Hello, everyone. We'll be starting in more 5 minutes. Thank you for joining. Please take this time to introduce yourself in the chat box and let us know about your educational background and research interest. Thank you. A very warm good morning, good afternoon, or good evening in whichever part of the world you are. After an incredible and insightful day one, I would like to welcome you all to day two of Omics Research Symposium. On day one, we heard from speakers on the academic front. We heard and discussed about vast range of topics from relevance and integration of omics into different types of studies, including COVID, genotype-phenotype correlations, machine learning to precision medicine, cancer systems biology, and different areas of analysis on Bioinfo platform, which included bulk and single-cell transcriptomics and metagenomic data analysis. Today is yet another exciting day as we will hear from researchers and professors from different industries and institutions. Today we'll hear about the application of omics research and relevance of the field of bioinformatics, from clinical informatics to the future of genomics. Not only this, but we'll also hear from industry researchers about the skill sets required for standing out in the industry. And finally, we'll hear from professors and principal investigators from National Association of Idea Principal Investigators and Louisiana State University about their research and how students can leverage the opportunities available. Now, without further ado, I would like to pass on the stage to Dr. Mohit Mazumdar, Director Business Development at Pine Biotech, to review the topics discussed on day one and introduce us all to the topics of day two. Over to you, Dr. Mohit.
Hello, everyone. Thank you, Sonalika. And also, if you can request the listeners to maybe share about themselves so that we can also get to know them and get to maybe work together someday. All right. So thank you, Sonalika, again. Hello, everyone. A warm welcome to all of you joining the second day of the Omics Research Symposium. To those who are joining for the first time, my name is Mohit Mazumdar, and I have a PhD in computational biology. And along with a great team, I'm heading the global business development and omics logic in Asia. In this introductory session, we will start with the recap of day one, and it will be also a great opportunity for those who could not join the sessions yesterday. Our team is already working on the session recordings, and they will be soon available and will be posted on our social media and YouTube channel. So stay tuned and also subscribe on the YouTube channel to get notification about the videos. So as Sonalika already described what we did, and I think we did a lot, as I was saying that there was so much to learn yesterday from the experts from academia and research institutes about bioinformatics and its data-driven science and machine learning, why machine learning is hyped, and we talked about and saw the application. So we learned about applications, we learned from case studies and discussions on how to be trained, get trained, and what is the future of this field. So in today's session, as I said, that we will start with a recap. To start with that, yesterday we had Ms. Sonalika, like always, started with the Omics Logic Global Portal and describing how we see bioinformatics, describing what are the different bioinformatics training challenges that are related to this tremendous growth in this field, enormous computational power requirements, lack of coding knowledge, faculties lacking the required facilities and the structure. So Sonalika explained how Omics Logic addresses these by standardization, organization, and systematization, applying Bloom's taxonomy levels, seeing biology as data science, and employing the hybrid learning approach. So followed by which the session by Sonalika was followed by the introduction of Asia Initiative, which I did, and the team responsible for daily interaction and development of resources and collaborations across Asia Pacific. And then we discussed about that with the help of our international collaborators, partners benefiting from this community. And we announced the partnership university program yesterday. The partnership university program is designed for the university partner to benefit from the collaborative association with Pine Biotech and their partners. So this special program is designed by utilizing the best of resources which are hosted at learnomicslogic.com, includes the coursework, project-based training for beginners, intermediate, and advanced users, helping them achieve the objectives of bioinformatics or Omics Logic data science and bioinformatics research training. The Omics Logic Partnership Program, which we described in much more detail, we described that it follows a unique curriculum designed for personalized training and research experience that enables the participants to develop critical scientific thinking, utilizing modern bioinformatics and data science practices on industry-relevant problems. So the Omics Logic program for university also follows a in-depth structure, which we went through by looking at one of the case studies where we describe the different stages and resources. The stages include basic training, example projects and coding, preparing to work on a project by practicing on various data sets and algorithms, and finally, to be able to work on your own research project or a faculty-driven project. So we piloted our Omics Logic training with several universities, and we showcased a recent program as a case study that clearly highlights the effectiveness and the impact of the program. Now, this case study that we highlighted yesterday was about the collaboration and the partnership.
Hello everyone. My name is Samantha.
Okay.
I'm a final year student.
So there's a video presentation which got played. But what I was saying that this collaboration and the partnership allowed students to work on their topics and interest and do real research on noble projects which were curated by mentors and experts. And also, as well as for the students, to improve their ranking in college and get an impressive resume that highlights their achievement.
Hello.
And for those students, such as here Aditya Bajpai, and who has a research interest to work on several projects and to contribute to the scientific community. And as well as by working on pressing challenges that had made the society dysfunctional, such as COVID-19, and had a huge impact on well-being. This was said also by Dr. Ishan Gupta, the next keynote speaker, who started describing about the collaboration with AIIMS New Delhi and his work, which explained like how what is the importance of Indian cohorts while studying the COVID-19 or while doing the COVID-19 research. And then COVID-19, which left so many people dead, had no molecular data, especially from the Indian cohorts. COVID patients have it got concluded to the fact that the COVID patients have lung microbiome dysbiosis. The lung microbiome distinguishes between patient types. So that's a way to distinguish between patient types, leading to the suggestion of patient stratification. So it was a very important topic and research that was highlighted by Dr. Ishan Gupta, followed by Dr. Suchi Nagar, who talked about various courses which are offered by the university, DY Patil University, and discussed genomics, transcriptomics, proteomics, metabolomics. What is omics, what are the branches of omics, and the process of bioinformatics analysis. So there was a lot of emphasis on the workflows, which showed the basic tools also like BLAST, Eugene, Mega, then finding out variation, then find what are the application in the of genomics and transcriptomics in different fields such as molecular medicine, microbial genomics, risk assessments, bioarchaeology, anthropology, human migration. So there's huge application that we learned, that from DNA identification to agriculture to livestock breeding and bioprocesses. So I think the application that we learned from this talk was really helpful, and also the pathways. I mean, it's very useful for the students to know about this and maybe choose a direction. And that was followed by a great talk again by Dr. Anshu Bharadwaj, who was talking about genomics and its application and the current places and the shape it is taking. So she highlighted about the Human Genome Project, the global interest to understand human diseases and understand the genomic variations. What are the single nucleotide polymorphisms? What were the candidate genomic approaches and genome-wide association studies, and the global effort to catalog the genomic variation. She also shared a lot of resources, which I thought would have been very much helpful. And as I said, that these sessions will also be posted online. Then we had that very interesting educational panel discussion where we discussed about the new educational policy in India and the actual need for data science education, and got to know about from the experts that how we can have data science education being imparted to students through basic training and a project-based learning. And then we discussed about the new education policy and how it encourages the hybrid learning, and what are the challenges that the universities faced while doing the hybrid learning, and what are the main taking away points, positive points, and how that has led to a hybrid setting of education, which is very effective. Then we discussed about some of the promising areas where bioinformatics is needed in Asia and India, and also with the respect of academic standpoints, academic experts. So job opportunities in academia, and what are the different ways to do and go about this, doing a master's or PhD, and all of those questions were answered and discussed in detail in this session. Followed by that, we had a very interesting poster presentation competition that covered omics and bioinformatics, artificial intelligence, machine learning, astrobiology, space omics, computational biology in pharmaceutical industry. So students were showcasing their work, and that were from various backgrounds and from various research areas. And that kind of concluded the first part of this day one. And then we had different panels. After the Asia panel, we moved to the African panel, and we learned from the panel where we learned about the Omics Logic African programs, partnerships and outcomes discussed by Ayanfe and Edikunle, where we learned about the bioinformatics tools and resources that have been provided, the data science for biologists program, and then discussion about discussion with the panel about all of these different topics, where we discussed about scaling bioinformatics education and training, and what are the future opportunities, and how bioinformatics is evolving in Africa, considering that it has such a diversity in its genomic makeup, considering the number of infectious diseases, and also the clinical information that we get from Africa is much more structured than in India. So these were some of the things that were highlighted. And then from the African panel, we moved to the US panel, where Mr. Brodsky, CEO, highlighted that what are the advances in enabling technologies, and then how bioinformatics and these events such as hackathon, and then the understanding of how bioinformatics is applied in wellness and health, then how to build a team, build an application, publish and share initiatives. He discussed about our initiatives in the US. He discussed about the virtual programs that we did, and also the research fellowship program, and our participation in EdTech Cup, leading to the semi-finalist position. And now we are moving towards that competition in this coming month. So that was the introduction and start to the US session, and we had follow-up important talks on scientific topics, which I think interested you, many of you a lot. And though for those who are joining for the second day, we have many interesting talks and industry input coming up, and I think it will be another very interesting and informative day for those who all are joining. So again, thank you all for joining today's session. We are going through a recap of what we did on day one, and after the Indian panel and after the African panel and the introduction to the US, Dr. Nikolai started, who is a senior bioinformatician at the Department of Biology, Lund University, Sweden, talked about the machine learning view of multi-omics data integration. He was discussing about the multi-omics integration across, across several features and several samples. He discussed about the hidden patterns in individual omics, and then he discussed about the curse of dimensionality, and also gave practical example from his lab, from his work, and to get an idea and work and know what they do. That was followed by another interesting talk, which was focused on bioinformatics application. So what we imagine for future is these dashboards, and these dashboards will give momentum and prove to be an excellent instruments for sharing biological data sets together with results. That is what Dr. Julia Panov concluded with. But she discussed about dashboard integration, a very important aspect of cloud computing, and doing this and developing these dashboards, which are like pipelines, which get answers, which give answers to specific biological problems. So she discussed about the easy data visualization and interaction, how it could be shared, allow easy navigation and analysis of different models, exploring ways to add code for R and Python. So these are some of the important aspects she discussed, and I think it was a great session, followed by the session by Elizaveta. She is the research associate at the Tauber Bioinformatics Research Center, and she discussed about the analysis of metagenomic data on T-Bioinfo platform, where she described about the KIME tool that we use to analyze the metagenomic data, and which is now available on T-Bioinfo platform, and gave an overview of metagenomics. How can you upload files? How can you run algorithms? And how can you design those pipelines and get outputs, and then understand by visualization, and again the incorporation of dashboards. So this was from Tauber group, describing about the advancements that have come up in the T-Bioinfo platform. Followed by that, we had the session which was about the forensic sciences, like how omics is now used in forensic sciences. So Dr. Sarah, she worked on interdisciplinary forensic sciences and integration of omics technology. She told us how the future research will be able to expand on these current studies, hoping for a leading to a wider implication of omics technology in forensic sciences, where she discussed about the application of genomics, transcriptomics, proteomics, and metabolomics for forensics, and what is STR profiling, which is the short tandem repeat profiling for identification of victims or perpetrators, and then what is forensic DNA phenotyping. And then there were a lot of keywords which would be really interesting for those who are interested in forensic sciences, and understanding that from soil microbiome to all this omics data, how it is helping in investigation of horrendous crimes and sometimes other applications as well. Now, after this initial discussion and all of the keynote speakers, we had Lucas, who is an undergraduate student of statistics and is a representative of Omics Logic team in Latin America, talked about his opening remarks. In his opening remarks, talked about bioinformatics education in Brazil, and introducing the bioinformatics training, and how we are working on different courses which are now translated in Portuguese and will be available and is available for students who are more comfortable in the native language. Followed by that introduction, we had the speaker, Dr. Luis Sokamoto, who talked about the precision medicine in Brazil. The reality and the trends and he's a precision medicine he's a pediatric oncologist, described about the pediatric cancer and frequency in children, the survival rate, the importance of childhood cancer for health system, and then what are the ways to do the molecular classification, what are the actionable targets, using evidence-based somatic variation, and also shared clinical cases. Then we had this session about system biology, as Sonalika was saying, that we had so many different sessions from different research areas, where Dr. Kasturi Kanan, who is an associate professor at Translational Molecular Pathology, discussed about bioinformatics and cancer system biology. So system biology initiatives for brain tumor, how there is a unified goal to create a computational framework to integrate all available patient-centric brain tumor recurrence data to generate these hypotheses, which is more unbiased in nature because of the entire incorporation of data for actionable target validation and for predicting diseases outcome for initial clinical trials. So all of this was concluded with this panel discussion, which kind of summarizes that how to apply these technologies to solve challenges, as we saw in the case of Sarah, and then from Nikolai, from physics to life sciences, how he got fascinated and by physicians and clinicians to mine more knowledge, and how Dr. Kasturi talked about his journey and he got curious and get self-taught bioinformatics, and talked about what are the skill sets that are required by students, coding databases, and all of the tools that we are seeing is something that Ilya discussed. And this was, again, one of the sessions where we concluded all of this, and we discussed about how interdisciplinary fields are integrated with multi-omic strategy to evolve students in bioinformatics and data science, which is the future of healthcare and well-being. So from this, this was the recap of day one, and I hope that this recap is helpful for those who could not join yesterday or who could not complete all the sessions, as we had 9 hours of intensive talks and sessions for students from our global community, joining from India, joining from Asia, joining from Africa, joining from Europe, and the US and Latin America. So I'm really excited that we had such a wonderful time yesterday, and now really hoping that after this quick recap, we are back with yet another day. And today we will hear about the perspectives of different researchers and industry experts from US and India and Africa. So today we kind of highlighted of some of our collaborators and industry collaborators. Many of them would be available today to help the viewers or the participants to learn about what are the advancements in the industry, how do you get into these different careers, and what are the opportunities, combining what we did with an industry, with an academia, and now learning from an industry perspective is a great way to go about the depth of where this is heading, and to be prepared. So today we will listen to our great panelists who are industry professionals from NEPI, as Sonalika has already described what we are going to do and gave an overview. So NEPI and its network universities and institutions, as well as industry partners such as Faba, and bioinformatics companies such as Elucidata, as well as NGS companies such as Prima's Life Sciences, to application-driven companies such as Oncophenomics and Equimind, and Mentis Hive, who are helping students to transition to industry, will be some of the speakers and keynote panelists. So let's quickly take a look at what we are expecting and what kind of sessions that we will be having. So we will have our first session in few minutes, perhaps, with Dr. Jeet Sarkar, who is a physician scientist and would be talking about clinical informatics and use cases and challenges. We are really looking forward to the session because you would be able to understand how clinical informatics is being applied in industry and what are the opportunities as well. Similarly, then we go from there to Dr. Shibi Chakrabarti Kanan, who is an entrepreneur as well as a scientist working on so many different cancer therapies, almost the cutting edge technologies that are being used worldwide, and working from India and bringing some of these technologies and research in India. So it will be something that will be a great session for all of those who are interested in cancer research. Then we have Dr. Nidhi Khurana from Faba, who are our very close collaborators in India, located in Hyderabad, and the network that Faba represents all the biotechnology industry and academia, where she will be talking about the different opportunities that Faba Academy is bringing and how our collaborative work is also helping a lot of students and also universities to develop these programs where they can, where they can kind of bridge this missing link between academia and industry. That would help actually a lot of students transition from academia to industry without the struggles that we usually see students and through our own experience, right? Now, after that, we have again a very interesting industry panel discussion, and that industry panel discussion will focus on some of these topics that you can see on the screen that would tell us about the future of Indian healthcare sector, how bioinformatics in industry is different from academia. Like the questions that you would want to ask is that how to prepare for a job that you have thought of, that you have dreamt of in a company without the stated experience. What are the key points that recruiters notice in a CV? So there are a lot of opportunities to learn from these academic experts and ask your questions. And we will be looking forward to answer some of your questions, as well as the questions that we have prepared for the panel discussion. That will be followed by some of these important skill sets. And we have been discussing about the participation of industry and the skill sets. So Dr. Rajavarman Kittu will be talking about that. And then we have session from Miss Padimoyo from Africa talking about the diplomas and CPGR grant program. And then we have the like more of specialized sessions, which is for a larger audience who are interested in protein-protein interaction and how these grafted peptides are used as a strategy. And then bioinformatics in beauty industry, a very inspiring kind of a talk, and we are looking forward to learn. Actually, it's this new for many of us, that how bioinformatics is used in beauty industry, what are the applications, and then learn from speakers from University of Malta about the life science research in space. So how life sciences research in space is being done through Dr. Joseph Borg, who is an associate professor and principal investigator, and also working on single cell. So then we have the prize distribution ceremony from the students who presented and submitted their posters. And then we have sessions by the NEPI founders and NEPI directors. So Dr. Gus Kasulas, director of the Division of Biotechnology and Molecular Medicine, Biomed, and a renowned professor of virology, will be talking about the NEPI IDEA program. And then we have Ilya Brodsky, who will be also describing our efforts with NEPI network and what are the different expectations and outcomes that we have seen. Then we have some specialized sessions again, which is to understanding of viruses and microbes. So these are for those students or for those who are interested in infectious disease research. Then we have machine learning, where all of this research is being utilized further to develop models, models that could be predictive in nature. Then we have from bench to a computer, where we are discussing about application in cancer therapy by Dr. Emmanuel M. Ruiz from the LSU School of Veterinary Medicine. And then we have the panel discussion for NEPI, which will conclude the day 2, and where we will learn about the different aspects of how to improve the training that is across 23 states and Puerto Rico. So I think we are really, really excited for all of these upcoming sessions. And I hope that all of you who have joined us today and will be joining us throughout this event will be able to learn and will be able to ask and will be able to experience the discussions and the talks from our respected experts and dignitaries. So without now wasting much of this by discussing what will happen, let's see what will happen. And with that, I will like to invite our first speaker, Dr. Jeet Sarkar, who is again passionate researcher who is mining and doing biomedical data analysis for better clinical outcome, and has been contributing by applying knowledge by doing translational research and finding solutions in multi-omics analysis, as well as learning and applying it for clinical informatics. So over to you, Dr. Jeet Sarkar.
Thanks, Mohit. So am I audible?
Yes, yes.
So I'll just share my screen. So is my screen visible?
Yes.
Okay. So thanks, Mohit, for the opportunity to talk on this topic. As far as we all know, clinical informatics is still in a very immature stage, and considering the deficiency of proper curated structured clinical data, and considering the impact of this particular field, it's still in a developing scenario. So I will just cover some of the use cases and some of the models which we have been developing, and which I have been developing to address various clinical problems. And I'd also brief in these three particular sections. So first, I will talk about the solution categories. So what are the possible categories in which the clinical use cases can be bucketed, followed by a solution life cycle, because clinical data is rapidly generated, and so the clinical models need to be rapidly updated, and so on. And lastly, I will just give a walkthrough of the use cases, a few use cases from clinical informatics. So starting with my background, so I have done my MBBS almost 6 years back. So by training, I am a clinician, followed by my expertise in data science and mostly building models for metabolic diseases. So that's how I wanted to combine these two areas to develop these clinical models. So to summarize, we can have we have been working. I have been working on 6 different type of solutions. So starting with the left one. So this is the broad category of predictive analytics. So this is more of a supervised modeling, where there are use cases like days to get hospitalized, length of stay prediction. It's a very important marker for the hospital performance, chronic disease risk prediction, and of course, COVID-19 prediction. So these are all predictive models where we have the data and we can do some predictions. Next comes the cluster identification, where you can identify disease subtypes. We can identify subspecialists. So today, things are going to a sub-specialization model, that we go for retina specialists, we go for strabismus specialists. So this is the domain of the ophthalmologists. So how we can identify these subspecialist doctors within a domain of ophthalmologists? These clusters can also be identified from clinical data. We can also identify disease clusters, whether what are the disease age which tend to co-occur in a certain population group of a population of a certain age. And we can also identify patient clusters. next we come to time series analysis. Now, time series is a very important part given the advent of these variables and Fitbits. So it's massive amount of data generated each and every second. And so because the vital parameters of human body are rapidly fluctuating, so we can predict the blood pressure or respiratory rate, or so using some time series models. So from the perspective of hospitals and healthcare providers, you can also predict footfall prediction. So because there are certain diseases which are acute and seasonal, so there is a trend and seasonal nature of patient coming to the OPDs and clinics. So in order to properly manage the burden of patients and the doctors, so we can also do footfall prediction. And lastly, vitals prediction in ICU, which is a major, because in the intensive care unit there are lots of parameters constantly measured almost every minute or even every hour. So those prediction also comes under the purview of time series analysis. Next, we come to image analysis. So it's a very much emerging field with last decade, where X-rays and CT scans and MRIs and even USG, these all things can be automated to a certain extent. Also for the pathological images, the tissue sections, this can also be automated to a certain extent. Another important part is the handwritten text analysis. This is very important for healthcare domain because healthcare still has massive amount of data in hard copy format. So digital formats you don't always get in countries like in Asian countries and in multiple countries where there has been the healthcare system hasn't yet been almost digitized. There are plenty of handwritten texts and scanned PDFs and all those things, and converting them to digital data and structuring them, cleaning them, then extracting medical entities to get insights. This is a very big domain in clinical informatics. And anything can be a combination of all these 5 categories, and it will be a customized model, and that model can also be developed. So clinical informatics covers this broad range of offerings of healthcare solutions. So how does a solution life cycle look like in clinical informatics? So suppose I work with clients where customers can come with some their data or their requirement. So there will be some introduction meeting for the use cases, and followed by that there will be some identification of possible model solutions for the use cases. Then will come the data exploratory data analysis, which is just to explore the data, how it looks, to provide some basic data insights, and then to propose the models. Now, this portion is the most vital part for from data EDA to the best model proposal. And here there is a constant iterative process to look for more suitable models which can best solve the clinical question. And lastly, and perhaps the most important part is the deployment and integration of these models to the clinical workflow, because the clinical workflow is already super burdened, be anywhere in the world. So integration of a new technology, of a new model to the existing clinical workflow, assigning nurses or physicians to use that clinical model for their clinical predictive purpose, or assigning a new person who will do this job on their part. This is a very important part, and that's where it also ensures a repeated iterative process where this usability is again and again tested unless the model is properly fit within the clinical workflow. So now the use cases. So just an overview. I will discuss today about, I just give some examples of supervised models, unsupervised models, graph network models, and some time series models. So we can just build some business insights. We can optimize the cost of expenses of the hospital. An evolving concept is value-based care. value-based care means where there is every clinical treatment or a clinical intervention is can be quantified as something valuable. So the healthcare system is gradually shifting towards value-based care. The insurance companies in the US and other countries, they are also focusing on value-based care. So so models can also be used for risk stratification, and then identifying high-risk patients, catering to them more frequently, so that the care is more of value, and that serves the purpose of the insurance companies also. Clinical insights, data labeling, model validation, risk stratification. So these are some use cases. Now, an important part is feature engineering, because in clinical domain you have lots of features and lots of different types of features. So it's a very mixed type of data set. So engineering those features to get proper features which can be fed to the model, and which will be having a high predictive value. Identifying those things and feature engineering is a very important part of clinical informatics. Model selection, translational insights is very important. And so predictive models, clustering models, OCR, where we can just scan and extract handwritten notes, image analysis, some use cases are here. And then finally, the application development, IoT management, wearable integration, and remote monitoring. These are also some possible use cases. So now, doing a deep dive, so I will just walk through the skeleton of a supervised model workflow, just to give an idea of how the thing can be designed. So on the left, you can see the data sources. So we are having electronic health records. We can have wireless wearables, medical devices, clinical notes, pharmacy records, then regular surveys, lab investigations. Now, these data types are of various different structures. So first of all, you have to structure it, and we have to make it machine readable. So these all deposited to the database. So you also have to connect all these things via suitable hardware, so that this data is ingested and properly curated on real time. Then comes the data preparation, and from there you can get certain variables. You can get socio-demographic variables. You can get comorbid conditions, lifestyle habits, self-taken surveys, real-time vitals. Now, each category will be having multiple features, and those will be fed to the training data set, will be a validation to develop the model. And now this model can be used to predict, as I already told, length of stay prediction, chronic disease risk prediction, hospital readmission prediction, so predicting post-op complications, vital parameter prediction, and all those things. And the model can be customized and updated as per the clinical need and other things. And we can also do some sort of feature importance coding, where we can identify that these 3 features or these 4 features are the most important for predicting length of stay. So maybe these 3 features should be the more frequently monitored features for this particular set of patients. So this sort of model can help us predict these sort of clinical outcomes. So for the unsupervised model workflow, I just presented one use case. So suppose we had data of ophthalmologists, and they have been treating particular diseases. Now all the diseases have some standard ICD codes. So if a ophthalmologist treats, suppose a disease code like 6 G138, so number of cases of G138 he or she has treated is in a tabular format. Now these all these ophthalmologists will be clustered in an unsupervised manner. And after clustering, when we saw the visualization, what we found is, so there were certain, so these, there was a difference in the clusters. Suppose these blue dots, these were mainly those ophthalmologists who were catering ocular, muscles, and eye movement refraction diseases. This green cluster were those who were providing moldy choroid and retinal diseases. And we found that these green physicians were mainly retina specialists, and these blue one were mainly pediatric ophthalmology and strabismus specialists. So, so you can already identify these subspecialists who are particularly expert in a subdomain of a particular ophthalmological domain. Now, this can be of multiple help. So maybe in this case, so because if so all of you, I guess, understand a PCA. So a PCA means these blue and these red axis, they are more or less at a 90-degree. So blue people don't contribute to green one. So that means retina specialists don't cater service for pediatric ophthalmology and strabismus, and the also the other way around. So if I can identify a specialist who is having both these skills, then that is a very common, very rare combination, and those persons can be a sort of a common door to these subspecialists. So these sorts of business insights and healthcare insights can also be extracted from these supervised, unsupervised model workflows. So next, I was talking about the graph network models. So suppose in a population, we know that people generally in the geriatric population, they suffer from multiple diseases. So the person has diabetes as well as hypertension, as well as dyslipidemia or hypothyroid in these cases. So if we could identify that at which age group and given a gender, what are the most common co-occurring diseases for that particular group. If we could find that, then given a new person comes, we can just provide a much more personalized screening program that, so you fall in the category of 60 and you are a male, and you are having diabetes. So in that case, you may be having hypothyroid and hypertension and eye disease. So you should be screened for these 3 or 4 diseases. So to achieve this, what we did, we took the disease conditions of all patients, and we calculated the frequencies. So the nodes are the frequencies, and if the 2 nodes are connected, that means these 2 diseases tend to occur more frequently in the population. So suppose one person is diagnosed with long-term current use of aspirin. In that case, there is a good probability of that person suffering from hyperlipidemia, suffering from insomnia, suffering from primary osteoarthritis, suffering from vitamin D deficiency. So this person should be screened for this surrounding 5 diseases. Suppose another person comes with another particular disease. So for that, the set of nodes will be different. So this will help us to cater a much more precise and personalized screening, health screening workflow from person to person. So in this case, we just designed it. So just to showcase this thing. So these 7 diseases are closely related to the long-term use of aspirin. So any person coming with this diagnosis should also be screened for these 5 conditions. So there are already some screening protocols in place where one person having over 40 should be diagnosed, should be screened for this condition, but this will make things much more precise and personalized. Lastly, the time series model, which is perhaps the most important, or because it's a very huge data. So here you can see this is the respiratory rate of one person fluctuating with time. So every 2 seconds, the reading has been taken. Now you can see in this particular condition, there is a huge increase of the respiratory rate compared to the other findings. So here there must have some abnormality or some exercise outburst or anything. So if we could predict this hyper value from the preceding values, then we could have prevented or alerted the patient that you might be having an increased respiratory rate in the coming 2 or 3 minutes. So be cautious. So these sorts of things are also right now a bit more hypothetical, and these are being tried by people to model, but this is how the use case looks like. So here we can apply something called change point algorithm. So what it does is it takes the entire stretch and breaks the stretch into certain slices, and for this slice the mean standard deviation is different from this slice having a different mean and standard deviation. So we can identify these abnormal slice where the mean and SD is significantly different from the preceding as well as the succeeding areas. And in this case, there's one problem is data has to be real-time synchronized and analyzed. So that is a different problem. It's more of an engineering problem. And in that case, we can also set parameters and rules that if the value is over this certain thing, then give an alert. And we can also build data-driven insights that if the data tells that the person is going to have an increased respiratory rate or heartbeat, then please alert the patient and the surrounding physician, doctor, ambulance, and all those things. So just lastly, coming to the end of my talk, I will just briefly give overview of how end-to-end workflow for chronic disease may look like. So we can just get the data acquisition from the wearables and medical devices. We can do the data prep, feature engineering, model building, clinical decision support, which will be validated with the physicians. And lastly, we can use that thing to produce automated alerts so as to help predict abnormal events, help recognize skipped medicine doses. So if somebody is having a abnormally increasing blood pressure, then we should check that whether the person has skipped his or his medicine for the last 1 week, or what is the compliance. And that can also be put in as an alert to the person and to the concerned caregiver. For mental health, it's a very important topic coming up, and predicting suicides, predicting depression attacks and panic attacks, manic phase prediction in bipolar disorder. And for this, the data source is much more comprehensive: clinical data, genomic data, vocal expression data, because the vocal expression, how the person is talking over the phone, that changes, and the facial expression changes, social media and regular survey. All these things can be structured and fed into the model, and accordingly we can do prediction. It's definitely very critical. There are certain ethical issues and lots of legal issues, but this is the one direction in which mental health is also going to proceed towards. So I'll just end my talk here, and I'll just take any questions for the last 5 or 7 minutes. Thank you.
Thank you so much, Dr. Jeet, for this wonderful, wonderful talk, where we learned about a lot of application or driven, how the industry is looking at the problems and finding solutions. What the students have learned, I guess, is also the same. And I think that we have some questions. So Sonalika, are there any questions from the participants? Or Shri Gauri? I think some Sonalika is putting it in the chat. So let me read it out for you, Dr. Sarkar. So what are the possible career options for people pursuing bioinformatics and clinical informatics?
Okay. So bioinformatics mainly consists of till now genomics, transcriptomics, proteomics, metabolomics, and maybe metagenomics. So a lot of these these omics approaches and the data generated from them, this is going to be used for coupled with clinical trial data and with clinical prediction. So the thing is not so mature right now, because as I told, there is a certain lack of bridge between healthcare providers and bioinformatics. But this is where the so there are companies who are trying to integrate omics data with clinical trial and clinical outcome data, and those are really big short things which are coming up. So if somebody can have the skill set of bioinformatics and is having the orientation to apply that in clinical care and clinical trial data, that would be a great career in the coming days. Almost a rare skill, I would tell.
All right, that's I think that is helpful. Next, there is a question by Dr. Chitrabrata. He's asking that graph theory can identify the group of closely related diseases. Can we predict which will come earlier?
Okay, so I guess graph theory. So in graph networks, there are also temporal graph networks. So if we can build temporal graph networks, in that case, we can do predict. But I will tell one thing, the major problem is with data. So, so suppose a person is having 4 diseases. So hypertension, diabetes, and hypothyroid and dyslipidemia. So what is the particular chronology of this disease is something very tough to decipher. The person had gone for a clinical visit, got tested with diabetes, and then also found that he is suffering from hypertension. But whether hypertension came before or diabetes, that is a very difficult thing to comment for a real world patient. But this should be something which should also be tried. And given we can have longitudinal data from a certain age where disease doesn't set in and we are having annual checkups, then we can have those data, and temporal graph networks can really identify this sequence of diseases. Yes.
All right. Thank you. Thank you so much for that answer. One last question. Risi M. Vesonga, she's asking, thank you, Doc, for a great presentation. Could you shed some light on your work with metabolic disorders?
Okay, so my work was mainly on type 2 diabetes. So now, as you know, type 2 diabetes is known to be a obesity-driven disease, that people who are obese tend to have diabetes more. But as you may know, that there are certain studies coming up which tell that type 2 diabetes can also occur in so-called non-obese persons. Now, those non-obese persons may have some increased visceral fat, lower muscle mass. That's a different discussion. But why do these non-obese type 2 persons also become diabetic? Because these persons, they contribute to over 50% of the Indian population. So in India, over 50% of type 2 diabetes patients are having a low body mass index, a body mass index less than 25. So my study was mainly to explore the underlying pathophysiology, and I covered, so I studied the adipose tissue of non-obese and obese diabetic persons biologically, and using cell biology, and I studied epidemiological data sets and also clinical records. So that there are multiple levels of the entire thesis, and you can find it on ResearchGate or just by searching me or on Google Scholar citation, and you can just have a good understanding.
All right. Okay, thank you so much, Dr. Sarkar, for this wonderful presentation, as well as for answering the questions. And as we are on a short time, so we will be able to share with you some more questions as they come over the email. And I hope that you will get the opportunity to respond to some of those.
Sure. Thanks.
Thank you. Thank you so much again. Now, moving to the next session, I think we learned about some of the aspects that Dr. Sarkar just discussed. We learned about finding meaning out of biomedical data and leveraging them for better clinical outcomes. As you learned about the omics data, either genomics, transcriptomics, proteomics, such data types, and how can they be used for different clinical outcomes. Now, I would want to go further with this, and now we have the next session, which focused on cancer research. So we have Dr. Shibi Kanan along with us, Dr. Shibi Chakravarti Kanan, who is the CEO of Informatics. Just a second. Sorry about the slide deck. Yeah. So let me introduce Dr. Kanan first, so that we can learn what he's going to talk about. So Dr. Kanan is an entrepreneur and a physician scientist, and also a computational biologist. And we have been fortunate enough to have several of these discussions with Dr. Kanan and learned so much about translational cancer research and its application in India. And I think it's a great opportunity for the audience and for everyone who is here today to learn from his experience, where his focus of work was cancer genomics and on utilizing different sequencing platforms for immuno-oncology, for drug discovery and development, that led to cancer immunotherapy, developing those protocols for cancer immunotherapy. And there are so many different keywords that are associated with what is going on with the with precision oncology and all of that. So it's really, we are very excited to learn by you, Dr. Shibikan Chakrabarti. And anything, anywhere, anyone getting ready for the future of genomics, over to you, sir.
Yeah, thank you. So, can you guys see my screen?
Yes, yes.
Okay, thanks Mohit for the wonderful introduction. So basically, anything, anyone, anywhere is basically from Nanopore's official website. So you can see here. So let me go to the beginning. I also have a presentation. So I wanted this to be a more interactive session. I don't want to bore the people, So if you are an NGS, next generation sequencing, whether you are on the wet lab side or on the bioinformatics side, when people say NGS, what do you think about? You always think about Illumina and ion torrent and other short read technologies, but the world is changing. The world of genomics is changing at a very fast pace. So we started adopting nanopore technology around 2017, 2018, when the technology was still at a very early stage. And a lot of people used to say, oh, nanopore is not good, it's only for research. It doesn't have, the accuracy does not meet the level of accuracy of Illumina, but that's not true anymore. So it's a fast evolving and a fast developing technology. Maybe I'll make the screen a little bit bigger so that you guys can see. Yeah. So I want to give a taste of what's coming in the near future. So, and I'm not alone. I am part of a huge community of nanopore users. More than 30,000 laboratories across the world have adopted nanopore, primarily for studying microbiome, bacteria, yeast, fungi, virus. And very recently, during the COVID pandemic, nanopore came as a very big solution for real-time genomic surveillance of how the COVID virus is mutating very fast and spreading all over the world. And all the countries started using. So the real adoption of nanopore technology happened during the pandemic. So that's something Illumina could not have done that because of it's a capex heavy equipment. You need to invest millions of dollars to have an NGS facility. But nanopore is like they have different small instruments to big instruments. So here they show, let me just scroll a little bit. So here you can see the range starting from a flongle or smidgion all the way to the minions, the gridion, and then we have the prometheon. So they have the complete range. And nanopore is one technology. You're not tied to the lab, so you can literally take it wherever you go. So people have done it, even done sequencing in the space, in a space station, and they've done in remote corners of the world. So there's a lot of information available on the website. So as a broad spectrum, what is the main use of this technology? Just like any other NGS technology, it has a very multiple use cases. Most common, how nanopore became very popular is metagenomics, especially when they have this what's in my pot analysis. I give you a tube, I go to collect some environmental samples or some bacterial samples or I take a sample from poultry farm, or I go to Himalayas and take a sample of the water, right? So what's in my pot? Can you tell me what's inside that tube? The colorless liquids. That's the metagenomics is the most important and most popular use case. And then of course there's agrigenomics. And then because of the long reads, you're able to have N50, the contigs. We have like, we're not limited by the lens. Whereas in Illumina, there's always a size limit. It's like 150 to 300 base pairs. So here there's no limit. So you can have very, very long reads. We have seen in our lab, minimum of 1.5 kb to 40 kb fragments with very good overlaps. So it becomes very easy when you want to do a de novo sequencing and align and map the genome. And of course, the most common use case is studying the bacteria, the virus. And now recently, a lot of work is being done on human genome. That's where we come in. So we primarily focus on cancer diagnostics. And we do something called a comprehensive genomic profiling in our lab. So we also look at the tissue biopsy and we also do the liquid biopsy. So nanopore technology has been used since the beginning in our lab. Earlier, we used to do some comparisons with Illumina data and ion torrent data. But now the accuracy has improved a lot. So we no longer do these orthogonal validations. We just go direct with nanopore because we are confident we are able to find the mutations very easily. There are several unique advantages with nanopore. Of course, you can see there are so many long-read technologies. There is PacBio and so many other guys like HiC, BioNano. Almost every week we see a new company pops up, right? So we have Singular Genomics and so many new companies coming into the space. It's very popular. And then we have the MGI, which is a clone of Illumina, but they do it in a different way. And they do have some unique advantages. It's more affordable compared to Illumina. But generally, entire NGS world is separated into short read technologies and long read technologies. Among the long read technologies, PacBio and nanopore are the leaders. Unfortunately, PacBio is too expensive. So the per sample cost is as high as $300. Sometimes if you want 100 GB of coverage, it's more than $3,000. For a small laboratory like us and most developing countries, this pricing is not affordable. Unless you are a university project, you have some grant money, right? So for clinical use, it's not making sense. So we have been very happy with nanopore. The per GB cost is as low as $30. So it's a PacBio is $300, here's $30. It's literally 1:10 kind of ratio. So I don't think anybody can beat nanopore on the pricing side. The other major important advantage in nanopore is it's a real-time sequencing. So in Illumina, it's like a 1 week long pipeline, both the wet lab and bioinformatics, 3 days for the wet lab minimum, and another 2-3 days for the bioinformatics. So if I get a sample on a Monday, I can give a report on a Friday or a Saturday. Sometimes, some holidays comes or something happens, some delays happen. Sometimes it may take longer than a week, 7 to 10 days. That's why most NGS laboratories and genomic service providers, they don't give a turnaround time less than 3 weeks. They give typically 3 to 4 weeks. But in nanopore, I can do it the same day. I start from the morning, do the DNA RNA isolation, do the library prep in the afternoon. By evening, I can load the sample. So this picture, you can see the MinION device, which is a most affordable device. Anybody interested in genomics, they can start buying this. The instrument comes free of cost. You're basically paying for the flow cells and the reagents. The startup cost is only like $5,000. It's very low economy. Anybody can start doing sequence. Once it's anything, anywhere, and anyone. You don't have to be an NGS expert. And it's kind of, the kits comes with excellent protocols, both on the wet lab side, also on the bioinformatics side. They do provide a software, online cloud-based software for the data analysis, especially if you're doing metagenomics projects. It's one of the easiest to start. Start with the rapid barcoding kit, and you can start doing metagenomics in the lab every day. It's very easy to adopt. So we have made a mission to help everybody to adopt nanopore technologies free of cost, right? So if you are interested, call me. I will help you set up the lab. I'll help you with onboarding. So we have a team of people who can support you. So we don't want anything in return. We just want people to start using nanopore technology. Forget Illumina. Those are the dinosaurs of the past, right? Let's move on to the future. That's why I said getting ready for the future of genomics. The way we see it, the next 10 years, long-read technologies are here to stay. And more competition comes, and the price is going to come down, and it could be as low as $9 per GB. So in NGS, you always go with the per GP cost, right? So it's making a lot of economical sense, especially for emerging markets like India, Africa, South America, Asia Pacific. Everywhere you can do it. And again, the time taken is just 24 hours to 48 hours. Maximum you can do. You can completely max out the flow cell in 72 hours. So that's a huge advantage in Nanopore. The real time, as soon as the data is being captured, it's uploaded to the server, and you can continuously monitor the data. Within 6 hours, for example, COVID virus, we can do it within 6 hours. We can actually tell you what variant it is. So they have a very nice protocol. Of course, you'll have data in the first 30 minutes itself, but we wait for 4 to 6 hours to get some confidence on the variant. Is it a Delta variant or Omicron? Even if it's a Omicron, what's a sub-variant? So we can do all that. So we have a NABL lab. We do COVID testing also. And we also do for research purposes under ICMR guidelines. We also look for variants because we have a mRNA vaccine project where we use Nanopore extensively. So without Nanopore, I don't know, can we even do this kind of work? So kind of the time turnaround time and the time it takes to develop the data. So it's a very simple prepare your samples, then sequence and analyze. So it's 3-step process. Let me quickly walk you through the 3 main products. So you can see MinION. It's starting from $1,000, so it's a starter packs, usually $5,000 when you add the kits and the agents. And you can see GridION starting from 50K. And Promethean, they have two kind of offers. It's actually a 2TK instrument, but you can go for the P2, which is more affordable. So if you are a university lab or a commercial lab and you want best of both worlds, there's a difference between the MinION flow cells and the Promethean flow cells. So I would recommend go with the GridION. So GridION gives you 5 flow cells. And the P2, so you have 2 Prometheans. So you will have the flexibility of using both MinION flow cells and the Promethean flow cells, depending on what type of data your customer wants, right? So, and here you can see in the product specifications, what can we do with Nanopore? Everything you can do in Illumina, you can do with Nanopore. It's not like, only Illumina is good at SNPs and genotyping. We can do SNP detection in Nanopore. In fact, it's a lot better. Homopolymer detection is also very high. And recently, people have done benchmarking studies. There have been Nature publications, many publications. Illumina versus Nanopore versus PacBio. They have done the neck-to-neck comparisons with known samples, and Nanopore has come out as a clear winner. There's a page, I have not opened that. There's a page that talks about the accuracy and base level accuracy, and also at the genome level and the chromosome level accuracies. And Nanopore always comes at a huge winner and at a very affordable cost. So this is like a superior technology at a more affordable cost. And another big advantage in Nanopore is the methylation. So no other technology gives you the advantage of both genetics and epigenetics on the same run, on the same experiment. Imagine if I have to do Illumina NGS, I have to, for example, Illumina is a company called Grail. They have done liquid biopsy for multi-cancer early detection. They have to split the DNA sample into 2 vials. One vial goes for normal variant calling pipeline DNA mutations. Another vial goes for the epigenetics, the bisulfite sequencing. So they have to split it. Now, what is the ratio, the proportions? Did you split it exactly equally 50-50, right? So the sampling bias comes in the picture. But in Nanopore, you don't have to split it. The entire sample runs through the same native barcoding protocol, and you get this raw data. The raw data can be analyzed in 2 different pipelines. So splitting happens at the bioinformatics level, not at the wet lab level. So there is no sampling bias. The same sample, you get base modification, especially the methylation. So that is the one of the main reasons many people are now switching over from Illumina, from bisulfite sequencing, switching over to Nanopore is because of the methylation detection. And another huge advantage, especially for cancer research, we not only look for small mutations, we look for big mutations, anything more than 50 base pair, called the structural variants, SV detection, and also haplotype phasing. So this is something not many people know this. People report EGFR mutations, for example, the cis and trans. So it depends on, let's say I have 2 types of mutations, the L858 or sensitivity causing mutation, or T79TM, which is a resistance causing mutation. If these 2 mutations come on the same strand of the DNA, then the resistance biomarker overtakes the sensitivity biomarker. But if they come on the opposite strands, then the patient is still sensitive to TKI. So this has a direct implications in tyrosine kinase inhibitors. So we call it companion diagnostics, like drugs like erlotinib, gefitinib, and all these TKIs, tyrosine kinase inhibitors, which target the EGFR, KRAS, or BRAF. Very important to know the cis and trans. How can we do that? You have to do haplotype phasing. You have to know if my cluster of mutations, are they coming on the same strand of DNA, or are they coming at the opposite strands of DNA? So haplotype phasing is coming up in a big way in cancer research. And another hot area is the transplantation medicine. So where you have to do HLA typing. If you're going for bone marrow transplantation, so cancer patients, leukemia, lymphoma, so many patients, they go for BMT, also hematopoietic stem cell transplantation. So typically they go with relatives, some matched donors. Sometimes today donors are very difficult to get. You don't have siblings, you don't have any family members to give you bone marrow. Then you go for matched unrelated donors, MUD. In a MUD case, which is more common nowadays, what we do is something called haploidentical. That means 100% is not a match, but 50% match. But the 50% match is on the same strand of DNA, so haploidentical. So recently there was a Nature publication where they showed there are 21 doppelgangers for every person walking on this planet Earth. 21 people in this planet who are exactly like you. They also look like you. They also have a similar genome, the facial features and everything. So it's very interesting paper. So finding an unrelated, unmatched donor at haploidentical is becoming more and more easier nowadays. So this has truly revolutionized transplantation medicine. That's where HLA typing, an entire HLA alleles, the MHC class 1, class 2 loci, very easy to do on the Nanopore platform. And again, in agrigenomics and plant genomics, wherever you're working on a new species, you don't have a reference genome. And now we have the pan-human genome, pan-genome project. And we also have the org.1 and so many other projects. So assembly, de novo assembly, right? So this is the key. So highly recommend, go to the Nanopore website, nanoporetech.com, and click on the resource center on the resources. So here you will find all the latest videos and stuff. And let me quickly walk you through the community. So anybody can have a free account. And here you can see I'm logged in, I'm in the dashboard. And you can see there's a huge community, active discussions are going on. Also, you have all the new releases, software downloads, and you can check the protocols. It's very easy to getting started with Nanopore. You have all the protocols here, and for different kids, it's very easy. It's a lot more simpler than Illumina. If you're coming from Illumina, it'll be a delightful experience working with Nanopore. And they have different protocols. For example, extraction protocols. Are you starting from DNA? Are you starting from RNA? What is your material? Like that, they make it very easy. There's also a protocol builder, which can basically walk you through the different sample types and what type of validations you want to do. So you can see tissue, cell culture, fluids, whole organism, agarose plug, environmental, FFPs. You're not limit. There's no limit. Any sample type. And then depending on the organism, so they have different protocols. They want one for mammalian, one for plant, like that. So pick the protocol that you need and follow that to the line. Don't change anything. It is guaranteed to work. That much I can tell you. I'll just quickly show you this video. So let me go a little bit back. So here, so this is the flow cell. Okay, so you see this small area here. This is where we drop the sample. So it's very simple. And we just, there's a small, we have to open that spot on that lid, and then put drop, and then it goes inside. So it's basically like electrophoretic chamber. So there is an upper chamber and lower chamber, and then there's an ASIC chip on which the nanopores are being embedded. It's basically a protein nanopore. And once the DNA goes inside, it's basically threading through the nanopore. So then the ASICs are reading the electrical signals, which the software will interpret. That's where the base calling comes. So this is where real-time base calling can happen. So you can monitor the data in real time. And once you exhaust the DNA in the top, you can basically stop it. And this is as part of the library, this is what you're doing. So this is your DNA sample. And what you're doing is basically you're attaching this motor protein on the adapter sequence. And here you can see these are the A6 chambers. These round things are the membranes on which the nanopores are embedded. So there's a 2048 membrane wells, each containing at least one. Sometimes it may contain more than one. But typically now the optimization has happened a lot. You'll only find one nanopore per. Sometimes the nanopore is not fully formed. Maybe it's broken. You do find a few channels. By the time you receive, a few channels, the protein has denatured or it has folded, it's not working. But majority of them, 70–80%, they still work. So even though you're guaranteed 2048 pores, by the time you receive it during the shipping and logistics, you may not get 2048. So they say minimum 1500 or 1700 is covered under warranty. If it is lower than that, then they will replace, they'll give you a brand new flow cell, so nobody gets 100% capacity. That's something that you need to consider, Yeah. Okay, so this is the important step. So you can see this tether is going to catch this, right? Okay, so this is how it works. So basically, when the DNA is being threaded, there is a disruption in the ionic current, which is detected. Earlier, I used to think it is threading one base at a time, but that's not correct. It's actually threading 4 to 5 bases at a time. And different pores have different speed limits. And depending on the speed, the faster it goes, the lesser the accuracy. The slower it goes, the higher the accuracy. So it all depends on, you want a structural variance, a bigger. I'm not caring about SNPs or single nucleotides. I only look at large things. Then you can go faster. But if you're doing variant detection, you are looking at single nucleotide, base level things. Then you have to go slower. Okay, so I think you got the picture. So let me open the presentation. I'll just quickly show you. So why we call it as the future of genomics, right? Illumina has been there for a long time. Illumina and other guys, Ion Torrent, they haven't done much innovations. It's the same technology they had 20 years ago. They still keep selling the same things again and again. But Nanopore comes up with a lot of new information almost every year. So they have this huge conference called London Calling every year. So these slides are taken from their plenary lectures from the London Calling 2022. I'll just pick some important slides in that. So the first is, you can look at the evolution of the technology from 2017, 2018. So they had the mini-ions, the grid-ions, and then they launched the flongles because some people said mini-ion is too expensive for smaller laboratories. Then they came up with the smaller ones, which only has 126 channels. The mini-ion has 512 times 4, so you have 2048. And the Promethean has 3000. So Promethean has a bigger capacity. If you're doing a plant genomics project, de novo sequencing, you are depending on the size of your samples, how big the genome is, how much coverage. If you want 100 GB to 300 GB kind of coverage, then you have to go with the Promethean. And again, the accuracy has improved a lot since 2017, 2018. Now in 2022, we are already at a very advanced level that every technology has its own product development life cycle, and it's already at a very, very advanced stage. I'm not sure how much more new things they can introduce. They already introduced so much new things. This is at the pinnacle of it's everything that we have hoped for. And earlier it was called R9.4. Now it's 10.4. The R10 series has clearly have a huge performance update. How? Because mainly you can see in this graph. So you have this different the bases per second BPS. So it used to be 520, then 400, then 216. So the slower it goes, the more accuracy the software is able to recognize. So basically, it is artificial intelligence running behind the scenes. So we used to have AlgoCore in the beginning, then we have Guppy. So today, Guppy, there are different versions. So always check the version, install the latest Guppy when you analyze the data. And we highly recommend the Minion MK1C, which comes with a screen. It has an onboard GPU and 1 terabyte storage. The regular Minions, they have a temperature problem. If you keep it in a normal room, I had this problem. We have like 5-6 Minions lying around in the lab. So there's too much heat creates a problem, especially in India. So I recommend go with the Minion MK1C, which is similar costing, no big difference. Plus, you have the advantage of a very good thermostat. It controls the temperature very well. The standalone menus don't have that option. Then if you have the budget, go for a grid, right? So that's something. And now the good news is it's backwards compatible. So if you have the older R9 flow cells where accuracy is not an issue for your data, you can still work with that. And they're also forwards compatible. So you can basically use any kind of flow cells. And the different ways to run on the Promethean, so you can run single samples, you can run, you don't have to, if you have a P24 or P48, no need to put all 24 at the same time. You can just run 2 or 3 Promethean flow cells at a time. You have a lot of flexibility, especially if you're doing human genome. We recommend minimum like 2 or 3, maybe up to 5. 5 is a lot, but 2 to 3 is fine. People have done very promising, like here you can see Matt Lewis has more than 170 GB as data has been generated from the new Kit 14, which is currently running at 400 BPS, right? So the new Kit 14 chemistry, the Kit 12 came, then the Kit 14 has come. So it's an early access. So the existing current users can get access to this. People who are interested in the accuracy. And they've done a lot of improvements on the motor adapters and also the chemistry. That's what's happening in the Kit 14. So this is currently now available. So initially they launch, they'll say it is early access, but after wait for a couple of months, it will be available in the regular store. So I highly recommend, if you're looking for accuracy, go for Kit 14. If you're looking at structural variants and large chromosomal kind of things or metagenomics, but you don't need Kit 14. You can go with the Kit 12 or the older kits also, not a problem. The more interesting part here is if you have the Gridiron or Promethean, the bigger instruments. Now I have a tuning. I can increase the speed, I can decrease the speed, how fast the DNA is being threaded. This is not possible in any other technology. You'll only see this in nanopore, and I can literally control the accuracy. Here they have shown the data for simplex accuracy and the duplex accuracy. So it's a tunable run settings. This is like like a science fiction, like a dream come true. Depending on the kind of and then there's I forgot to keep on slide. There's another very interesting feature in nanopore called adaptive sampling. Today, if you do like Illumina, everything is amplicons. You need to design primers. You have to make the PCR products. You have to amplify certain regions of the DNA, right? And then you have to sequence it. But adaptive sequencing is like no need for amplicons, nothing. In the software, we program, these are the regions. Let's say I'm working on chromosome 8. Chromosome 8 is my interest. I'll give the reference genome for chromosome 8 as one of the approved file formats. So I'm doing a whole genome project. I'm literally putting an entire whole genome of the human DNA in the sample. But when the DNA is being threaded, the software will identify, okay, this does not belong to chromosome 8. It will reject it. It will spit it out. It's called adaptive sampling. And when the next strand goes inside, it says, oh, this looks like chromosome 8. I'm going to continue sequencing. So I don't need amplicons. Why do you need amplicons? Just go with adaptive sampling. The only challenge there is you will not get hundreds of GBs of data because you are getting rid of 90% of the unwanted DNA. You're focusing on the highly focused DNA. But then you are looking at the DNA in the native conformation with the base modifications. You are not changing it. Same with RNA. Another huge plus point in nanopore is the direct RNA sequence. So there's so many features in nanopore. Once you start learning about that, then say, man, why are we still doing Illumina or any of the short read technologies? Nanopore, once you go into nanopore world, you never come back. So again, for a long time, nanopore used to be just a single reads. So now they're introducing the duplex feature. The kits are not yet available. It's coming soon. So this is how the duplex read works. So basically what it does, it threads one half of the DNA first, but then there's a connector link, and then it goes to the other end. Same like how Illumina works, but they have done some modifications in that to avoid the IP infringement with the Illumina. And now we have this 3 major pores. So we have 3 major flow cells. So we have the R9 series, we have the R10 series, and then the new RX series, the R11, 12, like that. Something will come in the new series. Currently, the R9s and R10s are available, 9.4.1 and 10.4.1. We have completely stopped using 9 series. We are now using the 10.4.1. This is giving very good accuracy for us. And we are able to get, like a complete whole genome coverage at a 3x to 30x. And adaptive sampling of those 500 genes, something like foundation medicine or garden health, the comprehensive genomics. There are 500 genes which are important in cancer. So we are doing adaptive sampling for those 500 genes we focus on that. And this is some data they presented about homopolymer accuracy. It's very nice. And also, like the raw read accuracy, theoretical maximum output in 72 hours, how much you can generate, And then where are we now? So we are now with Remora. So Remora is the latest for the methylation. And here are some examples. You can see the precision and accuracy. It's giving the best bang for your buck compared to bisulfate sequencing. So if you are into methylation, Nanopore is the best option for you right now. You can do both genomics and epigenomics. I think that's the last slide. Yeah. Yes. So we'll take questions now.
Thank you, Dr. Shivi, for this wonderful overview and understanding of the nanopore technology. And there was also a comparison. So for our users and viewers, I think many of our users and viewers have been students and faculty. And for students, I think one of the things that have been a gap, or perhaps many of the biotechnologists and biochemistry students would want to know much about how the data is generated. So I'm just summarizing that this has been really helpful for those who would want to know how the data is generated and what is the industry standards, because many times, especially with faculties who are also working and want to conduct experiments, are kind of outsourcing this to the industry, this sequencing work to the industry, and then they're getting reports, and then they are learning about what is the possible insights that they can gain from the data. However, having an in-depth understanding from this session is going to be very much helpful in designing the experiments, and even before designing, hypothesizing the experiments. So thank you so much, Dr. Kanan, for this insightful talk. And we started from clinical informatics by Dr. Jeet Sarkar, and then we learned about how the data is being generated, and then what we want to know also. There are some questions, so let me read that out to you. So one question that I'll read now is, keeping some of the limitations of nanopore technology in mind, in your perspective, what are some of the ways the technology can be further improved in the future? So where do you see that the future with nanopore technology, how they can evolve further from this?
Yes. So like I said, the base calling algorithm can keep on improving. So one of the huge advantages in nanopore is we have done some sequencing way back in 2018. That raw data is still lying around. Every time they release a new version of Guppy and the new version of all the software, Medaka, Remora, and everything, I can go back and reanalyze the raw data. I can retrieve more information. So that's the beauty of nanopore. Because the base calling algorithm is improving, it runs on artificial intelligence behind the scenes. The more training data set you use to train this algorithm, the better it becomes, right? It also opens up the possibility. Let's say you are working on a very unique kind of project, which nobody has done. Everybody is working on model organisms. Let's say your organism, let's say you are a diabetes professional working on Gila monster. We don't have a reference genome for Gila monster or the Komodo dragon, for example, right? But the saliva has the diabetes treatment. So you're working on that. You don't have a reference genome. So you're completely doing something from out of the scratch. So take monkeypox, for example, right? And nobody has built a monkeypox reference genome. And you are the first guy doing it. So then you can basically train your own algorithm. So the software gives you that ability to train based on your own training data set. So you have gigabytes of data lying around in your software database, in your hard disk. You can train it and you can customize it, especially I highly recommend there's one talk from one of the nanopore team members. They talk about how to customize Albacore or Guppy or Remora for your particular lab scenario. And I also highly recommend Epitome Labs. This is a tutorial website, so they have plenty of tutorials, and you can see they have different versions also released, and they have one for
also share this in the chat. So there was a question. Next question was to a full guide or these tutorials, I think.
This is the place. Yeah. So all students go to labs.epitome.io, and you have plenty of tutorials. So let me click on one of them, and you can see you first start with the installation, right? So you need to install Docker to access the server. So you can just install it on your desktop and then start with the basic stuff like introduction to FASTQ, the VCF file if you're interested in variant calling, the BAM file, FASTPy, do the basic QC tutorial. Start with this. And then here you have adaptive sampling. And if you're into plant genomics, do the assembly. Highly recommend you do the metagenomics, the epitomy sequencing, 16S analysis. Also, they have other examples also. For example, if you're in COVID virology, highly recommend the SARS-CoV-2 analysis. So let me just open it quickly. I'll show you. So basically, if you're familiar with Jupyter notebooks, so this is how it looks. So right now it's a static page, but once you connect Docker and you have the epitomy labs up and running, I can click on any of these code chunks and hit enter, the code will run. So now it's just a static HTML page. So here we'll have like instructions followed by chunk of code. So it's very easy to follow these tutorials, especially if you're new to working in command line. So Jupyter notebooks, if you are into bioinformatics, you need to know Jupyter notebooks. That's something I recommend everybody get familiar with working with Jupyter notebooks. Yeah.
Thank you. Thank you so much, Dr. Kanan. And I'm just looking at the website. I'll share it with our audience. And I think we are running out of time. And thank you so much for your participation in this keynote session and helping all of the listeners with the advancements and the technology. So we will be able to share with you some of the questions that are still there. Now from, as I was saying, that from clinical informatics to understanding about the data and its application, now we have our next keynote speaker amongst us, Dr. Nidhi Khurana, and a close colleague and head for academic coordinator at Faba Academy. So Dr. Khurana is currently the head for academic coordinator with Faba Academy, which is aimed at imparting industry relevant skills to the life science students. So besides being a molecular biologist by training, she is also a life science mentor and has mentored over 200 more plus students till now for career building in the life science sector. So with that, I would like to invite Dr. Khurana. Hello, Nidhi.
Yes, hi Mohit. I'm sure you guys are having a great day, and congratulations first of all for this event, and thank you so much for introducing me to the audience. Okay, so shall we get started?
Yes.
Great. Before I get started, everybody, I would love to know my audience. And I would also appreciate if we can be as interactive as possible. So let me start by asking all of you your, the current status, like your educational qualification, your stream. It would be nice to know your background, like in terms of where you come from, as in educational qualification. Your MSc, PhD. Please type in the chat box. I would love to see some answers. Because, my talk is about finding that missing link between academia and industry. People are talking about the gap in academia and industry. So what is that gap? And what should we do as professionals and as students, to fill that gap, to make a bright future for ourselves? So do we have an interactive audience here? My first question. Let me know. PhD. Wow. Thank you, Jyoti, for answering. I'm expecting a few more answers, everybody. Are you BSc, Masters? Okay. MSc Bioinformatics students, Bachelors of Pharmacy. Very nice. Thank you, Tiffany, Rupa, Divya. Great. Well, Biotechnology PhD, MSc Biotech, BSc. Great. PhD, beginning. Glad to know, Tracy, MSc Genomics and Precision Medicine. Okay, that's an interesting mix of audience that you have, ranging from undergraduates till PhDs and researcher. Okay, so I will now share my screen and you can keep answering in the chat box. Let me share. Okay. Can you all see my screen? Give me a thumbs up. Great. All right, everybody. So that gap that I talked about between academia and industry, it is hurting the entire ecosystem, the entire ecosystem of life sciences industry. Why? Because on one end, the companies complain that they cannot hire fresh graduates or inexperienced people because they're not adequately skilled. And on the other hand, the complaint by the students is that companies do not hire them, and so there's a misconception around number of job opportunities or the training, the number of training courses that can enhance their skills. So there is some kind of a missing link there. So my talk is going to focus on what are the expectations of the industry and what is academia, based on my observations, what is academia currently doing, okay, to help the students make that transition. But before I delve into all of this, let me start by a common expectation from that we have from ourselves when we enter into college. That first day of college after our school, and what do you have in mind when you join your college? The expected professional path by the students looks something like this. You ought to complete your graduation or post-graduation, and then you apply for jobs. You expect yourself to get a job, and then you perform well, you get promoted, and then live the life you desire. So tell me, at least I had this expectation when I joined the college. I would love to know from you, after your BSc, MSc, PhD, whatever course you're doing, what is your professional expectation from you? Does it align with what you're looking at the screen right now? Yes or no? Give me a thumbs up. Do you expect to get a job from whatever you're learning in your institution? Isn't that the sole purpose? To find a career for yourself? Yes or no, guys? Okay, great. So exactly, most of you are saying, yes, that is the expectation, but do you think that this expectation is being fulfilled just in that manner that you are thinking about it? So the reality is, a student completes graduation or post-graduation degree program, then they apply for jobs, and then what do you see? Struggle to get hired. Maybe most of you may have also experienced it. If you actually tried applying for jobs, you do not hear from organizations for interviews. You send out your CVs to hundreds of jobs, but you do not get back with any reply. You're told that you do not have the required skill sets to work in the life sciences industry. And maybe if you get an interview, you turn down because, again, as a fresher, you're told that you do not have relevant experience. How many of you actually relate to either of this scenario here? Andy here says no jobs even after masters in life sciences. So I do not want to comment on that right now, because there are jobs, Andy, I can assure you that, but it's just the approach, and because nobody's preparing us to approach for those jobs. So that is why we are not taking the right steps at the right time to really benefit from the opportunity. So here, the reality that that I have shown you on the slide, it resonates with most of you. And this is what my observation after talking to 500 plus students in groups on personal basis, most of you come up with this scenario. So do you see a problem here with our expectation from academic institutions and basically the outcome of the entire scenario? We expect our institutions, our colleges, our universities to prepare us to build a career in life sciences, but somewhere something is missing, because after we complete our education from that college or university, we actually end up struggling to get a job, right? So what is that missing link? What does the industry tell most of the time to the candidates who approach them for a job? That you do not have the adequate skill sets, And I'm sure, and after talking to a lot of students out there, life sciences students, our academic institutions do not even, lay enough emphasis on the term skill sets, enough. So most of you, or, if I have an intelligent audience here, good, because most of the students who I have talked about, they are not even aware of the term skill sets. The term skill sets confuse them. So let me just, give a brief insight into what a skill set is. It is actually the knowledge, the ability, or experience needed to perform a job. It could be any job, guys. It could be R&D, it could be management, it could be, accounting. So based on the skill sets that you have worked on and, you're working on to hone them with your everyday practice or your experience or your exposure, you develop these skill sets which you can use for your job later in the future. Okay. So in terms of skill sets also, people talk about two kinds of skill sets. One is soft skills. It's basically nothing but your personality on the whole, how you react personally to your surrounding in various aspects. But there's another, so academic institutions do contribute towards improving your soft skills, but most of the work should be done by you. When it comes to hard skills, these skills are actually quantifiable and teachable, and they include the specific technical knowledge and abilities required for a job. So they are technique oriented. And it is the hard skills that the academic institutions, or we expect the institutions to prepare us when we go for college, go for a course, to prepare us in terms of hard skills, so that when we approach for a job at a company, we are not turned down because of lack of adequate skill set. So here, now I would like to point out various, these are all my observations, and what is academia focusing on instead of actually preparing you for a career in life sciences. So academia focuses on completing the syllabus and giving good CGPAs to the students. Let me know in the chat box if you agree to these points or not. I would love to see your answers. See, we have an educational curriculum to follow, okay, and academia restricts itself to that curriculum. But what do I mean by giving good CGPAs? Actually, most of us come from the background where, during our school time, if you're toppers, if you're among the top 10, we really, our parents take pride in us, and we really make everybody proud, our teachers, our family, and everybody. So we have this mindset that when we enter college, that is the sole requirement by a candidate to get a good CGPA. Most of us think when we enter into our bachelor's program that having good CGPA is what is going to get us a very good job. But how many of you really know the example of people who are doing great in their career, but did not have the good CGPA back in the day? It's actually not about the CGPA. CGPA absolutely matters, because that is where your basic understanding of the subject is coming from. However, you need to implement the techniques, the subject knowledge that you're acquiring, okay, and put it in practical aspects. So what matters is exposure. So having CGPA is important, but you need to move beyond CGPA to literally build on that career for yourself. Second point, academia actually teaches about applications. I'm not saying in our colleges we've never we never knew the applications of any technique that we were studying. But the restriction here is they teach about applications of a technique, but only theoretically. For example, RDT, recombinant DNA technology. How many of you have answered the question, where RDT is used on a paper as a part of your answer and listed the applications on the answer, and that's it? You did it to score good marks. How many of you literally delve into the detail of the application of RDT in solving real life problems? Which companies, which solutions are, they're favored by RDT? So it's only theoretically that the focus is fixed on. The third aspect is actually, although basic and advanced knowledge of the topics are covered, but how these are implemented to find a solution or create a product is not emphasized upon. Let me know, you are allowed to give your views here with respect to the points that I'm putting up, guys. So when we study about any topic during our college, yes, we study about its basics. Yes, we study about its advanced. But what we fail to study is the implementation of the technology to really find a solution, or how using the technology a product is created in real life scenario, right? So that is where the gap is in understanding the entire operations behind life sciences industry. Another, academia actually thinks industrial visits are enough to teach students about how the work happens in an industry. So let me know, while I was, putting up this point, I was laughing to myself, but actually that's true. In my MSc also, I had an industrial visit, and I'm sure in your universities also, most of the places, they arrange these industrial visits to understand, to give you a sense of how industry works. But is that what is required? What does it even do? For like a 5 hours or 6 hour visit, how does it help a student? But colleges do it, and basically they should do something more than that to really help the students understand the entire scenario of life sciences industry. Another point, they do not educate the students about the concepts of skill sets required in the industry. As I've emphasized upon it earlier, that the term skill sets, how to build on them, how to really analyze your strengths and weaknesses, to develop those skill sets for you so that you really enjoy what you do going forward, is completely lacking. And then ironically, very few institutes try to place their students after their graduation, but the entire process of those placements that occur for life sciences student is hollow. It does not have any meaning and does not help the students much in their career building, guys. Now that's the truth. You like it or not, most of you, how many of you literally continue with the job that that you're placed after your college? Does that even align with your strengths and weaknesses? Does that give you a sense of whether you are built for the job or not, whether you're going to enjoy it or not? Nothing. There's no preparation behind that scenario. So academia does a lot. There are a lot of points here, but I think the way it executes this thing needs to have more meaning to it. Let me know if you agree to me or not here. Give me a thumbs up if we are aligned on this, at this magnitude. So give me a yes or no if you really connect to what I'm talking here. On the other hand, guys, I need you to focus on what the industry wants. Okay, till now we actually saw what academia is providing us in terms of preparing us for career building in life sciences. But what is it that the industry wants? So everything is based on the applications of the techniques in the industry. Okay, we have talked to many people from Faba itself. We conduct many panel discussions. We go into one-on-one personal meetings with the industry leaders to understand the gap. So everything in industry is based on applications of the techniques. Industry is actually product or solution centric. Okay, it seeks individuals who understand the basics and applications of the techniques used in the company. When they say that you do not have the adequate skill set, what do they imply? So for example, they're looking for somebody who knows about the use of HPLC. Okay, now if you're someone who's used HPLC before, you fit right into their pocket. But the thing is, if you are not very well acquainted with HPLC, if you do not know how to handle the equipment, if you do not know how to work with it, how to generate data with it, how to interpret its data, or troubleshoot the experiments that are performed with that experiment, then you cannot add value to that job. And industries look, when they go through your resumes, when they go through your profiles, what they're looking at. They're looking at various ways how you can add value to them, how you can help them grow. And that is where this lack of a skill set is coming in. Our educational curriculum is too much focused on providing us basic and subjective knowledge. When it comes to practical, it's mostly restricted to whatever we learn in the laboratories. But that does not do justice to what is required in real time at industry level. Okay, so industry seeks individuals who know how to handle the equipment, can generate and interpret the data, and most importantly, is able to troubleshoot experiments. Sometimes industry would employ somebody who would not have a hands-on experience on HPLC. So there is this another factor when you are interviewing for that position, or you are telling about yourself to that recruiter, potential recruiter. It's more about your attitude. Your if you would not have had the hands-on experience on HPLC, but you should know what HPLC is, what is its principle, what is the insights behind generating data from it, how does it help their projects, like why do you use HPLC. So at least these are the aspects you need to know, and then as a fresher, having hands-on or no having hands-on can be tackled at some level. Okay, so for that only, there was this one statement in the chat box which said that there are not enough job opportunities after MSCs. No, there are, guys. Okay, it's just that we need to approach them in the right manner. And life sciences students actually need to put in a little bit more effort as compared to the other streams, engineering, IT, computer science, anything, because they have been established from a very long time ago in this scenario. And for life sciences students, mostly the path till now has been BSc, MSc, PhD, postdoc, and then becoming a professor, right? But now you see a lot of companies, you see a lot of industry, organizations that are coming up with projects, with healthcare projects, with life sciences, biotech, pharma. It's always been there. And now it's time to open up our minds also to explore these opportunities. But life sciences students need an extra push, an extra nudge to really approach those opportunities in this case. So now we know what industry wants from us and how academia is not able to provide us that kind of training. Here, I have actually put up a snapshot for you about the typical academic syllabus that we have. So this is a general, some of the general topics that all of us find in our syllabus, be it MSc Biotech, Microbiome, Biochemistry, and everything. And then below, I actually have industry skill requirements. So you see in our academic syllabus, we have these big terms, bioinformatics, genomics and proteomics, bioentrepreneurship, nanobiotechnology, bioprocessing engineering, and fermentation technology. These are actually industry relevant subjects or topics. The problem is how the academia teaches us about these subjects. For example, genomics and proteomics or bioinformatics. If you really think academia was doing enough to really, impart that skill to us, which was industry relevant, then an organization like Omics Logic, okay, which is doing great work in this scenario, would have had no meaning. But Omics Logic, you are attending this webinar, you are a part of their courses, you know what they teach, how nicely project oriented they are. So case studies are important. You need to work on real life problems to really understand how bioinformatics or genomics or proteomics or nanobiotechnology is helping you find a solution. That is what is needed. Okay. When it comes to industry skill requirements, I want you to see the difference. So industry skill requirements are advanced technical concepts in molecular biology, in vitro assays, in vivo models, preclinical drug discovery and development, biopharmaceutical production and analysis, facets of industry R&D focusing on product development and correlation between functions. industry focuses on physiology, pharmacology, toxicology, and regulatory studies. You tell me which of these topics are covered in the academic syllabus in a way that we should be prepared to get a job right after our degree. Like negligible, guys, negligible. So here is the gap. When I say that skill set is the missing link between academia and industry, what I mean is this, okay? Going forward, what needs to change to really have a positive effect on this entire scenario? What academia can do at their level to really help the students build their career in life sciences industry? Starting right at the bachelor's level, there should be mandatory industrial internships, and preferably for 3 months. Why do I say this? So one day from Faba, we were organizing this workshop on drug discovery and development, and basically our partner from the US, he randomly said that in our curriculum, the students start taking internships from the very first year of their bachelor's. So there, in the US, the bachelor's is like 4 years. So by the time they complete 4 years, they have an exposure of working in 4 different industries at 4 different roles. And so that is why when they graduate, they actually feel ready to perform in the industry. Okay, you are no longer lost because you've had prior 4 times experience of reflecting what you like, what you don't like, what are these various job opportunities, how I can perform best in this particular role, having been connected to people at 4 different places, you have built a community and network for yourself so strong that after 4 years, after those 4 internships, you are actually connected to people who can help you get the job. That's what networking is. And that's what skilling looks like. Another thing that the colleges can, ponder upon is empowering the culture of working on projects based on real life problems to add to the students' experience and foster entrepreneurship. Right now, these colleges have these placement cells and entrepreneurship departments and cells. But the thing is, some proactive approach needs to be taken to empower a culture where the students are, they are required to team up, to work on real life problems, to find a solution at their level. There could be many solutions to one problem, everybody. And so when you do that, you actually get real-time hands-on experience and you foster that mindset of entrepreneurship, the solution-centric mindset. Another thing, exposure to the pilot scale manufacturing projects and quality control studies for students should be conducted by collaborating with startups or industries to impart industry relevant skills. College also needs to educate them about careers outside academia and the necessary skills that each of those careers require. Because it is also important to understand that research and development is not the only option after your bachelor's, master's, or PhD, everybody. You do have different roles where you can outperform by your skill sets, by your likings and disliking, by reflecting, by building on your strengths and considering your weaknesses. You can go for other careers other than research and development. It's not mandatory as a master's student to really just go for research after your, after getting the degree. So college needs to educate them about various career opportunities and how to prepare for them by telling them that this particular job requires these strengths. And if you're a person who has those strengths, go for that job. So the last thing, the least that the college can do is encourage the students to build their career based on their strengths and weaknesses rather than following the herd, just what I just mentioned. So over here, guys, let me know in the chat box that are you with me here, or do you have any contradictory comment or your point of view or your perspective about this? So because I think that this is where the gap lies, this is how the academic institutions need to act upon to really build, that mindset, to really build experience, to really give the confidence to the students for career building in life sciences. Going forward, now there's a scenario that was a scenario that academic, institution need to do at their level. But that's, I don't know how far that is from practicing. So what you can do at your level, guys. So considering what the industry wants from you, considering the limited amount of work that the academia can put in preparing you for getting jobs, it is your responsibility to take proactive steps in these areas that are shown here. So what you can do to make your transition smoother from academia to industry, go for internships, everybody. Go for internships at least once a year. While you're doing that internship, reflect on your strengths or build your experience around it. Okay, you need to reflect on your strengths, if you're liking it, if you're disliking it, or if you need to consider that on an everyday basis, would you be enjoying this role that you are a part of right now? Because having an internship gives you a lot of exposure that is required to really build that fulfilling career going ahead. Second is focus on networking, online and offline. I cannot say it enough. And I don't know if you're following me on LinkedIn, most of my posts, they focus on this particular point, networking. As life sciences students, nobody talks about it, nobody encourages us for it. And so whenever we talk to people, whenever we talk to the students, they always say, I'm not a conversationalist. You do not need to be a conversationalist to actually network. Network is when you connect with people, when you deliver value, when you take, help them and take their help and build a community for yourself. So work on it. When I say online and offline, I'm talking about platforms like LinkedIn, and offline, I'm talking about conferences, symposiums, your class, your seniors, your professors. Make full use of conferences and symposiums. Talk to people. You should go prepared when you go for a conference so that you go with a targeted approach. Then you build connections, reach out to your alma mater, reach out to your professors. The entire strategy can really help you find that job for yourself. It can easily get you hired if you start with this strategy at the right time. It is not late for anybody, guys. Okay, but if you start at a younger level, then you actually have ample amount of time to build around these. So the last is increase your visibility. Talk to people, let them know you exist. Until you tell them you exist, how are they going to find out that you are some person who is strong at this and maybe perfect for this job? So if you increase your visibility, if you talk to people around, if you network right, if you approach people and build connections, there is no reason or there is no scenario that you cannot get hired. So these are some things that you can do at your level to really bridge the gap and make your transition smooth from academia to industry. So guys, I'll just take last 2 to 3 minutes to tell you how Faba Academy is helping in this space. So to educate the students and support the students as they explore and further understand themselves in the career options and to gain valuable experience, Faba Academy was launched in 2020. And what we do is we conduct webinars which are career oriented. We conduct these workshops which upskill the participants in industry relevant skill sets, which gives them better career growth. So just giving a gist about who is Faba Academy for. So it has a lot to offer to every kind of domain of life sciences sector, graduates and postgraduates, PhD postdocs, industry professionals, faculty, everybody. So our strengths are, we have upskilled actually 400 plus participants, including early career professionals. Our speakers are, for these workshops are invited from India and abroad to give you really wholesome, exposure about the advancements that are happening in technology, the career opportunities that are coming up in this field. And so these are some of the workshops that we have conducted, like drug discovery and development, basis of chromatography techniques, hands-on training on DNA fingerprinting, biosafety, biosecurity, upstream and downstream technologies of biologics and biosimilar. And we do conduct workshops in collaboration with Omics Logics also. Just like this we did last year, biomedical data science and drug discovery and development this year. So now we're moving on to doing more hands-on workshop, because until 2021 it was more like virtual. But when you gain these extra skill sets which are industry relevant, you are actually equipping yourself with those adequate skill sets that the industry is looking for. So with this, guys, these are some of the webinars that we have conducted. For example, RNAi technologies and career opportunities. This was about next generation sequencing, and this was about science communication. Rewrite your strengths to become a science communicator. We invite the experts from India and abroad, their CEOs. They are outperforming themselves in their field, so they bring their own wisdom and experience to teach you about these aspects. This is about one of the upcoming webinars with Dr. Gautam Das, who is the co-founder and managing partner of Bioinformatics, and it is on 8th September. I would love for you to explore this opportunity and register for this webinars. Our webinars are free of cost, and so you be a part of it and gain some value out of it. So guys, with this, thank you so much. I would love to answer some questions if you have, and let me know if there is any way that I can help you with. Thank you very much.
Thank you so much, ma'am. That was indeed a very interesting talk, and we have a bunch of questions to ask. But as we are running out of time, I would email you these questions, and I hope it would be fine if you could answer them, and we'll get back to the participants.
Thank you. Thank you so much.
Thank you so much, ma'am, once again for the wonderful talk. And now that we know what is the right kind of skill set that is required for industrial research, how can someone acquire these? What kind of internships or job opportunities should one look for in such scenarios? So now we would be starting with our academic panel discussion, and I would like to welcome our academic panelists, Dr. Kalyani Korla, Dr. Shibi, and Dr. Sonali Khandra. Dr. Kalyani holds a PhD in biochemistry and is a mentor at Mentis Hive, and Dr. Sonali holds a PhD in cancer biology, and she's the CEO of Informatics.
Thank you, Sonalika, and hello, everyone. We are now heading towards the industry panel discussion, and I think what Nidhi did was like to give us this amazing platform with her energetic talk. And Nidhi, as you are also a part of this panel, and if you can also join in, so that was a great discussion, and we covered like a lot of points that we were hoping to discuss, and actually that makes sense now to go into this next session, which is to discuss some of the opportunities that are there, and also to summarize some of the points that we have put together. Let me share my presentation. So we kind of we've been discussing here since yesterday about how the academic research is shaping up in India. We have had great experts, and also participants from all around the globe listening from the experts about how their research has influenced, plus what they see that how like bioinformatics can be a part and can be integrated into several ways, and the pathways for students to learn about it, and to maybe transition to the industry by having those understanding of those hard skills that Nidhi was just talking about. But we would want to start with this discussion that as an industry, as an entrepreneur, as a leader, how do you see this industry evolving? And that brings me to my like next slide, which is like this recent report about the healthcare being reinvented in India, and the numbers around it, and the different areas where the survey sees that there is a huge application and hence the opportunity. So again, like about Make in India programs from Indian soil that have nurtured much of these biotech programs, but somehow it remains nascent. So what is then the solution? Like there are tech parks, and I know the one that had opened up in Hyderabad, and Sonali has been part of it. So like industrial collaborations, Silicon Valley like ecosystems. So I mean, with that question and with that note, I would want to maybe go to Sonali first. and start a discussion about this current situation, and where are we, and where do you see that where are the opportunities for the future of Indian healthcare sector, and also for those students who are now freshers and who are listening for the first time about something like this.
Hi, good evening, everyone. Am I audible?
Yes.
Okay, thank you. Thanks a lot, Mohit, for giving this opportunity. Really, so glad sharing, my evening talking to all the other, along with all the other panelists. And it's really a great platform, and the work what you guys are doing, educating young, individuals and all talents, and of course, giving them a path or the direction moving forward next, what's happening in the industry. So that's really commendable, because at least if I remember 10 years back, there were no one to show us the path. So I would say that, you guys are really doing good job for the next, the millennial or the future scientist.
Thank
So
you
yeah,
much.
coming back to the investment in this space and the industry itself, how it is going. And I think, I can take my own example and why the whole opportunity we have seen and we have invested in this space, and we have, and that the whole genesis of Ekiminds happened. So just for the people who are not aware, I come from life sciences background, have my PhD postdoc in that, but of course always felt the gap and always felt that there was, the missing things, which was definitely, bringing more technology into it. And then I worked with a couple of, tech startups, some VC firms, to understand, how the whole space moving in this, like the medtech space, how the investments are coming, and how the business model should be built. Because when you're a researcher, you just focus on the science. You've never been allowed to even look at the business part of it, right? Like how to actually take the whole science to the market. I think that's the most saddest part of it, because, as a researcher, you don't really look at who's going to use it, who's my customer. You don't use this word at all. Who's the customer? Who's the user? You just do your research, right? So I think that's the whole change in your mindset, your perspective, when you come to, the industry and you look at who's going to use this. And when you know that who's going to use this, your entire perspective towards building it completely changes, because there's a person, there's a patient, or there is a user who's going to directly use my product or my services. So your perspective change. This is no more just for mice or rat or for animals. You're going to really change people's life. You're going to touch humans, right? So that's a huge change which you see from your academia to industry. But coming back to your question, yes, and then we started bringing more questioning more, like how we can bridge this gap between people who come from life sciences have no understanding of technology, and on the other hand, people who are tech, who are really good coding skills and techies, but they don't understand the domain. So how do we bring these two worlds? And when I was at IIIT, I was heading the innovation center. We had amazing startups, but all they were like tech people. They have absolutely they jump into startups, kind of trying to solve what is required and what is the unmet need in healthcare. But the challenge was, because they don't come from domain, it's always tech first kind of approach. But that's how it doesn't work that way, because it has to be a problem first approach. Like, what's the demand, what is the challenge, right? And then you try and solve that problem using technology. So it's always the other way. But I think the missing thing was, having that interdisciplinary knowledge and kind of, awareness that you come from the domain, but you also know what kind of technology can be used to help these people or to solve this challenge. So that's the opportunity we saw. And of course, that's whole was the genesis of Ekiminds, where we, it's basically a bio IT or a tech-led healthcare company, a life sciences company, very, very, very focused on, bringing these two worlds together and try and see how we can, basically leverage the talent and bridge and basically skill them to the next level of job which is required, which is demanding, and what kind of skill sets are required in the industry. And that's what we are doing. Coming back to the investment in this space, it's really incredible at this point of time. In fact, when I just started, I kind of got a couple of calls from VCs and messages that they want to invest, but that time we had just the seed fund. So I was so happy to see that yes, investors are looking at this space quite, I mean, they are seeing the future of return on investment on this space, which wasn't the case a decade back. Life sciences has been always a very riskier space to invest in. If you look at the other space like fintech, edtech, there's a huge investment been pumped up in last 1 or 2 decades, but nothing has been really happened on the health tech or the life sciences side. But now what I've seen is like there are very focused VCs has come in, angels have come in, who are advocating, who are brand ambassadoring, who are really interested in spending on this space. And I think we all have seen that post COVID, the way things have changed, the adoptions have come. And if you just look at the number of startups, in fact, before this, I was just trying to figure out some numbers, and I was amazed to see like there are almost 5,000 plus health tech startups are born in India on the healthcare and health tech side. So that itself suggests that how people are trying to get into this space, invest in this space. And of course, for freshers, I would say, please open your ears and eyes and look at the opportunity, and just sky is the limit in this space. Yeah, thanks.
That makes sense. I think with all of that, your experience and the points that you have mentioned, which is like, I would say, for an entrepreneur or person who is thinking about to do something with it, like startups and all, the interdisciplinary nature of this field is kind of something that requires them to know about. So general awareness, I think, awareness about investments, awareness about technology, awareness about what is happening in the fields around it, and what are the innovations. These are some of the very important aspects someone who is diving into this and thinking about to do this. And again, like another point that we discussed about, that having a prior experience or doing these internships where the startups are working actually could help a lot of people, a lot of these students to overcome, to understand the challenges, for example, first-hand challenges. So I think great value there to this to address this question. And if Dr. Kanan can maybe add something to it, like how does he see, because Dr. Kanan, you are working on the frontiers of oncology research and using these new technologies. So how do you see this market, and how do you see that, what kind of opportunities? Because what is happening also is that like these specialized skill sets are something that the postdocs are getting an exposure in the US. So how do students in India get exposed to those cutting edge technologies that you are working on, and there is a scope of capacity and intake of such students?
Thank you. That's a very good point. So if you live in US, if you are a student in a university, either a PhD or a postdoc, you frequently go, almost every weekend. We used to have Fridays and Saturdays. We used to have like institution-wide meetings where you interact with everybody. Also, we get to travel. We get to, so in oncology, we go to 3 main conferences: American Society for Hematology, ASH, then American Society for Clinical Oncology, ASCO, and AACR. So if you're a basic science guy or doing some kind of product development, you go to AACR. If you're a clinician, you go to ASCO. And if you are in hematology, then you go to ASH. So, and then if you live in Europe, you go to ESMO. So I had the opportunity of attending all these conferences when I was in US 10 years. Every year we used to go all the meetings. The university used to sponsor, my PA used to sponsor. One thing I really miss after coming back to India, that physical presence of attending these meetings is not happening because it's too expensive, right? First you have to travel, then you have to book the flight tickets, you have to get the hotel and all that stuff. And then there's a conference registration fee. But one good thing happened in the pandemic. They say every crisis comes with some lot of bad things, but also some good things. The good thing that happened, many of these conferences are now online, and virtual events have become the mainstay. So right now we are in a virtual event. No need to travel, no need to spend money. And you still have to pay certain conferences. They still charge you for the which I don't understand. They still charge you a registration fee, but it's a lot more economical than actually physically traveling. So highly encourage the student community here in your area of domain of expertise, follow these institutions, these organizations. Like we follow AACR and follow them on Twitter, on LinkedIn. So as soon as the event is announced, immediately register. So during this meeting, I just registered for a MIT bootcamp that's specifically for healthcare entrepreneurship. So it just took me five minutes. When I was listening to Sonali, I was just filling up the form on MIT bootcamp website. So it doesn't take much time. And what I do is, whenever I see some notifications, whenever I visit some good websites, I highly recommend every organization, whether it's a for-profit or a non-profit, they advertise the webinars and CME events for clinicians. Webinars are very common nowadays. And they also have all these global events like the London Calling event I was talking about. I have been attending London Calling virtually since 2019. I've been to London once or twice, but I didn't get the opportunity to attend a live nanopore conference. All the nanopore community meetings, London Calling, everything I'm attending virtually. The good thing about nanopore, they have the video section in the resources page where you can watch all the videos. So you can also, if you register, you attend in live, you can also interact with the audience, interact with the speakers. Also, you can interact with the staff members. You can ask all your doubts. So like that, many companies do that. So that's something, an opportunity, even if you miss the live session, but I still register, even if I cannot attend all the meetings, I will get a recorded session. They email me, they get a recorded session. You can watch it in your free time. So subscribe to newsletters. Any Zoom link you see, any conference, any webinar, click the register button now. Right? That's the way. That's the way to stay connected. Because once you leave academia and come to industry, nobody's keeping you up to date, right? You have to take your own initiatives to learn what's going around in the rest of the world, especially for people who are in India and other emerging economies who don't have access to travel. Something happening in San Diego, something happening in Chicago. Obviously, I can't travel now. The embassy is closed. They're not issuing visas for at least one more year. So it's a huge backlog. So, but the same meetings are available virtually, even if they charge you $1,000 or $2,000. I would say it's totally worth it to register for the meetings, because one thing, you get access to all the abstracts, all the plenary symposiums and everything. Plus, you'll also get the contacts, the networking. So most of the time, I'm not even listening to the speakers. I'm actually in the chat box talking to people, on networking. So I highly encourage these three strategies: subscription to newsletters, attend as many webinars as possible, and when you attend a real live session, try to network with the attendees. So here we have like more than 70 people in the group. So try to interact with them, direct message them. Don't clutter the common group. Do direct message to the person you want to talk to. So that's my three main takeaways from this virtual meetings. That's how you stay connected. That's how you stay up to date with what's going on around the world. So then you are not left behind, right? So you are on par with your colleagues in US and Europe.
Yeah, I think great points and hacks there. I mean, we do know about it, but we don't know how to utilize them and where to find these resources. So I think one of the key to finding information is also searching properly, searching for these keywords that are necessary for you to know exactly where these resources are. And most of the things are available online. And as Dr. Kanan rightly said, that how the COVID-19 pandemic has changed the entire way of knowledge transfer, and also gaining access to all those events. And especially on an online setting, the opportunities are a lot. There's a lot of opportunity, and many of these events are also free for students and users. And also recordings are available on YouTube. So there's a huge resource out there where you can learn about these new technologies and maybe connect with people over LinkedIn who are working on these specific technologies, because I think that is one great place to find people who are working on it, something that that like motivates you, and then connect and then have a meaningful discussion. So I mean, with that, I would go to Kalyani and ask her that how's her experience in Mentis Hive, and how do you see, Kalyani, and very good evening, how do you see this academia to industry transition, and those points, like when you prepare to be like maybe get highlighted by the industry, or get your profile being highlighted, so that the relevant person can reach out to you, or you can reach out to relevant person as in and get that impression.
Thank you so much, Mohit. Can you hear me?
Yes.
Great. Good evening, everyone. And thank you so much, Mohit, for giving this opportunity. And it's great to be talking to a lot of upcoming scientists over here. So I was listening to Sonali and Dr. Shivi and Nidhi before this. So I think all these things that these people have told has a lot of value that they have brought into this space for the students who are here. So quickly adding something to what Dr. Shivi was just saying about conferences. So I think students can relate to this. When we were students, we also used to think of conferences as an outing, that when you're going for a conference, it's more like, okay, I'm going to ISC for a conference, so I'm going to go there and have fun and all. But what Dr. Shibi has told just now is something that you should all note, that conferences are not just for fun or food and all. It's more about networking over there. And one more point that I would like to add along with the 3 points that he has mentioned is, you can target people over there. So suppose if you want to connect with Dr. Shibi sometime on a personal level, he just mentioned that he is going for a boot camp. And if you are interested in something like that, that's the place that you can target. That is just an example that I'm giving you. So if you have been following a professor for your next postdoc, or for a job position going forward for somebody who is into industry, make sure you find out, you follow them on LinkedIn, keep following up that which events are they going to attend in future. You keep track of those events, attend those events, so that you have a context for a meaningful conversation with them when you go there. And even when you want to connect, you can say that, okay, I heard you in this particular conference, you spoke about this thing. The moment you put this personalized messages over there, that is what will bring the limelight to you. So I have been into industry, like I have transitioned, I've done my PhD, my postdoc, then I was in a healthcare startup that I was working in. So for 3 years I was there. Then I completely pivoted. Now I'm a product manager in a video publishing house from US. So these transitions, so you can see I have been in the trajectory where I have experienced all these things. And I would strongly recommend that networking is something that plays a big role, Mohit. And I'm sure you also understand because you're also in industry now. So that is something that students should definitely think on. And one point that I want to elaborate a little bit on what Dr. Sonali was also indicating about mindset. So this is something where we are all getting stuck. It is not a very big thing. People have been making a huge hue and cry around transitioning from academia to industry. It is not very difficult if you are taking the right approach, if you have that mindset, if you are prepared for it, even before your masters, when you join masters, what kind of field you are interested in, take internships. Everybody has been telling about it, so there's nothing new about it. But when you're taking internship, when you are looking at the particular job that you are doing there, also look at people around you. Does anything else apart from your own job interest you? You can maybe look at some project manager who is there in your company, and you can look up to them. You can see, okay, this person is doing this, I would like to do a job like this. Set goals like that, and then take a top-down approach so that you can make a career path for yourself. That's something that we keep telling our mentees always, that the problem with our system is we are never taught to think beyond our next degree. The moment you take admission in PhD, you are like, okay, 5 years, I'll just focus on my PhD. No, that's not the right thing, and that is not something that you should be doing. You should know what is your next step going to be after that, and you start preparing for it. So like life is like a chess game. So when you have heard this, that in a chess, a grandmaster will prepare for a move 7 moves ahead. So why can't you do that in your life? I'm sure your life is more important than a chess game. So I think these are a couple of things that people should start focusing on, Mohit. And we were talking about the healthcare segment. It is booming like anything, and it is not the not the boom that we have seen in EdTech. It is going beyond that, because something that is related to us, that all of us can relate to. And even the investors are seeing this, as Dr. Sonali was also indicating. So I actually urge students always that you go for a startup job first, because my first job was in a startup. You get to learn a lot. When you are in a big company, it is like you are given a small chunk of work and you do the same work day in, day out. And I'm sure Dr. Sonali would agree with me on this, that if you are in a startup, you get to wear many hats. You get so much of exposure. You get to learn so many things. So early in your career, if you're hustling at that level, you learn so much about yourself, about your domain, about your industry, that going forward becomes much more easier for you. At least that's what I feel when I have transitioned from academia to industry, then to another category altogether. So these are a couple of things that we should keep our minds open to. Keep looking around people, who are there, what kind of upskilling they are doing. Upskilling is a very important thing that we should always focus on. That is where omics research, omics logic comes into picture, biotech comes into picture. You should keep upskilling yourself, never get stuck, even after your PhD or postdoc or master's. I did my postdoc, a 3-year postdoc, and even after that, I wanted to upskill. I wanted to grow beyond that. I didn't want to get stuck after saying that, okay, postdoc is the highest degree. What more can somebody do? But I wanted to break that thing, because I wanted to reach out for more. So never think that, I have done this and this is enough. It's never enough. There's always something to learn. I think that answers your question, Mohit.
Yes, absolutely. And there is another important question, like I'll come to that, but yes, absolutely, you're right. And we got to know about, like, I would highlight networking, one of the things that all of the speakers have put an emphasis on, and the different ways to do this, and what is a meaningful way to do this, because we all try to do different things, but there's a structure to it. And that's what also something that Nidhi showed us in her presentation, that there is a structure to it and you can get trained. So there are a few questions, I mean, which are quite relevant. I think one question which I could maybe go to Sonali about. So Parak Sehgal, she's asking that what are the opportunities for women on how one can do research in bioinformatics or data science while working from home, because it's also like an opportunity for women who are like at the early stage and maybe involved in family and have those limitations that we are very much aware in India.
Yeah, so I mean, as a subject, it's not really women or men. It's about, the bioinformatics. So if I would just, talk about bioinformatics as a subject, it's a very interdisciplinary area where you need knowledge of biology, where you need knowledge of computational biology, a bit of coding skills, depending on which direction you are more. So you can have a combination, or it can skew between from very domain side to the coding side, or in between anywhere of this, the whole a scale of the bioinformatics. There is definitely, I mean, since it's a very collaborative space, and it's a very, very scientific area, there's definitely a need for, be there along with the team, working along with the scientists, because so bioinformatics is not the other areas where, like the IT, right? Like where you just work on some project remotely. Yes, that can be done for some time, but there would be some touch points. And also it depends on how the projects are designed. Like some of the projects which we do, basically we have a project manager who would be client facing, who would be understanding, and that person is basically who understand the biology and the life sciences, right? And they would relay this to the data scientist guys, and data scientist guys can sit anywhere across the country or the globe and can actually work on the logics or the problem statement which is defined to them, right? So if somebody is interested work remotely, yes, there is absolutely a possibility to work remotely on the bioinformatics area as well. But then again, I mean, are you just willing to do the coding part of it, or you also want to, you know, go a little bit more advanced on the science part of it, right? So I think that's the beauty of it, right? You just don't want to restrict yourself on you know, knowing the algorithm or, you know, building the algorithm, because you're working on a very interesting data. These are not like, you know, any other non-biological data. These are very live, very, very interactive data. And when you work with data, you would want to work collaboratively. You would want to work with the scientific team who has actually generated the data. I mean, let's say, for example, of NGS sequencing, right? So you get all the data coming out from your Illumina or Nanopore or whatever sequencing machine, and then you try and analyze this data. But then you will also have to understand how the data has been generated, what was the objective of it, what was the outcome you're trying to achieve. So there's basically a lot of interactions and collaborations happens among this. But having said that, there is definitely a possibility one team can sit remotely and can, you know, work on the coding part of it, as long as they can effectively communicate with the scientist, you know, remotely. It's absolutely possible to deliver even the bioinformatics projects as well.
Perfect. I think that kind of answers the question that Parag wanted to ask, because what is happening is that, like, we saw that many, especially in the context of women who has maybe dropped out and have a long gap, they are coming back and saying that bioinformatics is something that could be done effectively in a home setting and maybe in a hybrid setting. So makes sense. Thank you so much, Sonali, for that expert advice. Now, a question, I mean, for Dr. Kanan, as he was talking about networking. So there's a question by one of the students, and it makes sense. Like, after you network with the expert in the field, what tips do you have on sustaining that professional relationship for a long-term approach? Because we do start with that, but how to sustain that? Like, how do you find those, ways to keep it engaged, and give that engagement for a longer time or longer timeline?
Yes, that's a very good question. So during the conferences or webinars, we make the initial meeting and greeting, right? But how do you continue that relationship outside the webinar? So the best place is LinkedIn, obviously. LinkedIn and Twitter. So Twitter, I was not using, I was one of the early adopters. I'm like 5,000th person who joined LinkedIn and 500th person who joined Twitter. So I'm like always looking for new tech, and I'm one of the early adopters of all these new technologies. In fact, I was using WhatsApp before people in India knew about WhatsApp, right? So I'm a very, very early adopter, but then I stopped using Twitter in between because it was full of junk. Then later on, I realized the entire scientific community is on Twitter, right? So if you want real-time collaboration, instantaneous response from anybody, for example, Clive Brown, CTO of Nanopore, I simply clicked follow 1 second. Within 5 seconds, he followed me back. I was like, wow, the CTO of Nanopore is following me back. And then instantly we connected and we were talking. Similarly, during COVID, I was working on the pan-coronavirus vaccine, mRNA vaccine. So we have a lot of interest in mRNA vaccine nowadays because of the success stories of Pfizer and Moderna. So there is this Dr. Florian Kramer, and he was putting all this, and also Eric Topol and all these other guys. They're putting all the latest updates on COVID research. So I kept following them. And when they put something interesting, then I engage with them, right? I message, I reply to them, and they reply back. So that's the key metric. So it's not just you bombarding them with messages, they have to reply back. Whereas if you take people like Elon Musk, millions of people follow him, he never replies to anybody. He very rarely replies unless it's something unique, something out of the ordinary. He never replies to anybody. That's not an engagement. The engagement is you message somebody and they reply. So there's a to and fro, there's a conversations are happening. So social media is the best place to go. Don't waste your time on Instagram or Facebook. Focus on these two platforms, LinkedIn and Twitter. LinkedIn, you will also get the history of that particular person, which university they went to, where are they working right now, right? And if they are hiring, so nowadays they have that hiring tag. And similarly, employees and students and postdocs also looking for a job. I'm open for opportunities. You can put a tag on a profile picture that kind of enables. Plus in LinkedIn, one big thing which I recommend everybody is follow or connect with your mentors and peers and ask them to give recommendations. Similarly, you give recommendations to them. So it's like I give you a recommendation, you give me a recommendation. No, it's a social exchange. And also update your skills, make your profile 100%. I've seen many students sending me CVs and other things because people are always looking for jobs. But then you open the profile, they don't even put the picture on the profile, right? So then suddenly imagine the human resource person or somebody who is interested, or let's say you are applying for a postdoc job with the faculty, they don't even see your face. How do you expect them to respond to you, right? So it's all about the human connection. So keep your profile 100% updated. So if you take any online course you've done, for example, you attended this event, for example, then you can post it in LinkedIn and you can put the hashtags, put the links and everything. Automatically you will get that recognition, right? It's all about social proofing, recognition. You need to be in control of your own domain. And you have to showcase that you are going that extra distance, you're taking initiatives to learn things and exchange ideas and connect with your peers, also connect with the faculty. So it's not like suddenly pop out of nowhere and say, hey, Dr. Kannan, can you give me a job? It doesn't work like that, right? Even if we have some openings, right? There are so many qualified candidates. Why should I pick you over 10 other candidates who have gone through the selection process? So you have to stand out from the crowd. How do you stand out from the crowd? You have to let your voice be heard. At the same time, you want to engage with the audience. So we have a saying in startups, in entrepreneurship, your network equals your net worth, right? So build your network, and these two platforms are amazing. So in Twitter, what you do, if you see any check mark, blue check marks, the verified accounts, unfollow everybody, unfollow all the celebrities, unfollow the guys who never reply. Like I don't follow Elon Musk, I don't follow Jeff Bezos, I don't follow Bill Gates, but I follow Bill and Melinda Gates Foundation, because the foundation will put announcement, when is the next grant coming? I need to know the deadline so that I can apply the grant. Follow the people you need, unfollow everybody you don't need. That's the best case scenario. Same with LinkedIn. Don't randomly follow people, don't randomly spam messages. Connect with them on the particular topic that's being discussed, right? So there has to be some relevancy in the connects. That's my take on that. So LinkedIn and Twitter, two best places to be right now. Also learning opportunities, plenty of learning opportunities.
So many tips and tricks for the community. It has been a great experience, Dr. Kannan and Kalyani and Sonali. Thank you so much.
If I may add a couple of actionable tips over there. Do we have time for that?
Yes, of course. I was coming back to you.
Okay, great. So just adding on to whatever Dr. Shibi said, so for students that, when we mentor them, sometimes we tell them these things, they don't know where to start. And LinkedIn as a profile, when you just join, it can become a little overwhelming, for anybody who has just joined new. So few actionable tips I would like to give here. If you have a pen and paper, make sure you, make a note of it. So when you're connecting with somebody, suppose if you are sending me a friend request or a connection request there, make sure you mention it there. Hi Kalyani, I heard you over this particular Omics Logic conference that I attended. I want to know more from you. I want to learn from you. Your profile is inspiring. Pick up something from my profile that you can connect with. Like, okay, you worked in systems biology. I'm also currently working on systems biology. Try to make a personal connect over there in some fashion. It should not be just like, you have just clicked on the button. Then you have not made an effort. Unless you make an effort from your end, how would the other person realize that you are sincere in your efforts? So that is the first thing. Never send a connection request without a message in it. And as Dr. Shibi was indicating, that you engage with them. So how do you engage with them on a continuous basis? You comment on their post. Don't just comment saying that, okay, this post is inspiring, thanks for sharing. Put your own thoughts to it. So if you have done that 5 times, the 6th time the person would know that, okay, this person has been commenting on my post. So if at all an opportunity arises where you have to reach out to them for a certain job or any favor, you can always say, and that person would recognize you because, oh, Kalyani, I've seen her, she keeps commenting on my post. That means she has some knowledge about the field. So these are the ways that you're establishing your credibility over there. So when you are on LinkedIn, you also have to post there. So if suppose you are interested in cancer biology and a new paper has come up in cancer biology, why don't you post it there along with some thoughts of your own, that what does this research mean? What is going to be a future impact of this research overall cancer healthcare? So the moment you put in some meaningful thoughts there, that's when the person realizes that, okay, this person is intellectually sound. they have some credibility in the field, they understand the field. And once you keep making those kind of engagement, that's when your name is visible always. You are somewhere in the radar. So even if you are not connected with them on a day in, day out basis, at least, you are there on their feed every time. They can keep seeing your name. So these are some of the ways that you can, highlight your profile, kind of create a brand for yourself when you're on LinkedIn. And similar goes with Twitter also. You have to keep engaging with their posts. And of course, meaningful engagement. Whenever I'm saying engagement, it is meaningful engagement where you write something about that they have done or they have posted and how your comment is adding a value to it. So keep these things in mind whenever you are going to LinkedIn next time.
Perfect. Thank you so much for all those insights. And it has been an honor to be a part of this panel and to be a part of with all of you here. So I'm thoughtful, and I think that students have been and also like the faculty have been able to gain some of these points and could maybe transfer this knowledge further, and students can apply some of these points and get the outcomes they are looking for or looking towards, because there is a sense of doing things quickly in this newer generation and without putting the effort which is required in there. So if there is a constant effort, you will get to the results, and that's what experts are saying. Again, thank you so much to the panelists for being a part of this research symposium. And Dr. Kanan is again filling another form. So yes, thank you again for being here and helping all of us. And we will be able to see you again very soon and maybe talk to you on different opportunities. Over to you, Sonalika, now.
So much, Dr. Mohit, and thank you so much all the panelists. That was really interesting to know about your thoughts.
Thank you so much.
Different things. Thank you once again. And now I would like to welcome Dr. Raja Varman. So to expand further on the topics that were discussed by Dr. Nidhi and the panel discussion, Dr. Raja Varman is the Senior Bioinformatics Specialist at Premas Life Sciences and spends most of his time analyzing and reporting cancer genomic data sets from Illumina platforms in hospitals like Kokila-Ben and Adayar Cancer Institute, WHO. He has published his cancer studies in American Molecular Pathology Journals, Journal of Molecular and Genetic Medicine, and has trained several institutes in India, which were involved in major genomic programs like SARS-CoV-2 surveillance. He has also written several articles on diverse topics focusing on clinical informatics, comprehensive genomic profiling, nutrigenomics, and infectious diseases in different magazines, blogs, etc. He previously has had exposure to precision medicine and non-clinical sequencing data analysis while he was working with several bioinformatics companies. We are pleased to have you with us, Mr. Raja. Over to you.
Thank you so much for that very detailed introduction, and thank you so much for the given chance. Let me share the screen. Somehow I feel my topic has been stolen by the previous panelist. Keeping that aside, I might reiterate the same thing in a different form altogether. Let's see how much it reaches. Without any further ado, my name is Raja Varman Kittu. I'm working as a bioinformatics specialist in Premas Life Sciences. Today, we are going to see the technical skills as well as the soft skills which is needed to be in industry. Again, it's not specific to informatics. I would probably consider this as a recommendation for any technical, probably the IT or the bioinformatics together, right? Wherever the data analysis comes in picture, this can be applied. So three things we are going to discuss. First is technical skills, and then soft skills and the mindset. Pretty much all these topics are covered by the previous panelist, but I will be adding little more extra points here. So speaking about technical skills, there are some unique skills comes with certain people, and as a team, we need to have some specific skills. Probably I would say people specific skills as well as the team management skills, right? So currently we are going to see, first, let's focus on individual skills. Suppose the given problem Yes. So say in the informatics, generally in the IT related companies or the informatics companies, you'll be given a problem or will be asking to develop a tool. So those kind of situations, first we need to understand the problem, but basically what do you want to do? Either we want to do the analysis for the given data sets, or we want to build some software. And then we'll be going for the plan, like say the architecture, like clear plan on the architecture. The second one is what are the infrastructure we need, then how many manpowers we need, how many people, and where are the resources has to be put on, right? And the last one is time frame. And then finally we'll be deciding what are my deliverables and how faster I can give. So this is a basic benchmark, or probably the need or the workflow which has to be followed, right, in both the cases. So be it it's happening on the cloud or local, this is the ultimate goal for any given cases, right? Say we are going to develop a product, say be a software product which is being done in the local, or you're developing a product in the cloud, or you're developing a database altogether, right? Or there are some cases, since currently people are so much fond of going for artificial intelligence or machine learning, or probably the SVM is one of the classical machine learning techniques, neural network concept or deep learning concept. Any of those cases, apart from even the data management, first of all, we need to understand what is the given problem or the situation, come up with a detailed plan, and we need to know the logic as well. Before even starting any given coding, we need to know the logic. We need to convert the whole problem into the scripting language, by the way. We need to be very good at the maths as well as the logical thinking. Only then the scripting can be executed, right? And finally, once the execution is being done, we need to go for certain validation. So again, when I say validation, in the beta stage, we'll be doing some validation where the known data sets will be given, and we'll be checking, are we getting the expected results or not? Again, you might need to fine tune different parameters or specifications to reach the expected value, which confirms your product is good enough. And then the final minimum refinement, if it is needed, will be going for. This is specifically for product development. As far as analysis is concerned, it's slightly different. We'll be taking a different data set which has been already established or been published. We'll use that same data set, build a pipeline and confirm the results. And then we'll be using the real-time data set for getting more confident on that, right? As far as data management is concerned, we need to know the policies very well. First of all, like say, you are a kind of a service provider where you are building a data management policies or the products. We need to know the recommend better or their policies, and then we'll be building the tools. Again, they're also pretty much the same concept. You need to apply the logic, you need to execute it, and you need to validate it, and then you'll be giving the deliverables, right? It's pretty much basic benchmark, or probably the bottom scenario which has to be followed. So the key point here, first we need to be understanding is probably the scope of this particular given problem, and what is the highlight, like what do they want to achieve out of this whole problem or the product. And the last one is limitation, what you can give or what you can get out of the products, and what are things we can't do it. So the more we know, the better we'll be coming up with the better solutions. That's what the ultimate, the probing is one of the important key. This cannot be done in the middle of the process or the last minute. Rather, the more we understand from the client end, or probably it may be a data or it may be a product, the more we communicate and get the details from the given clients, the better products we can achieve. Right. So this is kind of one of the busy slide with scary key points. To keep it very simple, again, this slide is specifically for informatics, I would say. If you are an informatician, be it you are an analyst or you are a development side you are in, the basic thing which you need to be knowing for sure is Perl and Python. Of course, there are many cloud computing services, there are GUI-based web browser, there are multiple options are there. But to do a minimum file manipulation, even if you want to do a counting, it's not good to do using a simple Excel. So in that case, the basic level of scripting is always recommended if you are in an informatics field. Again, very basic knowledge for Perl and Python at least, right? And then before even jumping into the scripting, we need to know the schemes better. Like say, what is a given problem? Which are all, what are all the concepts I can apply on the given data sets? And then the systems. In this given configuration or given setup, will be able to execute or not. So we need to know the computation also better with respect to the scope. right? So Perl Python is kind of, I would say, if you are in the informatics field, then it is compulsory. And then there are next level of products where people are nowadays, not even nowadays, I would say down the line, 10, 15 years before also, people are more fond of coding in the cloud, right? So where Hadoop Scala is kind of widely used, if you get a time and if you are a tech savvy, so in that case, you are very much fond of learning the technical skills, try to understand how cloud computing is being executed, where Hadoop ecosystem, which covers almost the data management, product development, as well as the full-fledged product development can be learned in that particular platform, which you can try it out. Again, it's very, very advanced level, not for everybody. People who are fond of tech, they can get into this. And the last one, which I'm saying, is basically Nextflow. Why it's kind of trendy, and it's not something very new. It came from Java and then Groovy. It's a like third level of Java, I would say, at this point. But why it is important, currently most of the cloud solutions is getting developed using this particular scripting. So if you get a chance, again, if you are very much fond of tech learning, then this is a thing which you can try it out, as it is trending, right? So these are all not again compulsory. If you have your own, as I told, there are web browsers are there are Windows-based softwares are there are free open source tools are there are cloud computing services are there. Those things can be explored. This is for people who fond of learning new skills, developing new skills. As you have seen the previous, the whole panel was speaking about developing the skills. So if that is a concept, technical skills, if you want to develop being a core developer, then these are all the things has to be there. As an analyst, if you know bare minimum with the Perl Python, it's good enough. But if you want to develop something, it's better to have all these things in place. At this given point, again, down the line, in 2 years, this may even get outdated, because the new things, that the way IT skill requirement is getting probably moving is so faster, right? So in this case, today, this is kind of useful. You need to catch up with your skills always. That's what I'm trying to say from this slide, right? So switching the gear here, soft skills, as far as soft skills is concerned, almost people have told this. I'm just reiterating the same thing in a different way. At certain extent, you need to be having a technical skill for sure. But if you want to survive in the industry, the people skill is more important. Yes, you know very well, but if you don't know how to communicate better, plus you are not talking with anybody, you are always working on your own PC, it doesn't help. So in that case, people skills are very much important. How do you develop? You need to do enough of networking, which has been told before also. And you need to have good contacts. Again, don't run behind contacts. At least if it is reaching you, don't give up. Like say someone is coming and talking, try to develop a relation with them. Relation in a sense as a friendly relation with them, right? And the team spirit, what does that mean? If any given role is any given task for a team, try to distribute instantly rather than taking the full ownership. Try to distribute it so that everyone get a chance to utilize their skills, right? Plus, you will be also, if you're working as a team, obviously down the line, you'll come to know what are the individual skills as well, so that it's easy to distribute their own favorite skill sets. So that's one kind of thing which will be understanding in the down the line, right? So team spirit is one of the important thing which gives chance for the individual growth as well as the team growth as well. So that's one of the important thing which everyone should be understanding, right? And self-discipline, which of course people skill, basically if you are very extrovert, you don't want to work on this first point because you will be having this inbuilt, right? There's no doubt on this. But when it comes, introverts is kind of different. We need to be picking up. So otherwise it's very difficult to survive, right? And the second one, which is self-discipline. Why we are speaking about self-discipline? The whole, say you are working in an office where whole 8 hours you are going to communicate, or even if the clients comes in with the requirement, you are going to communicate with them. The communication is the key. Again, with respect to what are the words you use, what are the body languages you use. So you are almost communicating or conveying the message even without speaking. That's what the second point is, which is body language. Be it you are giving a talk, or you're sending a mail, or you're in the call, like say you are having even one-to-one conversation, everywhere, try to be with the full self-discipline. When I say self-discipline, you need to follow basic rules, which is basically, I would say, ground rules, which is these things. Try to be a try to have a pleasant conversation as much as possible, and try to spread the positivity as much as possible. And whenever you speak, make sure it's basically you're delivering the statement with full confidence, and the way you are delivering, people should get trust on you. So that's the ultimate purpose of communication all over, be it professional or non-professional. This should be the way, right? And if I want to put it in very simple term, try to write very clear, even simple context. So you don't want to be so much of using a jargons, like say very technical terms, even if the simple term would do to convey the message. So try to keep it very simple. Second is, when we are in call, even though the tone is very important, the pitch is very important. Why? People don't see our face. So in that case, we need to be very careful with what we communicate. So the pitch as well as the tone is very, very important in the calls specifically, right? And the last one is body language. This is mostly for if you are working on your own space, there is no issues. But when you are really working as a group or as a team, or even if you are in a meeting, it's very important to maintain a body language. Suppose people can easily figure out who is basically focusing and who is interested and not interested, right? So in that case, body language is one of the important factor. In fact, even if you are speaking and if your message is conveyed, and it may even reach in the right term, but body language, if it is wrong, people get it very totally different term altogether. So in that case, these are all the key points which has to be followed. These are all the basic, I would say, basic recommendations, right? So I think this has been told before, right? I'm just putting in a different context in the communication as well. So we need to know, when I say scope, highlight, and limitations, basically, given communication, say there is a communication which happens, we need to grasp the concept quickly. So suppose if someone comes and reaches you for some topic, try to get the like say crisp or probably the purpose behind this quickly, and highlight only the advantages, plus try to know the limitation. This I told for the technical as well. This is applicable for the simple communication as well. So rather than wasting time on unrelevant topics or like say unwanted things, rather understand the concept as much as possible. Try to ask as much as question in the first instance, get a clarity why this particular conversation is happening. Straight away get to the point, right? Plus you try to know where it is leading as well as soon as possible. So this is one of the effective way communication. This should be followed for sure, right? As almost we are in the last part, where the mindset people have already told also. Since we could able to see in the panelists, there are different kind of there was a consultant, there was a doctor, there is a CEO, plus there are informaticians. So we could see almost different background people in one particular panel which we saw. Almost everyone had kind of same mindset here. So everyone was saying, try to grasp whatever comes to you, don't miss out any meetings, LinkedIn, be active, communicate effectively. So almost they told everything. So all things comes out of one basic thing, which is basically they should have a very positive mindset, plus the eagerness to learn with full passion, right? So here I just brought out a little bit of plot where try to have an experimental mindset and be ready to take risk. Again, not extremes, at least the basic ones. You should not be giving a second thought, because as a student level, it's better to take a risk at the early stage, because down the line you may not even getting a chance to take a risk, right? So this is a time to take a risk. It's better to take and try to learn things quickly. Again, different people have different learning skill sets. Probably they may take a time, but as a motto, try to have a motto to learn quickly. So because that is one of the key factor to improve the skills faster, right? And if anything comes new, you can give a thought, get to know about pro and cons for sure. But adaptation is one of the important key factors in the industry, because every company is not going to work in the same way. So it's better to have a quick adaptation mindset. And then passion driven, almost everyone will be having this. If they enjoy the job which they do, in that case, it's by default, it comes in. If you're not getting it, obviously you need to switch the job. So in that case, what I meant, if you enjoy the job, you don't want to struggle more on this. So passion driven should be there if you want to see a growth in your career. And sprint, so obviously it will be coming in picture if everything is in place. Finally, the commitment. So if you miss any of these things in the middle, so probably the commitments should be given to all the segments which is being shown here. So that's the one which is going to be driven. So it's not like 2 days you are doing something and then you are leaving it. Rather, try to have a long-term commitment with whatever is being mentioned. So these are all the key, probably the parameters or the mindset we should be having it to, rather than saying survive, to have a better career in the industry, right? This can be applicable for research as well. It's overall, it can be applicable for both, but the skill sets which has been mentioned or the key points which has been mentioned is very, very important if you want to achieve something in your career. So with that, I would like to close the talk. Let me see if I have any questions, even though it's a very general topic.
All right. Thank you so much, Mr. Raja, for such an insightful talk. Yes, we do have some questions with us that I would like to ask. So one of our participants is asking, after acquiring technical skills such as Perl or Python, I would like to work on a project using these skills. What are some of the ways that I can showcase my work?
So about the projects, again, we need to know, as I told before, we need to know what kind of project it is. Plus, we need, so again, scripting wise, you can always choose any of the scripting. You have C, C++, Perl, Java, you have multiple things. We need to understand what is the problem is, plus which particular scripting will be best suitable for the given, probably to solve the problem or to develop the product. In that case, the more understanding we should be knowing the limitations as well as the highlight of the individual scriptings. Probably the programmings.
I hope that answers your question. And how do you network with experts in field who are difficult to communicate with? Perhaps they're not interested, or how do you convince them?
Okay, so rather than here, there is no convincing. Rather, as she have told very nicely, I think. So don't send even the LinkedIn request. When you say don't just send a blank message or blank invitation, you add in your key points, probably the catchy phrase. When I say catchy phrase, it's not like you are putting a one-liner to impress. Rather, you keep something, give a topic which is common between both of them. So of course, it may take a time. Some people are not open to start a conversation in the first go, right? If you really following someone, probably you are having passion to follow someone, keep in touch with them. It's not like you need to communicate down the line. Anyway, you'll be meeting them if you are following them seriously. So it's not like you need to force them to get them in your radar. So you just follow them and do your work. Down the line, anyway, you'll be reaching them for sure. I mean, be optimistic.
Yeah, that's a nice answer. All right. And how do the skills acquired differ while working on a project individually as well as while working in a team? That's a nice question.
Yes. So first of all, this mostly. So let me give a very simple examples. First of all, when the project comes in, obviously the teams will be divided individual based on their skill sets which they have acquired before. Even if they are not well trained with the skill sets, the team, the individual division or probably the job distribution which will be happening after the project has been settled. Say the business analyst who collects all the requirement from the clients, then there will be a job distribution meeting which happens very clearly with all the team members, where based on their skill sets, the problem will be different. Probably the workload will be divided. So that's one chance where people even switch, or people wants to learn something new, they also get an opportunity. So it's kind of well organized in the MNCs. When it comes small startups, as people have already told, you will be wearing multiple hats. You will be responsible for multiple things. So in fact, you will be putting your hands almost everywhere.
Right. And do technology soft skills set you apart from the crowd?
Let me change the perspective here. The crowd will be anyway will be following the trending part. That's not sure. Today one script, tomorrow one script. It will be anyway always following, right? So in your case, at this point, like today you decide, like today you are a student. So today you try to pick one skills and try to become a master down the line. Say at least within 3 years, right? Why I'm saying this? Because anyway the trend will be keep on changing. If you know one script today, it's very easy to adapt new one, or you can pick up. So whatever you learned in these 3 years can be converted to the new things which is coming. So rather than catching up on something which is basically keep on running, focus on something at this point for some time, and then you switch. It will be easy transition basically.
I hope that answers your question. Okay, we have a lot of questions today. How can soft skills provide an edge to a person in the industry, and what could be some of the tips to improve them? Yeah, we did cover it. But yeah, if you want to add something?
But no, I think it's covered. It's fine. Let's.
Yes.
Yep.
Okay. And as an introvert, how can I keep the conversation going while networking?
Yes, so this is very difficult for sure. But being introvert is fine. Still, you are in college and school. But when it comes industry, you will be missing out if you can be an introvert. There is no doubt. But when it comes something to your table, like if example I'm giving, you running behind someone and then saying hi is not needed. But at least if you get a hi, try to say hello. That's minimum requirement which the introvert should be doing. Plus, if someone who is matching your wavelength technically, right, be open to discuss with them more. So that's one of the thing which I can think about at this point. Introvert is one of the like characteristics which is a born skills, but there are certain topics which you should be able to discuss with people who have same interest.
Yeah, that's a nice point. And amongst technical and soft skills, which is important, which is more important?
So at the early stage, technical skills are very important, which is 90-10. And down the line, when you are well experienced with the technical, the people skill should be like 70% important.
I hope that answers your question. And do you want to add something for our participants towards the end, who are in their graduation and post-graduation and are dealing with their busy schedule of academics, but also want to upgrade their skill sets, but don't have the time or are confused which resources to follow, what to do, how to do, and actually are confused which specific skill set to pick? They have no idea that how can they?
Yes. So some skills, again, speaking about academic, there is a really very good gap, or probably the very long gap from the academic to industry. How do you fill the gap? Of course, the academic syllabus where you cover for, like say the academic syllabus is only for the course, right? You may grasp the basics for sure. Not everyone is picking the life skills over there. When I say life skills, it means the skills which will be useful for your career and the life, right? So in that case, these bridges can be closed by taking extra courses. It might be either probably, as you can say, the biotech is giving multiple courses where you get to have extra skills which your own classmate doesn't have. Simple example, in your classes, 60 people are there. Not like everyone knows how do we analyze the NGS data example, right? So there are nearly 16 to 20 different pipelines which has been developed in the cloud, which Biotech is having, where every individual pipelines has its own tools and different parameters. You can't pick up everything in your academic. Academic can give you only the introduction to certain areas. They can't go in deep any of those things. So if you want to develop your skills, you need to come out of the academic syllabus and learn more outside the classrooms, which is very important things. That's how you'll be differentiated from others. Again, there is no limitations here in the learning. The more you learn outside your class is going to be helpful in your lives, like basically in the career. So in that case, try to do more. This has been already told before. Also, I'm reiterating: conference, workshop, classes. Try to attend more. This improves you to pick up your skills better.
Right, Mr. Raja. Your answers were really helpful, and I thank you once again for being a part of the symposium and delivering this very insightful and helpful talk. And with this,
Thank you so much for the chance.
With this, we would now like to proceed ahead with our next speaker. So far, we learned about bioinformatics research in Indian industries. Now let us see what is the present status of bioinformatics in Africa in terms of education, internship, job opportunities, and research. I would like to welcome Ms. Padmayo, who works as the bioinformatics team leader at the Center for Proteomic and Genomic Research and works 50% of her time as the bioinformatics coordinator at Diplomics, a CPGR grant program. Her main role is in overseeing efforts to develop different methods to understand data generated from the different omics fields the organizations serve. She also enjoys learning new concepts from managing these projects. In these roles, she has aided or led in building successful products and complete projects. This has refined her marketing, project, product, and team management skills. She has enjoyed and has found that her strength is in diligently stitching the different components needed to deliver successful services. She is also pursuing her PhD program, which looks to explore factors that contribute to developing competencies in the evolving biomedical field that experiences a constant change of scope in work like at the CPGR. Ultimately, to improve on products and outcomes which attracts funding in business to continue to boost with a particular focus on work done in Africa. That was indeed an introduction though. Now I would like to welcome Ms. Padmayo. Over to you.
Thank you very much. Let me just share my screen. Can you see my presentation?
Yes, we can see us.
Perfect.
Yeah, perfect.
Okay, so thank you for that introduction. So I'll get right into it. I'll start off with the CPGR and I'll explain how the two other organizations fit into this. So here's a summary of where everything sits. So the Department of Science and Innovation in South Africa funds different programs. And one of them is the Technology Innovation Agency, which funds the CPGR, which is my primary employer. And the other two organizations that I'm going to speak about are Diplomics, which is a program. So the CPGR hosts that. And the CPGR also has a subsidiary startup, which is called Artisan Biomed. So in these different organizations, that's where the different sort of titles come out. So in Diplomics, I'm a bioinformatics coordinator. At the CPGR, I'm the bioinformatics team leader. And at Artisan, I'm a bioinformatics consultant. So all of these, although are connected, are quite independent and have different visions. So it's quite interesting to be in either of them. And then I'll touch a little bit more about what I do in them. I'll strictly be focusing on the bioinformatics aspect of each one of them, but they actually do quite a lot. So if you're interested in learning a little bit more about them, you're welcome to contact me. So I'll start off with the CPGR. So the CPGR stands for Center for Proteomic and Genomic Research, and it is a nonprofit organization that I mentioned is funded by the Department of Science and Innovation in South Africa through one of the programs called TIA, so that's Technology and Innovation Agency. So we sit in Woodstock in Cape Town in South Africa. It works mainly in the model of a contract research organization. So focuses on different services and training. It is omics focused, so omics research and life science innovation in Africa. So the main omics fields that it focuses on, it offers comprehensive services in genomics, proteomics, transcriptomics, metabolomics, and bioinformatics. So it houses quite a lot of world-class infrastructure. So the organization itself has a wide range of instruments. So if you'd like to know a lot more about the different instruments that are at the CPGR, they have an Unpacking the Toy Box series. So if you go to CPGR's LinkedIn, you'll be able to follow that series as they explain each of the machinery that they have. So the reason why I'm mentioning this wide variety of capabilities is that along with that, we offer bioinformatics support. So in terms of the work that I do for the CPGR, it's a sort of in-house support for all of the wide range of services that could be offered through all of this machinery. Okay, so CPGR Bioinformatics, an overview. So as I mentioned, the projects that happen in the lab can come through our bioinformatics. So it is an optional service. So if you need bioinformatics support for any wet lab services that you get, that's what we're there for. We have different clients. So the clientele is from industry and academia, but mostly from academia. The clients are researchers, are students, consortiums. So I gave you an example of a consortium that we're a part of. So it's a big consortium called MADCAP, so Men of African Descent, Cancer of the Prostate. And this is a custom array that was created. It is a project that is funded by the NIH, so that would fall under a consortium. And we also have commercial clients that are either doing research to better their product or their offering, or they're using our lab to run some of their services. So in terms of the bioinformatics team, because we have a wide range of projects that come in, although we have standard pipelines, we tailor make each product to each client, whether they're from research or they're a commercial client. We will tailor make the solution that we make for them. The reason why we do this is because, one, the clients are different, two, their objectives are going to be different. So even if it's an RNA-seq project, each client will want something different. So I will explain what I mean now after I explain the makeup of the bioinformatics team. So the bioinformatics team is made up of bioinformatic analysts. So the skill set in terms of the people that we hire is based on the analysis that we do. So it has to, when we're hiring someone, it has a focus on genomics and proteomics. The CPGR being a CRO, so a contract research, it means that you have more defined turnaround times because it's a service that you're offering someone. Unlike in academia, where it's a research project and you have, for example, a year to complete your master, the turnaround time is not as strict and as defined. So the core skill that we need for our bioinformaticians, so this will differ from organization to organization. It will differ from industry to industry, depending on what you're doing. So when I'm talking about core skill set, I'm talking about a core skill set that's needed for you to be a bioinformatician at the CPGR. So here, our core skill set when we're hiring someone is programming. Why? Because it's the main skill that's needed to implement the solutions based on the different services that we offer in the lab. Our core language, we use Python. Why? Because it's easy to learn and easy to use. We try to have a standard language that everyone uses. If you have another language that you prefer or you're well versed in, we still welcome you because that may be useful. Additional fundamental requirements is experience in R and Bash, because we also do use that, and some knowledge of biology. Why? Because most projects are very holistic. You're not only going to use Python. Sometimes you'll need to use Bash. Sometimes you'll need to use R. Sometimes what's really needed for the project is a good understanding of the biology. So all of these are important because we also collaborate with the wet lab. So that's another good thing about the CPGR, is that all of this is housed under one roof, from the wet lab to the analysis. So a client will have from start to finish, and you can actually go to the person in the lab and they can even tell you that. You can say, for sample 2, I'm seeing that the QC is looking a bit fuzzy, and then the person in the lab can tell you that, oh, you know what, these are the QC metrics, and when I was handling it, they didn't send, the aliquot was way less than the other ones, and I was just trying to make it work. So it helps you understand the data a little bit more. So all of these skill sets are needed because we apply a holistic approach to solving problems. And then the Bioinformatics platform or department itself, we work collaboratively internally. So as a team, we collaborate together if someone is experiencing certain issues. That's why I noted that it's important for us to at least have common programming languages or skill sets so that internally we can collaborate, and externally we collaborate quite a lot with the clients and with the wet lab. We have to make sure that we can integrate to assist each other. So whatever system is in place, it's important for us to have a common language, as I said, so that we can integrate into the same system. One of the things that our platform or our department has to consider is storage. So we have a lot of clients that come into the CPGR, we store their data. We have to consider how we're managing storage. We have to consider connectivity. Sometimes we have cloud solutions, or we have to upload or download databases. So connectivity is a big consideration. We have to consider high performance computing. So as you all may know, that some tools are really computationally intensive and you cannot do it on your laptop. You would need a high performance computer. You would also have to consider data sharing platforms or methods, because you also want to integrate externally into a client system because maybe they receive data in a certain way. So you would have to consider that. A very important one is you have to consider code hosting platform. We use GitHub. So you have to make sure that it's a very standardized system at the CPGR. So they are ISO accredited, which means there's a standard way that you do everything. It makes sure that you can always backtrack if you have a problem, that things are reproducible. So to fulfill one of those requirements is we put all of our code up on GitHub. It makes sure that anyone else can come and replicate it. So yeah, that's an important consideration. You also need to consider tools and software and related licenses. So sometimes you only need a license for one particular client. So you have to think that through to say, how will I complete this project? So those are the main considerations when setting up a bioinformatics platform. And then bioinformatics as a service. So this is quite different from academics. We've had bioinformatics analysts who've joined the team, and they're testamented this to say that it's a very different environment when you are now offering bioinformatics as a service. It's a bit more refined. You need to be a bit more sure about what you're doing because it's helping inform someone else's project or someone else's service. So the next line where I say a service means an offering that requires a specific result. So when a client comes in, they want something specific at the end. So whatever you're doing, you're trying to get to that point. In research, if you sort of reach certain blocks, you can, take your time to think another way to do it or to drop it. When you're offering it as a service, the client expects a result at the end. So you'll find that bioinformatics as a service, you employ quite a lot of business principles with a high emphasis on human capital. So in terms of my team, I gear it mainly towards soft skills. You'll find very quickly when you're offering bioinformatics as a service that your soft skills are going to be such a big component. So for example, something as little as communication, whether you're communicating internally as a team or externally to a client, you need to communicate effectively because it reduces scope creep. So scope creep is the project slowly getting bigger and bigger because people are slowly adding requirements. So you'll find with clients who are researchers, they're quite excited about their project. And what they'll do is they'll come and say, oh, can the data do this? Or can the data do that? Or can we do this? And can we do that? And that increases the budget, the turnaround time. And it just prolongs the project. So if you learn how to communicate effectively, then that sort of reduces scope creep. So you'll find that a lot of soft skills have to come in with your hard skills. So you have tighter, more defined deadlines. So there's a little bit more pressure in a service environment. So I think this environment works better for individuals who enjoy solving like varied problems. So if you enjoy solving, some people enjoy looking at a particular protein for the next 20 years, and there's absolutely nothing wrong with that. Some people like looking at proteins today and looking at genes tomorrow, and tomorrow looking at a specific organism. I think this environment will work well for the latter, someone who likes looking at different problems and solving them and being able to move on. Okay, in terms of service output, so it's dependent on the client's objectives. So at the end of the day, when we're giving a client a result, if someone comes in, usually this is what we give them. Either we're giving them data because they want the analysis done to a certain point and they're going to do downstream analysis by themselves. So some people might just say, give me my raw data. Some people might say, QC my data and then give it to me. Some people might say, analyze the data up to a point where you have your OTUs and then give it to me. Or sometimes we do analysis and they will interpret the results themselves. Sometimes we'll do analysis and training because they would like to analyze future data sets by themselves. So that's also for researchers who are continuing with their project. They might want you to do it the first time, and as they get more data, then they might want to do training so they can do it by themselves. Training, so you could train someone because they would like to analyze their own data sets from scratch. Collaborative, which means that the client would like us to significantly contribute to their project. So they would like us to analyze and interpret and actually give results and then include us in papers and so on. Sometimes it's consulting, so they would like to build it themselves, but just need someone as a sounding board or someone to check in with. So they will purchase, let's say, 8 hours, and then 2 hours a week, we have a meeting where they take us through their solution. So here is just an example of training. So we get a lot of inquiries about RNA-seq, and we thought it would be a good idea to have an educational webinar where we just take people through basics of the analysis so that they have an understanding of it. And sometimes when people understand it better, then they can say, oh, you know what, this will be too much for me and I'd rather you do the analysis. They can make an informed decision about how you want them to help you. So from this educational webinar, some people will come up and say, actually, this is too much and I'd just like you to do the analysis. Or some people would say, oh, this looks manageable and I'd like to take a stab at it. So I would like to take this consulting hour approach where I go and I do it, and then we meet every week if I'm experiencing problems. So those are the basic service outputs at the CPGR. So now I'm going to move to Diplomics, where I am a bioinformatics coordinator. So Diplomics, as I mentioned, let me just go back to it. It is hosted by the CPGR and it's an infrastructure program. So the approach involves mainly collaboration, capacity development, and optimization. And this requires, amongst a lot of things, but partners, experts, and cooperation from my point of view. And this is just a summary of what Diplomics does. So our aim is to build world-class omics infrastructure. And how do we do that? It's either we're placing instruments, and this is the impact that it has. And these are the impacts, whether they're going to be direct, induced, indirect, or cumulative. And over time, you can sort of tell like an indirect impact and sorry, I've got a chat. An indirect impact. And then enables access to students and researchers, and that's done in various ways. So in terms of training, in terms of high visibility, high impact projects, and then the different impacts are, you get world-class reputational gain when you do high impact, high visibility projects. So it speaks to our capability to do things. So if I link it back to what I'm currently doing for my PhD now, all of these, as I said, are, although they're connected, they have different focuses. So maybe just to speak about high visibility, high impact project for my PhD, where I speak about once we have a good reputation of doing reproducible and good doing research that has good outcomes, then we can have that reputational gain. And over time, that will attract funding or that will attract business. So that's like a cumulative or a long-term impact. But as time goes on, you'll see the impact of it later. So I'm going to speak about Diplomics again, mainly with respect to bioinformatics. But if you guys have any questions that relates to anything else, you can get in touch with me after. So Diplomics, there are 3 omics fields that it branches into. So genomics, proteomics, and metabolomics, and bioinformatics. Each is informed by a working group of experts, and each has an overarching domain expert who is the coordinator. So for example, I am the bioinformatics coordinator, and there is a bioinformatics working group that has a bioinformatics chair. in these different branches, so genomics, proteomics, and metabolomics, and bioinformatics, who've had many successful initiatives. So these initiatives range from either training, instrument placement, or support for high impact initiatives. So you'll find here that, for example, genomics would have a training outreach, or bioinformatics would have a training outreach, and then you develop skills or you increase awareness of a particular field or a particular technology, and then there are different types of impacts that happen over time. And then I'm going to talk about bioinformatics. So information gathering is key to all of our successful initiatives. So before we make a decision, you sort of research and say, maybe this is the need, or this is the current situation. And then you sort of now research a little bit more to be what is the best way to resolve it. So we would use this working group that has all of these experts either as a source or as a soundboard or as a source of initiatives themselves. So of the current needs with respect to bioinformatics, we have 4 main areas of interest. So establishing bioinformatics as a service, improving bioinformatics at project design, enabling or improving bioinformatics training, and inspiring the next generation. So all of the above help improve directly or indirectly overall outcomes. So of the list that I've said here, I'm just going to expand a little bit more about it. So bioinformatics as a service. So we launched Clarity, which is a bioinformatics marketplace. It hosts expert bioinformatics consultants who are managed by an experienced concierge. It is virtual, therefore it's accessible across the globe. Currently, it's subsidized to the researchers to help establish it as a service and its worth as a service. So it goes back to what we're talking about, building a reputation so that it either attracts funding or attracts business. So that's one of the main reasons why it's subsidized to researchers. We have a strong emphasis on good project design because we have found in terms of when we are doing information gathering and we're really talking about what are the struggles people are having with bioinformatics, is that when they're now doing the analysis, they feel that the bioinformatics is failing them. It's not necessarily that the bioinformatics is failing, is that if you put good data in, then you'll get good data out. So if your data is not, it's not that the data is necessarily bad, but it's not the right data to answer your research question. So for example, your data may have passed in the lab and the QC is good, but if you're trying to do whole exome sequencing, or you're trying to look for a variant and you have RNA-seq data, it won't answer your question. So it's the same way, if you haven't designed your project to get the data that will help you answer the question, then you're always going to find problems. So that's why we have a strong emphasis on good project design. So currently, of the projects that have come through our marketplace, these are mostly projects after data has been produced, which means that we would have already passed the stage of project design. So we found that clients may have tried to take a stab at analyzing their data and have found difficulties, and then now they'll go and find a service provider. And in that, other than just analyzing, we're really trying to roll back and get the best that we can out of the project. So again, this is also evidence supporting our initial finding that project design is a really important part of design. Considering bioinformatics at project design is very important. So bioinformatics as a service, we also get involved in high impact projects. So the nanopore projects that we currently have now, we have bioinformaticians from our marketplace working with the different partners in the Diplomics network. So that's how Diplomics handles bioinformatics as a service. So speaking a little bit more about bioinformatics and project design, so 46% of projects fail because of poor project design. Why? They did not properly consider the needs for data analysis. So if you want to look at, I'm just going to use an analogy, if you want to count how many apples there are, that means that when you go to the market, you must get apples so that you can count apples. So sometimes when you're designing a project, what you need is replicates in order for you to have confidence in your data. And if you don't put in replicates at the beginning, then you will not have confidence in your data at the end. It sounds like a very simple thing, but it's a small gap between like molecular biologists and bioinformaticians. So we strongly encourage that bioinformaticians are involved at the beginning of the project. So some of the efforts by Diplomics have been mainly in awareness, because when we do speak to researchers or students, a light bulb sort of goes off where we talk about, project design. So just spreading awareness that it is important to, or they don't know that, it's something that they should do to involve a bioinformatician at the beginning when they're planning out their project. So awareness. So last year, not this year, we had a little campaign where we released some lighter adverts just to say, Patrick Shaw's Do It Right the First Time. So as I was explaining some of the projects that we've had, that after the data was already, they were trying to do patchwork, where we're trying to roll back and trying to get the best that we can out of the data. And then another one is a project without bioinformatics. It's like a car without wheels. It's not going anywhere. So if your car does not have wheels, it's not going anywhere. You can create your project, you can run the samples. But again, if you want to count apples, you have to go and buy apples at the market. So this is also just an explanation of how our marketplace works from our brochure. We sort of welcome you to it, give you why it's important, explain our process, and then tell you what bioinformatics is, and then just say, let's work together. Okay, and then bioinformatics training, we do this mainly through partners. We found that collaboration works well. If we don't have to reinvent the wheel, we won't. There are already partners that are doing it quite well. And the aim really is to enable through facilitating. There are two main groups that we are currently focusing on. So it's the bioinformaticians themselves, because maybe they may need to upskill or get a new skill, and the wet lab scientist. So it's actually important for my molecular biologists, for example, to have an understanding of bioinformatics. So it might be good and well for us to say they should include it at project design, but if they're also not aware of, downstream what needs to be in place or how things work, then although they know it's important, they don't know how to consider it. So it's also important to train, even if it's going to be a basic training of bioinformatics, even if they're not going to do their bioinformatics by themselves, it's important for them to at least have an idea of how everything works. That way, then they can best engage a bioinformatician to get the best out of them. They will, for example, already know that when I meet my bioinformatician, these are the things that we need to cover, or this is the critical information that I need to give them. So here's just two examples of training through our partner at Thermo Fisher, and training through partnering with ICGV. Okay, and then we're going to talk about future bioinformaticians. I'm just checking my time. I'm out of time, but I'm going to be done soon. So future bioinformaticians, so these are high school outreaches, so human capacity development. So we focus a lot on low resource schools, just to stimulate interest in science and negate quite a lot of misconceptions they have about science, and also to give them exposure to this, so they might not even know certain things are aware. So we take them through very high level scientific concepts, and we end, for example, in them doing like a mock code, and then I tell them a little bit more about bioinformatics and tell them all of the other different science fields that are involved, from example, from start of strawberry DNA extraction to the end. All right, and then we're going to go into the last one, which is artisan biomed. This is mainly focused on precision medicine. One way to think about it is that all of the research that is being done is in order to inform solutions. So Artisan sort of takes it from the step where they're now utilizing these solutions, but they have a focus on precision medicine. And they also have a focus on people of African descent, because as we know from research, African populations or genomes are underrepresented in research. So that's why the focus is there. And it also better serves their population group in South Africa. So precision medicine gives better insights to disease, and that's another reason why artisans sort of focus into this, better insights into disease, in particular for people of African descent, because if the research on people of African descent is underrepresented, it means that the solutions that come out do not best serve them. So they also have a training arm to them as well, where they want to empower clinicians and medical experts and different stakeholders in precision medicine. So here they're creating products for consumers and clinicians, and it's usually a commercial product. So I'm just going to go into a little bit more about the bioinformatics with respect to Artisan. So it is going to be the same skill set that we use for the CPGR, but you have to refine it to make a commercial project. So initially you're using it for researchers. So usually like a plot is the product that you're giving them, or data, or a bar graph. But with Artisan, because it is a product that's going to a client, it means that if it's going to be a report, it's going to be a more refined and commercial report. So in addition to the programming skills, you have to have additional skills that help you make it a product. So for example, additional skills can be outsourced and easily integrated. It could be a user experience engineer who comes in. And the additional skills as well are mostly non-computational. So sometimes you just need a clinical geneticist who's going to come and better inform your product. So the initial, as I said, the initial considerations are similar to the CPGR. The pipelines, everything that you're doing is centered towards a product, right? So you have to make it easy to use. It has to be well documented. It has to be easy to update. The speed and the storage and the quality are critical. So it's very different from academic research, where, for example, if you have a variant caller, you can use an open source variant caller that's going to take 2 days to run, because 2 days in 2-year masters is not a significant amount of time. But if you have a client using an open source, 2 days is like already too much. Someone is waiting for their result. So speed, storage, and quality are critical. So there's a wide range of products and services. It's simple as a COVID test. And although the testing may be simple, providing it as a service is what's complex. So I'm just going to give a quick example of one of their products, which is Be Happy to Be You. It's the most comprehensive ancestry test in Africa with an international client base. So it's forward-thinking. It's a forward-thinking effort because they would like to use the rich informative data to give further insights to people. So I'm just going to go through that. So this is actually an extract from my report, which shows my ancestry. So here, the bioinformatics considerations, as I've mentioned before, are going to be, you have to test. It's going to be a complex pipeline. You have to test it. You have to manage quite a lot of things, computing power, analysis, and all of those things. And then for the last slide, you're going to try to extract as much reusable information as you can. So e.g. with this ancestry report, because you're using the GSA and it has a lot of informative markers, you can use some of the pharmacogenomic markers to then give insights on drug usage that's related to, for example, in this one, COVID. So although it's the data that comes is rich and informative and it's complex to interpret and report, Artisan has had such a great start by taking time to understand all of these complexities. All right, thank you.
Good morning.
Any
and thank
questions?
you.
Welcome.
We are unfortunately out of time for questions, and we need to move to our next speaker, but thank
No
you
problem.
yet again for being here this morning. Bye-bye. Thanks.
Bye.
Well, good morning, and thanks to the global community again for getting us rolling for the day. And we're now going to roll into our Americas session, leading to another amazing slate of speakers. Yesterday, we really enjoyed several surveys, and maybe get to that yet again today. I want to welcome everyone to the Omics Research Americas session. And this is a time when we come to discuss the advances in biomedical research and all the education, hearing from experts that represent our community. In the US, we've been actively building out a network of experts that can help lead the way for young researchers and students to utilize omics technologies and the data science in biomedical research. And our first speaker comes from a background in pharmacy and will be speaking about drug design and the importance of omics technologies in this type of translational research. He's a professor of medicinal chemistry in the School of Basic Pharmaceutical and Toxicological Sciences, College of Pharmacy at the University of Louisiana. Sitharama has obtained his PhD in 1994 in molecular biophysics. So that is an amazing accomplishment. His research interests is in the area of peptidomotic, I think I'm going to mess this up, peptidomotic based drug design as a promising approach for developing therapeutic agents for autoimmune diseases, inflammatory diseases, and cancer. Dr. Joyce, I'm really excited to hear your presentation and correct all my bad pronunciations. The floor is yours, sir.
Thank you. Can you all hear me?
A little bit more volume would be great.
How about it now?
Yeah, a little closer like that would be wonderful. That would
Okay.
get us going.
I will try to come closer. I'll share my slide. Can you see this now?
Yes, and you'll want it to go to slideshow mode.
Yeah, I went
Go to your
there.
slideshow menu. There you go.
Is it visible now?
Yes, there you go. Perfect. Absolutely perfect.
Yeah. Thank you so much. I would like to thank the organizers and introducing me and giving me an opportunity to speak here. What I'm going to talk about is kind of a rational drug design. I'm not going to talk much of the bioinformatics here. You will see the end product here. Some of the information was obtained from bioinformatics. So my research interest is, as already introduced, grafted peptides for inhibition of protein-protein interaction. So I work on designing some of the molecules to target protein-protein interaction here at the School of Pharmacy, University of Louisiana, Monroe. So just a disclosure, some of the molecules I developed have applied for a patent. One application is already approved. Just to give a brief review of drug discovery, we start with the medicinal chemistry. It could be from natural product or rational drug design. And we go to test them in the test tubes and then in small animals. If we get positive results, we go through the toxicology, formulation, and then apply for investigational new drug application. And then the drug goes through the clinical trial. If it passes, approved by the FDA. So you can see that the drug is here, I am somewhere here. So I'm at the basic part. Let us look at the protein-protein interaction. So proteins, you know that the end product of the information pathway. So you do the bioinformatics and we work on the proteins. It is known that 50,000 different proteins work in each cell in human body. And you can imagine it's 50,000 proteins. If they interact with one another, there are nearly 600,000 protein-protein interactions that carry out any cellular process that is happening right now in your body. So that is an estimate. So if you look at the protein-protein interaction kind of map, this is just one map. You can see there are a lot of interaction between these proteins. So what we are going to look at is when this protein-protein interaction is deregulated, it results in the disease state. So we modulate the protein-protein interaction by inhibiting them. I'm going to talk about the epidermal growth factor receptor protein-protein interaction here. The human epidermal growth factor, there are 4 epidermal growth factor receptors known: EGFR are HER1, HER2, HER3, and HER4. So these are kind of a membrane tyrosine kinase receptors. Upon binding of the ligand, they undergo conformation change. When they undergo conformation change, they dimerize. That's why the protein-protein interaction takes place. Upon dimerization, they undergo phosphorylation and send the signal for cell growth, differentiation, and so on. So that is a normal process. But when this normal process is deregulated due to mutation, our overexpression of these receptors, more protein-protein interaction takes place, and then that leads to uncontrolled growth of cell, which is cancer. So EGFRs have important in cancer. Among these, HER1 to HER4, HER2 doesn't have any known ligand. So since we are talking about cancer, I just want to show the statistics. Basically, you can see on the left side, the highest number is breast cancer, followed by prostate and lung and colorectal. But if you look at the estimated death, lung cancer is the highest, followed by colorectal, pancreatic cancer, and breast. So since the number of deaths is high, we need to find new ways of developing drugs for cancer, because current therapies ultimately within 2 to 5 years develop resistance, and some patients don't have much time. So if we look at the information here, this information came from Bioinformatics. As I said, I'm the end user. So they studied a lot of information in patients about different types of lung cancer. Among them, 85% is non-small cell lung cancer among the lung cancer. And then they look at the genetic mutation and the genetic drivers that actually drive this cancer. Large part is still unknown in non-small cell lung cancer, but the remaining part, KRAS and then EGFR, these are well known. There will be overexpression of these receptors or mutation in these receptors or deregulation. And then we have HER2, ERBB2, BRAF, and so on. So what I'm going to look at is EGFR and HER2 today. It's one of the major drivers of this cancer. So if you look at the structure, these are the extracellular domain here, transmembrane domain, and the kinase domain. The 4 receptors are shown. If you look at HER1, HER3, and HER4, they have ligand binding site, whereas HER2 doesn't have any known ligand, and the ligand binding site is not that open. So in another case also, HER1 and HER2, HER4 are kinase active, but HER3 doesn't have much kinase activity. So that indicates that one doesn't bind to ligand, other doesn't have much kinase activity. That means these receptors have to work together in an orchestrated process. That is what you call protein-protein interaction. And these receptors, the extracellular domain, exists in closed conformation or open conformation. You can see the closed conformation here. I just give the example here how the domain 4 and domain 2 interact with one another, blocking the dimerization arm. We call this dimerization or socialization arm. Once the ligand binds here, it opens up. You can see that the dimerization arm is open. Now the two proteins interact with one another, giving the signal. So that's what you call protein-protein interaction. So now what is the clinical importance of this? These dimers and protein-protein interaction. HER2, HER3 dimerization is important in 30% of the breast cancer. And EGFR, HER2 are also overexpressed in 30% of non-small cell lung cancer. If both the dimers are present, then you can see that the survival rate is very poor in non-small cell lung cancer. In some cases, as short as 9 months, if both are expressed. So remember that EGFR, HER2, HER3, protein-protein interaction is very, very important. How do they measure the status of this? Of course, they measure the overexpression of proteins or overexpression of genes. So that's where the bioinformatics come into major play. And they classify them 3+, 2+, and 1+, depending on positive or negative. Now, what is the clinical importance of this protein-protein interaction? We wanted to check that EGFR, HER2, HER3, as well as EGFR. We took the tissue sample from the cancer center here in Shreveport, and then did something called proximity ligation assay. In this assay, if the two proteins are close together, very near to one another, and they're interacting, we can direct primary, secondary antibodies to them. And the secondary antibodies are actually tagged with DNA fragments, which can complement to one another, and then they can do rolling circle amplification. So basically, and then detect using the probe, DNA probe. In this case, it's a red dot. So basically, if the two proteins are close to one another interacting, you can detect them using this assay. And here I have shown EGFR, HER2, HER3 protein-protein interaction. You can see from the probe, the number of red dots in EGFR, HER2, HER3 is more compared to EGFR homodimer. So heterodimers are very important in the non-small cell lung cancer. That is the clinical significance. So we use some cell lines which are derived from human. You can see SKBR3 breast cancer cell line and MCF7 breast cancer cell line, but these two are different, HER2 positive, HER2 negative. And BT474 breast cancer cell line and lung cancer cell line which show 3+, that means HER2 positive. So these are some controls we use for non-HER2 cell line. So now, in EGFR related or HER2 related non-small cell lung cancer, there are 3 mechanisms: HER2 protein expression, HER2 gene amplification, HER2 gene mutation. Apart from that, mutation can happen in EGFR and double mutation can happen or a MET amplification. So all this information came from bioinformatics data from human patient samples. So we use this data to design the drugs. So now what is the current targeted therapy for non-small cell lung cancer and different types of cancer targeting EGFR and HER2? We have antibodies targeting extracellular domain and kinase domain, lot of small molecules. But remember, these kinase, they're not specific for particular kinase, even though they're somewhat to some extent specific. But they develop resistance because kinase is an enzyme. It changes the active site. So within 2 years, all these develop resistance. Hence, there is a need to find better ways to treat the non-small cell lung cancer. So our idea is to inhibit the protein-protein interaction. So we have EGFR, HER2, HER2, HER3, HER2, HER4 dimers. If you look here on the left side, HER2 interacts with any of the other EGFR partners. So we call it, it readily dances with any of these molecules. It is always in open conformation. So it forms actually protein-protein interaction, sending the signal for cell growth. Suppose in the case of cancer, we target this protein-protein interaction site, which is domain 4 here, using a compound here. We inhibit the protein-protein interaction. There is no signal for cell growth, and hence therapeutic effect for cancer. That is the idea. So how do we design? I'm a medicinal chemist. We look at the protein-protein interaction and look at the structure of the protein. So here is the structure of HER2 domain 4, and it interacts with Herceptin or the antibody. We took that crystal structure. You can see arginine, a positive charged residue interacting with a negative charged residue here. Central part, a hydrophobic interaction and hydrogen bonding. We kind of measured this and formed a template here, arginine, spacer, hydrophobic group, COOH. So based on that, we designed a compound with a rational drug design. Arginine, naphthyl group with a peptidomimetic, and then phenylalanine here. We tested them in cell line, which overexpress HER2, which does not overexpress HER2. So there is around 800 nanomolar, and in non-HER2 positive cells, 16 nanomolar. So there is a selectivity here. So with that lead compound, we also look at the protein-protein interaction of EGFR homodimer, EGFR HER2 heterodimer. You can see here, the protein-protein interaction site is dominated by leucine, tryptophan, phenylalanine, and threonine. So these are hydrophobic amino acid. Then we look at the literature. They have found that they have studied several thousand proteins and look at the protein-protein interaction site. These are called hotspot residues. You can see the tryptophan, arginine, tyrosine, phenylalanine, isoleucine, leucine. If you look at our compound, arginine, a tryptophan analog, phenylalanine. So hotspot residues are there. We believe that because our compound has hotspot residues, it interacts and inhibits the protein-protein interaction. So this is a small peptidomimetic. If you put this in vivo, it is going to be digested by the enzymes. So we modify them. So we can modify them with different functional groups, D-amino acid substitution or cyclization or end capping. So I'm not going to talk details about that, how we do that. Here are different ways I did N-terminals modification, C-terminal modification, other peptidomimetic modifications, cyclization. So we did all that and studied several compounds. When you want to cyclize, we can't just cyclize a protein chain, a small peptide. It'll flip or snap off. So then what we have to do is just like a plumber, we use particular amino acid at a particular site, like a T joint or a L joint here. So in this case, designed based on the previous compound, you can see arginine, naphthyl group, phenylalanine, aspartic acid, we use this. We repeated this sequence and then cyclize one side. The other side, just like a plumber uses L or T-shaped connection, I use proline. Proline is known to induce what they call beta sheet in peptides and proteins. So why did I repeat this? I repeated this because there is a good chance of binding if both sides have the same amino acid residues and peptidomimetic residues. So we did that, hoping to get better binding. We tested them on HER2 positive, HER2 negative cell line. You can see that in lung cancer cell line, the activity of IC50 is 18 nanomolar. If you take the HER2 negative, it is 50 and 40 micromolar. So there is a huge difference. So our compound is cyclic now, it works. Now, our idea is binding to HER2 protein domain 4. How do we check that? With the surface plasmon resonance. Here we have a chip, a dextran chip, on which we can immobilize the protein. So on the immobilized protein, we pass our compound 18. If they bind, they give the signal using refractive index. Here you can see that there is an incremental index as we increase the concentration of the peptide. And then we wanted to see whether it binds to domain 4. We took this domain 4 in particular and titrated our compound. You can see that it binds. And then we use the control peptide mimetic, which doesn't bind. So this clearly says that our compound, in fact, binds to domain 4 and probably inhibits the protein-protein interaction. But how do we know protein-protein interaction is inhibited? Once again, we did the proximity ligation assay, EGFR HER2 and HER2 HER3, both the dimers. You can see the positive control. There are a lot of dimers here. There are each red dot corresponding to a HER2 EGFR dimer. This is the negative control. One of the antibody was not added. And then when we add our compound in different amounts, you can see number of red dots compared to the control decreased. So we can see that there is a decrease in the number of red dots, indicating protein-protein interaction is inhibited. Not only HER2 EGFR, HER2 HER3. You can see that it inhibits both EGFR, HER2, HER3 interaction. So it's a dual inhibitor. If you remember my slide in the beginning, in lung cancer and breast cancer, both are important. So we are inhibiting the both. So now that is a peptidomimetic or a peptide. Most of the peptide have a limitation. They have to be administered via IV. So how do you make orally available peptide drugs? That is my question. So for that, I want to go to a story here. So this happened a long time ago. During 1960s, a Red Cross doctor went to Congo and he saw that some of the women were drinking a tea from a weed to increase the childbirth. He studied that what is that ingredient. He found that it's a Calata B1, a uteroactive peptide. Then he wondered how a peptide can be stable by boiling and drinking via oral route be stable. But he could not solve it until 1995. David Craig, University of Queensland, Australia, solved the structure of that. He said it is an unusual structure, a cyclic structure with multiple disulfide bond, and 3D structure looks like this. So these are very stable structure. You can boil them, you can put in acid, you can put them in trypsin. They don't digest very easily. So those can be used as a template for design of drugs to make stable. So in this particular case, my compound is here. I did not go for cyclotide. I went for another peptide, which is stable, which is from sunflower trypsin inhibitor, from another plant, sunflower seeds. So you can see the sunflower trypsin inhibitor structure here, a cyclic structure with a disulfide bond, and looks like a beta sheet structure here. So those are comparable. So what we did was we should not disturb the disulfide bond here. That is very, very important. So remaining part, we replaced with some of the amino acids and peptidomimetics here. That's why I call it grafting, because I took some amino acid here and grafted onto stable peptide. So now we have an active compound, we have a stable compound. So the resulting will be active stable compound. So I just want to go through the design. The design cannot be done very easily. We have to go through structural biology and protein structure and compare the 3D structure. Just to show you a sample, this is SFTA peptide. This is compound 18. The grafting is shown in yellow here. We tried different possibilities here. And then when we tested them in cells, HER2 positive cells, you can see the one of the compound SFTA G5, 280 nanomolar, it works. And we tested them in CALO3 cell line, which is non-small cell lung cancer, 73 nanomolar. In MCF7, which is HER2 negative, which is actually 25 micromolar. We have also a control compound which doesn't work. So a compound which is grafted works. Once again, I come back here and show that it binds to HER2 protein and HER2 domain 4. We did the surface plasmon resonance. You can see here concentration dependent increase, and HER2 domain 4, it specifically binds here, and control peptidomimetic doesn't bind. So once again, we look at the protein-protein interaction between HER2, HER3, EGFR, HER2. You can see the control lot of red fluorescence, and we added the compound, red fluorescence is decreased based on the concentration. Similarly here, HER2, EGFR. So our compound binds to grafted peptide binds to actually HER2 domain 4, inhibit both protein-protein interaction of EGFR, HER2, HER3. So far I talked about the 2D cell culture. What is the 3D cell culture effect? Remember that most of the cancer drugs fail because of the 2D cell culture. 2D cell culture doesn't have a matrix. It is just they expose it too much to the media as well as the compound. So compound can kill very easily. But actual tumors are different. So we wanted to see in 3D spheroid model, we grew MCF7, BT474, A549, CALO3. You can see these three form very good spheroids, but CALO3 form loosely formed spheroids. We went ahead and treated them. You can see SFTA-G5 compared to the lapatinib and the doxorubicin and the control. You can see here SFTA-G5 is here, reducing the tumor growth in 3D model. Similarly, in CALO3 cell lines, which is loosely formed spheroid, you can see compared to the control or compound in a concentration-dependent manner inhibits the growth of 3D spheroid. So we also did the PL assay. Nobody has reported this before. You can see number of red dots here. Adding the compound, the number of red dots decreased here and a control here. So now with cellular work, we went ahead with the xenograph model of lung cancer. And here is the CALO3 cells injected into nude mice. 10 to 15 days they develop cancer. And then we treat them via IV 3 times a week. And we use pertuzumab, trastuzumab antibodies as control. And here you can see the graph, tumor volume versus days of treatment. This is the control which is shown in green. And then SFTA-G5 and pertuzumab, trastuzumab as well as pertuzumab antibodies, which work for HER2 positive cancer. So you can clearly see that there is a decrease in the tumor volume by treating our compound. We did the proximity ligation assay again to look at the protein-protein interaction in tissue sample of the tumor. You can see that the number of red dots more in control and red dot decreased in our compound. And phosphorylation here, decrease the phosphorylation of pHER2 in a concentration-dependent manner. So now our ultimate question is the grafted peptide orally available? So we use in vivo model here with the CALO3 cells, and via oral gavage, we administer 50 and 30 mg, 2 doses, twice a week for 4 weeks. We have a control and a positive control pertuzumab, trastuzumab. And you can see here the change in the tumor volume. This is the control. And 30 mg per kg, we did not show much of the significance, but 50 mg per kg showed significant compared to the control orally available. So we believe that the peptides can be made orally available by grafting technology. And overall, a lot of antibodies as well as tyrosine kinase are targeted for non-small cell lung cancer as well as breast cancer, EGFR positive ones. And most of these tyrosine kinase develop resistance. Most of these antibodies have problem. So what I have done here, I have targeted a unique site, domain 4 of HER2 and EGFR, HER2, HER3, which inhibits the protein-protein interaction. Not only that, it inhibits both dimers. It's a dual inhibitor. So with that, I would like to acknowledge the following people, graduate students who graduated and some of the postdocs and graduate students in my lab and the core facilities. Pertuzumab, trastuzumab obtained from Genentech, and Dr. Sita Vidars at LSU helped me to carry out some of the immunoassays. I didn't show that here. And here is my team for this project and funding from R01 grant from NCI. And thank you. If you have any questions, I have left some time. Thank you very much.
Thank you, Dr. Joyce. Yeah, we happen to have a few questions for you. Can you hear me okay?
Sure.
All right, great. So what is the significance of peptidiomimetic-based drug design?
Basically, we use peptidomimetic because, as you know, that peptides, if you eat a protein, it is digested in the body. First, in the acid in the stomach, pH 1 to 2, and then a lot of proteases near the breast border membrane as well as in the lumen before it goes to the intestine before it is absorbed. So most of the peptide protein drugs basically cannot be given via oral. Even IV, most of the drugs can be digested by enzymes and they're probably 20-30 minutes. So that's why we use something called peptidomimetics. where we introduce organic functional groups. In this particular case, I use naphthalene group in there. So that is the significance of that, making it stable for IV or oral administration, which the peptides cannot be done.
What are some of the limitations associated with those type of drug designs?
Limitation is, once again, even though it's a peptidomimetic, you can see particular enzymes can cut them at a particular site. Even though it is cyclized, there is a possibility. And the disulfide bond can be broken. So those are the limitations. Other thing is these peptides are small enough, they can pass through the kidney filtration faster. So that's why I use something called grafted peptide. That means these grafted peptides are quite stable. So hopefully you have seen that I could give them orally. The grafted peptide can be, resolve the limitation of these peptidomimetics also.
Gotcha. So for someone who's interested to start working on the peptidomimetic-based drug design, where do they start for theory and practice?
There are several books. So one can study rational drug design. What I do here is a rational drug design. So there are two ways. The natural compounds, like olden days, you can extract the compound from different sources, check them, and then filter them and purify them. But this is, you go through the bioinformatics, structural biology, and then design. So for this kind of work to be done, 10 years of bioinformatics, molecular biology, structural biology work has to be done. But now that is shortened, you can see that bioinformatics can be done with next generation sequencing very fast. Molecular biology is done. So basically, I would suggest that you learn some structural biology with the basic bioinformatics data, and then see how much data is there about the structure of any of this. Then start designing. That means you have to spend a lot of time with molecular modeling. You need to know how to visualize the protein structure, the cavity, and then basically learn from there. Fortunately, I'm actually, the American Association of Pharmaceutical Scientists had asked me to write a book on peptide-based drug design. Myself, with the collaboration with others, we have written a book. It's going to come out in a couple of months. I can give that information. I have explained in detail, step by step, how to design this. But still, you need bioinformatics, biochemistry, structural biology understanding. Look at the protein structure. That is where you start.
Well, that would be wonderful to have. I'm sure if it's submitted, we can figure out the right people to get it and get it published.
Sure.
Thank you, Dr. Joyce, for your remarkable work in immunology, cancer, and the heart disease work. It was fascinating to see all of that stuff on the screen. I'm not sure I understood it all, but it's wonderful. Thank you so much. And we really appreciate you being on today.
Thank you so much.
Thank you. Before we move on to our next speaker, let me remind you that we made an announcement yesterday about our upcoming hackathon, and that we will focus on this type of metagenomic data in the health and wellness domain. Now, that site is still being finalized, but later today, we'll be sharing more details about that event in November, as well as providing updates over the next few weeks. So make sure you stay around, and we'll get you that information. Next, let's meet Dr. Rhea Kanan. She's going to be speaking on bioinformatics in the beauty industry. Dr. Kanan has an PhD in computational biology from the Weizmann Institute of Science in Israel. She was the Royal Society Dorothy Hodgins Fellow at Cambridge University with over 20 years of experience in computational cancer genomics. Now, inspired by the paradigm shift in the one-size-fits-all to precision medicine, Rhea envisions similar changes in wellness and beauty. So she co-founded LifeGnome, an award-winning company that leverages biological, physiological, and behavioral data to personalize wellness interventions in the preventive health, nutrition, and personal care beauty industries. Rhea is both a researcher, academician, as well as entrepreneur. Rhea, we can't wait to hear how this type of data, the technologies, and all that are being used to bring about new treatments. But we also hope to hear in other domains as well as beauty. Welcome.
Thank you very much. Do you hear me okay? Perfect.
Yes, we can hear you fine and your presentation can be seen well. Thank you.
Perfect. So many thanks for the introduction and thanks to the organizers for inviting me. So this is going to be a very subjective overview on the increasingly important role that bioinformatics plays in the beauty industry. My name is Raya Khanin, and I'm a scientific co-founder of LifeNome, a genomics AI wellness company. So some of you may be asking what bioinformatics has to do with beauty. Actually, a lot. So when I started preparing for this talk, I quickly realized that applications of bioinformatics in the beauty industry have grown tremendously in the last couple of years. So I will highlight the most prominent trends and that have been driven by bioinformatics. I will give some examples of big companies and startups in this area. And also we'll talk a little bit about the challenges that bioinformatics faces in the beauty industry. So to put things into perspective, the beauty industry is actually huge. It is strong, it's getting only stronger, and it's predicted to exceed $780 billion in about 5 years. So what is beauty industry? It actually covers a wide variety of personal care products, skincare, hair care, cosmetics, fragrances, beauty tech, services, various related functions. So these figures do not include dermatology, but our conversation is relevant to dermatology as well. Okay, so let's talk about skin. As a computational biologist, I view skin, our largest organ, as a complex system of interacting biological components and the exposome. So the exposome is everything our skin interacts with. That's water, environment, temperature, pollutants, toxins, and also all the products that we put on our skin, from basic soap to very sophisticated skincare. And obviously, skin is a reflection of all the physiological processes that are going on inside our bodies. So skin is also our immune organ as well. So out of 20,000 proteins, human proteins, 70% are actually expressed in skin. And about 500 proteins have elevated expression in skin compared to other tissues. And these proteins are regulatory and signaling, their receptors and enzymes. So understanding their roles in skin homeostasis and skin aging is extremely important. Furthermore, there are various non-coding RNAs, from small RNAs like microRNAs to long non-coding RNAs that contribute to skin aging and skin disorders. And actually, we still don't know much about their functions. Then, of course, there is our second skin. Then, of course, I don't know, there is message to me or to general. Okay. So then, of course, there is our second skin, the microbiome. So these are all the millions of microbes that live on our skin. And they too change with aging, with environment, and various phenotypes. For example, dry skin, as I will show a couple of examples, acne, and so on. So for a while, studies on the skin microbiome have lagged behind research on gut microbiome. But actually, the beauty industry and the whole research on skin is quickly catching up. So obviously, you know, all these considerations are as important for hair as they are for skin. And as you can imagine, lots of research is going into understanding hair health and hair graying and hair baldness. So what if we could predict and reverse, for example, hair loss? But I will mainly talk about skin here. Okay, so in other words, there is a complex interplay between human proteins, human RNAs, and microbiomes, and also different fungi and viruses that live on our skin. At the same time, microbiomes produce and release different metabolites and peptides, and all this complex network of interactions protect the skin from the environment and interacts with the exposome and skincare ingredient. I call it systems biology of beauty. So you see here the Unilever logo. And this figure was actually taken from a special skin microbiome issue from Nature journal, sponsored by Unilever. In fact, all major personal care companies from Unilever, Procter & Gamble, L'Oreal heavily invest in bioinformatics and genomics sciences. They all actively work on generating proprietary genomics datasets, sophisticated data analysis methods, and they develop computational models and pipelines. But this has not always been the case. A few years back, that's another article from Nature journal called Molecular Beauty. They cite a prominent dermatologist from a leading hospital in Boston, who says that until actually a couple of decades ago, the beauty industry was almost allergic to science. The common perception was that consumers were scared of and uninterested in science. So companies didn't want this word to be associated with their products. But that aversion started to melt with the advent of genomics, which not only transformed research, but also captured the public imagination. And in this paper, they cite a scientist from the brand Olay at consumer goods company Procter & Gamble, who says that over the years, we've converged on an understanding that we should be doing really deep biology on the skin aging process and on products that can be used to improve skin health. It's just like any other biological endpoint that a pharmaceutical company would go after. So just like pharma, just like in pharma, bioinformatics and machine learning are expected to make the search for new ingredients, new formulas quicker, cheaper, and more effective. So perhaps initially the beauty industry was merely following what pharma was doing, but just because of the nature of its business and willingness of consumers to actually contribute their data, so-called citizen science, the beauty industry, with the help of bioinformatics and machine learning, can actually move faster, make more discoveries, in particular in anti-aging and longevity science. I will give an example of such a company in a few minutes. So I will now highlight a couple of major trends in the beauty industry that are being driven by bioinformatics. So firstly, an unstoppable trend is synthetic beauty, sustainability, and ingredient transparency. So biotechnologies are increasingly impacting the production of beauty ingredients, from algae to marine-based components. Companies produce some ingredients in the lab, and these ingredients can be some commonly used collagens, squalene, and palm oil, to some rare ingredients, such as, for example, unique scents of roses without actually relying on expensive rose petals. That's ginkgo biome. Perhaps the most fascinating example of a new generation of beauty company is Archaea, which recently received funding of $78 million from Chanel, Givaudan, and VCs. And the goal of the company is to create sustainable, previously inaccessible ingredients for beauty industry through what they call expressive biology. So this is a screenshot from their website where they call biology the most sophisticated technology on the planet. It has unparalleled precision and scale that modern means have never been able to replicate. At Archaea, they combine technology from the past and present, bridging biology with DNA sequencing, bioinformatics, fermentation, and storytelling to create new product experiences for beauty. Another huge trend is beauty further integrating with the whole wellness economy. Pandemic obviously accelerated this trend, the whole trend of self-care, focusing on one's health, wellness, with an added spotlight on immunity. And there is an overlap here with nutrigenomics, personalized nutrition, and also special emphasis on sleep and mental health. Additionally, there is increasing interest and research on longevity. So people are planning to live longer and they want to look younger. And in this respect, skin aging is obviously the most recognizable and often the most kind of psychologically difficult aspect of aging. At the same time, skin aging is potentially also one of the most amenable for intervention, prevention, and rejuvenation. So skin actually can be regarded as an ideal model for studying tissue aging. And I'm going to talk about a specific example here, company called OneSkin, which I find absolutely fascinating. So what they did initially, they analyzed publicly available data. Actually, that's methylation data. And here's all the data sets that they've analyzed. And they developed highly accurate skin-specific methylome biological clock for skin. So they showed, they validated it. They showed on an independent cohort its accuracy. What they did next was they used this method as a screening platform for different compounds. They screened hundreds of peptides and they found one, they call it peptide 14, that effectively decreased chronological aging, UVB radiation by modulating longevity and senescence pathways. So basically they showed that this specific peptide promoted skin health, reduced senescent cell markers, as well as the biological age of samples. They showed next that this specific peptide actually outperformed the current golden standard in the anti-aging skincare, the retinol treatment. So, and then this scientist, 4 female PhDs from Brazil originally, they raised funds and they started a company called OneSkin. And you can actually now buy their products, which they call skin supplements. The product is unisex and it's for skin and body, and I highly recommend it. It's very good. Okay, so the next trend in beauty is, and in medicine as well, is personalization. Obviously, it's not a new concept, but it's getting more sophisticated. It started with quizzes, it's moved to selfies, various skin and hair scanning devices, and now it is powered by biology. So biology is the ultimate frontier of personalization, and genetics and microbiome are driving this trend. So here I would like to highlight 2 studies. The first one is relatively old, 2009. It looked at many hundreds of twins and identified that about 60% of skin aging is actually determined by our genetics, while 40% is determined by the environment. Another study followed 1,000 people for over 10 years and found that actually some people age much faster than the others. And this is visible in their physical characteristics as well as in their skin features. So the authors of this study built an AI predictor using genetics and other biomarkers, and they quantified aging in younger people, thus allowing for intervention and prevention. So there are numerous genome-wide association studies on genetics of skin health, skin aging. There are SNPs identified to be connected to different skin features, such as early wrinkles or photoaging. So this information can be used to predict not immediately observable skin aging. And several companies, including LifeNom, the company that I co-founded, offer beauty-focused DNA testing kits, informing consumers on their genomic skin traits, curate ingredients and products, and develop personalized products and services. So DNA tests are becoming a commodity, right? I'm talking about genotyping tests. And in the US, you can buy them in your local CVS store. Probably over 30 million people have taken at-home ancestry tests. And as you all know, every chip contains about 600,000 SNPs. With imputation, that's a few million. So it's a lot of information that can be used by beauty companies to look into skin health and skin aging. So lots of very interesting discoveries can be made here. So at LifeNom, we are developing a beauty personalized platform that uses genomics AI. Well, we use machine learning. You probably heard the joke that if it's written in Python, it's probably machine learning. If it's written in PowerPoint, it's AI. So actually, when you work with beauty companies, everything is AI. So jokes aside, we built a huge knowledge base that integrates data from thousands of studies, including gene expression studies. And what our platform does, it utilizes consumer DNA with the survey and informs consumers about their potential risks and offers ingredient recommendation, product recommendation. We also collect feedback and obviously build new insights. We identify new genetic-based features, for example, sensitivities to specific ingredients such as retinol. I will now switch gears and talk about microbiome. So this is probably the biggest trend in beauty, the hottest trend. So cosmetic formulators are developing their own microbiome franchises. They design health-enhancing skincare products that contain bacteria, bacteria extracts, or ingredients that are meant to enhance skin microbe activity. And then Instagram and beauty magazines are educating their readers what are prebiotics, postbiotics, and probiotics. And as I mentioned before, all major personal care companies have been working in this area, and also a number of very innovative startups and indie brands have launched products that use microbiome. So this is from a company, from a Dutch company, DSM, and they developed an ingredient that they called Epibiome Beauty. This specific ingredient relieving skin dryness, and they showcase a clinical study where participants use this ingredient, and kind of the bad bacteria was decreasing with usage of this ingredient, while the good bacteria was increasing. Interestingly, tech companies are also partnering with beauty companies and attempting to solve similar problems. This is an example of IBM partnering with Unilever, and again, an article from Nature Journal. And they also study a similar problem. They study an issue of skin hydration. Here they use explainable AI, and they identify a signature of microbes that is associated with skin dryness. So that's, I think, is very interesting partnerships between tech companies and beauty. And as I mentioned, there is a number of very innovative startups in this field. Is Biomedic, Symbiome, Dermala. Each of these companies claims to harness the power of microbiome and address skin conditions like acne, rosacea, and different features related to skin aging. Other companies like Skin Trust, Sequential Skin, Dr. Elsa Jungmann offer skin microbiome tests. It's a big business, and at the same time, it's data collection, it's citizen science, and it enables future discoveries. So needless to say that behind all this brand messaging and marketing, all this work is enabled by bioinformatics. And in fact, some of these companies have bioinformaticians among their founders. So that's another example of a company, Givaudan. And in addition to doing taxonomic analysis, excuse me, they actually explore which jobs the microbiota can do. So they do functional microbiome analysis. Okay, so bioinformatics powers microbiome trends in beauty. So all these companies require development, improvement, and maintenance of robust bioinformatics pipelines for efficient and fast processing of large-scale metagenomics data. So they needed to detect known species, identify new species, and so on. And bioinformaticians would appreciate that it requires infrastructure for storage, further usage, and so on. So, and all the tools that bioinformatics has developed are also required here, from machine learning, supervised, unsupervised analysis, pathways analysis, and so on. And for those interested in challenges, there are plenty. Discovering functions of species, new microbial pathways, metabolites, and so on. So I see that the notion here that my time is almost coming up. So I'll skip a couple of things here and kind of say that there are lots of pros working as bioinformatician in the beauty industry. It's easy to collect data, it's fast turnaround, it's still a new area of application. There are many challenging problems to be solved and lots of opportunities. At the same time, there are many proprietary data sets, lots of secrecy, fast turnaround. You can see your failures fast too. And even if you have great science, you also need to learn some storytelling, branding, and marketing. So you need to learn some beauty lingo. And I'll skip here, and happy to answer your questions. And please connect with me, discuss with me if you have any interest in bioinformatics in beauty. Many thanks for your attention.
Thank you. That was really interesting. The personalization, it's going to be interesting to see where that goes in the next couple of years. If you want to pull your PowerPoint down, I do have a couple questions for you. Why do you think metagenomics specifically is so important for the beauty and wellness research? And then how can it be integrated with other types of omics data?
Why is it important? Oh, sorry, yes, stop share. Yeah. I mean, it's there are several. I mean, in terms of beauty, it's easy to measure. Somehow, people actually willing to share their skin microbiome. They're not as willing to share their DNA, but they are really interested to learn what microbes live on their skin. In terms of challenges, I think the challenges are the same as I think yesterday somebody from Sweden was talking about integrating different layers of multi-omics data. Challenges are the same as in any other area, but I kind of predict that integration in the beauty industry will happen a bit faster, again, because people are willing to learn more about their skin. They want to look healthy, they want to look young. So there is this
younger, yeah, younger. I'll take younger.
Me too. Yes.
Right.
So that's kind of what I think.
Okay. What's the integration look like?
What's the integration looks like? The integration
like
of
for other type of omics, maybe some fields or whatever.
It can be something that, you know, it's not just easy to detect using one type of data. Like, for example, microbiome and genetics, or metabolomics and transcriptomics. But by combining two types of data, you can actually identify the sort of the compound score, if you like. So if you take biomarkers from different types of omics data.
Okay. I have a question from the audience. I'm sorry. A question from the audience. Are you using ancestry data in the research, for instance, from the GSA?
Ancestry as in, you mean in terms of ethnic differences in aging? Yes.
Okay.
Yeah.
Yeah. Okay. There are other fields in the beauty industry where bioinformatics research is actively conducted. Do you have any examples?
Other areas other than those that I mentioned. I mean, all the skin disorders, skin conditions, acne, rosacea, sensitivity, these are all huge areas where, you know, lots of research is going on. Yes.
Okay. Have environmental issues such as global warming, pollution contributed to the growth of the beauty industry? And then if so, how does bioinformatics help in the process?
Excellent question. Yes. So I mentioned, I think, in the first trend was the sustainability concern, and this is why bioinformatics and synthetic biology, they're working on developing new sustainable ingredients, obviously. And this is going to grow. This is, I think, we're just at the kind of beginning of this development. And in terms of pollution and global warming, obviously it does affect our skin. That's for sure. And again, I just today saw an article how the anti-pollution skincare is going to grow in the next couple of years. Yeah,
wow.
Definitely a huge, huge area.
Where do you think this will be the last question, the future of personalized skincare solutions, where do you think that's headed?
I think it's actually heading where, you know, you'll have your little station kind of at home, and, you know, your microbiome and metabolomics of your skin will be measured at certain regular intervals, will be sent to some center, and then you will receive, you know, your personalized products for your skin, depending on seasonality, depending where you are in a big city, in the countryside, etc. So definitely we are heading in that direction. So it will be a few more years before that, but the vector is there. Yes.
Well, very good. Well, thank you again, Dr. Kannan, for your incredible work in this field. Congratulations on the success of your award-winning company. Thank you much for being here today.
Thank you. Thank you.
You're welcome. I want to remind everyone, I want to remind our audience about signing up for our e-learning portal. If you've not yet signed up, if you've not yet registered, I've just posted the link in the chat box for your free learning account. If you've not yet registered, I encourage you to do so. Again, it is free and sets you on a path to learning about this fascinating body of research and discovery. Sign up now at the link and then click the register for free or the sign up button, the accurate one on the page. Thank you so much for that short commercial. So it's been called the final frontier. Space, that is, and we're hoping that Dr. Joseph Borg takes us where no speaker has gone before, in this case, space omics. Joseph is an academic molecular geneticist and the principal investigator at the University of Malta. Primary interest is the understanding of developmental globin gene switching, regulation and control of urosthopathies. I'm not even going to try to do that one again. I'll let you correct me on that. And the next generation sequencing tools to uncover locked secrets of life. He recently led Malta's first space program for life science in space through the Malteth program. And he also actively participates in the analysis working group at the NASA Gene Lab for Multiomics and on the topical team for European Space Agency Space Omics. Joseph, my apologies for the Star Trek introduction. I'm sure I'm the first person that's ever connected your name with that, right?
Yeah, indeed, indeed. Do you hear me correctly? I hope you do.
I can hear you just fine. And if you want to go ahead and go to slideshow mode, I'm going to get out of your way. It's all yours.
So thanks so much to the organizers for the invitation, which I gladly accepted, and I feel really, how to say, honored to be part of this Omics Research Symposium, because indeed Omics is one aspect of my research work that I really enjoy doing. So yes, so background aside, and focusing on blood research and blood omics, today I'm talking to you about MALET program, which is a life science research program in space. And this is centered around diabetes type 2, with special interest on human skin microbiomes that are found on diabetic foot ulcers. And I will explain why we study these in the context of space, on Earth, and how we compare the two with each other. So as a brief intro, let me just minimize the view screen. Yeah, so all of this, the research program, the MALET program, started at the end of 2020. So during the pandemic years here in Malta, I had an idea to sort of conduct or take my next level of omic science in the context of space. It all came about serendipitously over the internet, over LinkedIn, having the idea and then meeting a group of people that can actually bring the idea to fruition. And at that time, I had taken the idea to our Ministry of Foreign and International Affairs. The idea being that sending an experiment to space could not be more foreign or more international than this. The idea was well received. And from there, basically, we continued forth with our program. So a very quick rough shot about the program. So we did a number of pilot studies in our labs on Earth in Malta to ensure that the human skin tissue samples, the way they are preserved, the way they are prepared in buffer, that the bacteria, the whole microbiome, is maintained for a number of weeks or months, and they do not lose their viability, and that we are able to both culture them back on agar plates, and also we are able to extract DNA and test it by NGS, mostly 16S typing, to determine the classification and the taxonomy of the bacteria. This project ties in very strongly with an ongoing PhD program of my student, who is testing roughly 150 or 200 diabetic patients from Malta. So we only had a selective number of patients that we also progressed to the space study. This work was done in Malta and also in Belgium, where we assemble our so-called biocube. I will show this later on. Basically, the cube itself is shipped from Europe to the States
from Cape Canaveral. There it is assembled on a SpaceX rocket. SpaceX, as some of you or most of you know, travels to outer space, and in this case, docks with the space station. Once it is present or docked with the space station, the astronauts get to work. They take out the cube, they assemble it to a particular place where the cube is powered up. And from here, from Malta, from our control room or center, we are able to connect to the cube and follow the experiment, so to say. Broadly speaking, the experiment per se is not too exciting to monitor, because I mean, the bacteria and the skin tissue don't really change macroscopically or visually, except that you see everything floating around. But the real work is done when the cube is returned back. That is what we did for Mallet 1. And for Mallet 2, which is a follow-up and a replica, because as all of you know, in science, you cannot really depend on one experiment and biological replicates, but you also need to validate your findings. And that is Mallet 2. And Mallet 2 is right now on its way from Orlando to Europe and on its way back to Malta this very week or the end of this week. So it's an exciting time as well. Okay, yeah. So of course, all of this precisely fits in the context of science. I form part of NASA Gene Lab as one of the working groups. Also, ESA, the European Space Agency, also has its own space omics team. And basically, in a nutshell, what these groups are is the experiments that we send to space and back, whether they are animal models, whether they are bacteria, whether they are plant or humans, astronauts. The biochemical and genetic data that we get back are deposited in these databases, especially the NASA Gene Lab repository. And there the data forms part of open science. And we call this open science for life in space. We published a white paper a while ago where it sort of drives the message home about how we can take space biology to the next era by ensuring standardization, applicability, and also a number of important tissue analysis and fixation protocols. We did already have a number of scientific publications on this topic, not necessarily on Mallet, but it also feeds indirectly. And these fall under the Cell Press umbrella. So it is an area, it is a topic where it is very well sought also for publication purposes. So this is Mallet 1. This is the first cube of last year. It is a 3D printed cube using special plastic. Those are the logos. And what is Mallet 1 basically? So Mallet 1, as I said, it centers around diabetes. Diabetes, as you know, is a major public health concern worldwide. And Malta, the country that I live in, is one of the highest prevalent in Europe. Actually, about 12% of the whole population is affected. A large number of diabetic patients are at risk of developing foot ulcers, which is a major cause of concern for hospitalization. There are different types of signs, symptoms. Generally, the people, a major problem is that they do not speak up, and therefore the infection and the wound ulcers tend to become more serious by time. The infections are polymicrobial, so you cannot pin down one particular microorganism. And a large number, almost half or more of these infections result in osteomyelitis and also amputation. In Malta alone, in 1 year, there are 400 cases of minor to major amputations. So more than one person per day, which is a very, very significant number. So this was the idea behind a PhD study, which is ongoing, and also the idea to marry this together with a space science research. This is a very conservative diabetic foot ulcer. Generally, they are much worse, much bigger. And yes, here I would like to mention my PhD student, Christine Gatt, who's a biomedical scientist. She works in a laboratory at our hospital, and she's conducting her PhD work at the university. And she's studying the genetics of bacteria that infect wound ulcers. Like Christine, I have a team of at least 3 other students one is also doing a PhD focusing on ophthalmic disorders, and 2 master's projects which are focusing on globin gene switching and developmental control of hemoglobin. So the way that we work as a team together, even though the students are not all on the same topic, we share the experience, we discuss with each other, even in terms of journal clubs, and generally this yields a rewarding and a rich experience. So this is a typical look inside the BioCube. You can see half of the cube on the right-hand side is focused with a Raspberry Pi and computer essentials or accessories. And on the left-hand side is the unit where there are cuvettes ready to be assembled with the skin tissue sample itself. Something small aside, I am a strong believer of training and inspiring the young generation. And so since in the BioCube, we had space, okay? Space. We had an SD card that can be utilized to embed messages or artwork from school children. We did this. We invited school children from Malta to have or to draw something that has to do with climate change, global warming, or send a message to the world. And we embedded all of these files inside the cube in such a way that we can open them when they are in space and stream them or show them on social media back on Earth. And this was a nice exercise, and we could see that the young children were really enticed. So this is just two quick pictures about the sample preparation. And on the right-hand side, you can see the samples being integrated. And this is a more closer look at the human skin tissue samples and how they are placed in each cuvette. Okay. Of course, the BioCube is one part of the experiment. On the other side of the story, that is on Earth and in Malta, we also require a laptop or a mission control room where we can plug in our system, and we are able to communicate or contact directly the BioCube itself for many reasons, some of which are for aesthetic or fun, the use of camera to see the samples to ensure that there were no major breakages or problems. There are also a number of sensors to detect the temperature, humidity, and one can adjust those sensors. And there is also LED lighting, and that you can change different colors according to the mood that one is feeling. But basically having a mission control center back on Earth ensures peace of mind that the experiment is going well. This is the cube itself positioned on a module, which is an exact replica that is found on board the Columbus module on the space station. Okay, IceCube stands for International Commercial Experiment, and you will see one of these also on board the space station later on. This is the cube in its final steps with all the samples integrated, packaged in a special box and shipped to the States. And from the States, as I said, the astronaut handled the cube, and you can see here.
The station to the IceCube activity is complete. Now looking for the crew conference. We are very much grateful for your support today. We also took note of your Japanese skills, so thanks for the entertainment. And the crew conference is ready on Space to Ground 4.
So there's just some talk between the astronaut and mission control.
It's good to see those new players in space, Malta and others. So congratulations to them for their success and moving forward.
That's when they mentioned Malta for the first time. And there you can see our cube alongside others placed on board the IceCube's facility, which is located on board the space station. Once plugged, that is powered up, and we have access to it from Earth. Once roughly between 30-35 days elapse, the same spacecraft that took the experiment to the space station is undocked. It deorbits around Earth and falls or splashes down in the Atlantic Ocean. There, a boat will go out. It will retrieve the spacecraft. The spacecraft is taken back to the Kennedy Space Center, and from there, all the experiments that were sent from the space station back to Earth are sorted. It's just like a postal service, and we receive the cube back to Malta. And there where the real work starts, where we open up the cube, we take out the samples, and there is a battery of tests that we do on all these skin tissue samples, which includes aerobic and anaerobic bacteria, the use of culture media as broth, and also culture media as agars and various forms. Of course, Christine Gad, being the microbiologist, is much more well-versed in all the different tests that we do. An interesting observation or a photograph that we had to do was when we identified that bacteria sent to space and back were actually cultivated on agar plates. So this showed us for the first time, for us in Malta, that we were able to send an experiment. The bacteria and the skin microbiome survived and came back to Earth to sort of tell the tale. Yes, so there's a large number of genetics and omic tools that we are doing. The first of which is concluded, is a first wave analysis, which we call a 16S typing. And 16S typing serves as a taxonomy classification of all the bacteria that are present in the samples. In the Mallet one, we had 6 different samples. So 6 different patients that came to our hospital, and each patient, a human skin tissue sample was obtained. The human skin tissue sample, as part of the surgery process, was split in 3. Okay, 1 was immediately processed and frozen. So we obtained a human skin microbiome profile on day 1. And the other 2 pieces, 1 was left in our lab on Earth, and 1 was sent to space and back. And therefore, for each single patient, which we have 6, okay, we have 3 different data points, as you like. Okay, so we have the omics or the 16S typing for each of the 3 timelines, and we can compare the differences between those 3, and we can also compare the differences between the 6 samples with each other. Okay, and there where you obtain a comparative genomics approach. This figure shows you a spread and the largest number of bacteria found together with the lowest number of bacteria found in the skin sample itself. Of course, this was replicated for all samples. Yeah, so our aim was to obtain a signature that is correlated with the tissue type itself. We had many other tissue types as well. I mean, we had a simple skin swab from healthy controls to act as a control. We had the foot ulcers themselves, those which were sent to space station and those which were left on Earth. We also had biofilms from certain wound ulcers, and we also had negative uninoculated controls. There is extensive informatics that was done. I did not do this myself, but a team of bioinformaticians, also with the help at NASA Gene Lab, which exclusively work on these type of analyses. And interestingly, from these analyses, before going into metagenomic sequencing, which is being conducted as we speak, so to say, which we are waiting for the Mallet 2 samples as well. But from just the 16S typing, we could see, or we could observe, that a small select number of bacterial species in space appear to thrive or flourish more than the same tissue type that was left on Earth. So there are these subtle differences, but meaningful, between one tissue type of one patient left on Earth, sent to space, and the differences between the 2. And we have a select number of bacteria that exhibit these differences. We can mention that later on. One key bacteria is Morganella morganae. Yeah, so the immediate next steps is that we are comparing the 16S data between these 3 data points over the 6 biological replicates. Something which we haven't done yet, but we want to explore, is that the patients themselves who came to hospital, we will also be sequencing the full genome of each patient, and this we believe will give added data, added information that we can sort of interplay or overlay, so to say, the genomic data of the patient together with the microbiome data obtained from the human skin. Metagenomic sequencing is a no-brainer, so that is being done on the isolated microbiome together with colleagues in the States. And of course, there are a number of key people that sort of what I have shown or presented in these few slides have made this possible. So primarily there's the ministry, for Foreign European Affairs. And for Mallet 2, there is also the Ministry now for Research and Innovation. A number of key people that are in the States, and these are Chris Mason, is a very renowned scientist who in the past has worked on NASA twin astronauts. Maybe some of you know Mark and Scott Kelly, who one of them is a senator. They are twins, they are identical twins. They are both astronauts. And there was a huge study where one of the twins was on Earth, the other twin was a year in space. And therefore, there were many blood samples being taken, urine samples, and other measurements. And one can conduct all the genomics and the multi-omics that one can think of and compare between the two. And it's amazing the results that one gets back. Braden Tierney is the one who conducted the Informatics for the Mallet program. Afshin was the original point of contact at NASA, who introduced me to the world of space bioscience. I am indebted to him. Kevin Kassar and team, he is a vascular surgeon at our hospital, and he's the one who physically operates and takes the skin tissue. I mean, we depend on these medical professionals. Arcafort and Evolve are private tech companies. It's impressive the interest that we see in such experiments or programs by the private sector who want to be part of this mission. Also, an interesting bartering idea, if you like, is that the biotech companies that agree to sponsor our work or sponsor our mission, they get to emblazon their logo on the cube itself. So that is exposure that is out of this world in the literal sense of the word. And of course, Christine Gattis, she's the PhD candidate who's doing all of the groundwork in the lab, physically working. Yeah, so this slide we're almost at the end here. The slide is sort of a collection of Mallet 1, which is the top left cube, and Mallet 2, which is the bottom left cube. You can see a color change. Mallet 2 is an improvement on the cube design. It is made up of a machined aluminum. So this is like the same as a MacBook laptop or these new fancy laptops that are made of aluminum. And basically you have a block of aluminum and they are machined from the inside to outside. And there was also an upgrade of the Raspberry Pi computer itself. And there on the bottom, on the center bottom, you can see a USB dongle, a key that is used to connect directly with the ISS. An interesting idea or notion is that once you plug in this dongle, okay, automatically the mission control room or the laptop that you're using disconnects with the internet and you only connect with the ISS. This is part of the encryption and security policy. And on the far right there you can see the SpaceX rocket that departed Earth to arrive to the ISS. This is a list of all sponsors and logos to date, and 3 pictures that shows you a close image of Mallet 2 cube. Yes, Mallet 2, I didn't speak too much about this. We had 5 samples and not 6 microbiomes, and we had 1 sample reserved for dried yeast, Saccharomyces cerevisiae. We have some ideas that we wish to implement when the experiment gets back. And as the cube itself on board the platform itself. And I believe that was the last slide. And if you have any questions or ideas, or maybe you have an idea to send to space, feel free to reach out to me on joseph@spaceomics.com. And watch out for some emerging publications on Mallet. So with that, I thank you all for being patient to listen to me. I think I cannot hear you, Grant. I cannot hear you.
Good old microphone muting, right? Does gravity play a role? This is from the audience. Does gravity play a role on aging process and the rate of cell division? I'm assuming he means in space, so in gravity. So does gravity play a role?
So there are some conflicting, of course, this is a little bit different from the Mallet program that we do, right? However, in the context of aging, we have been told that in space we age faster. However, recent evidence, and is also published, there seems to be observation that telomeres at the end of the chromosomes, instead of shortening, actually, in long duration, they elongate. And that means that these cells might actually become more younger. So maybe we'll be having trips to space to become younger as opposed to aging.
Well, that'd be interesting. A little expensive, but interesting nonetheless, right? So how is the MALITH program used to understand living in space?
Yeah, so MALITH program, okay, so it's ambitious. It's a very first program for Malta. And the idea or the overall objective, besides the core, is a life science experiment on diabetes type 2. We also know that astronauts in space generally are very healthy and athletic. Okay. However, astronauts in space also have difficulties with vascularization and the bloodstream itself. They endure space anemia. So understanding wound ulcers, foot ulcers, and the complications that arise on Earth can feed in directly with complications that we observe in space, especially if space is opened up wider for astronauts who might not be completely athletic or fit.
Like some of the people that can just pay to get there, right? So do you think there are specific measures that astronauts take before or after going into space? Maybe talk about one or two of them.
This is a very, very good question. I think to date, it is still not completely set in stone, but there are definite measurements. I mean measurements that are done at the brain, the EEG, and cardiac function. I mean, these are logical tests that need to be done to ascertain that the individual, the astronaut, is fit, especially to survive a 3-month, a 6-month mission. An interesting thing is the introduction of omics testing, genomics, DNA, RNA expression profiling. This is a very, very, very recent entry. And as we progress, we're going to see more and more omics testing on astronauts.
Fascinating. Well, thank you yet again. This has been wonderful. I apologize again on all the Star Trek references, but that was fun. Glad you played along.
Excellent.
I'll avoid any more cliche space movie references like your work serves the needs of the many, but now it won't go there. So thank you again, Dr. Borg, for being here. And we're going to move on to our poster presentation, prize distribution. And we're excited to announce the prizes from our poster competition. So I'd like to invite Sonalika Ray from Pine Biotech. Sonalika?
Thank you so much, Grant. All right, so we are glad to announce the results of the poster presentation competition now. This year, we got an overwhelming response from students and researchers from different backgrounds, from different parts of the world. The abstract committee identified 16 posters from a pool of 25 posters for poster presentation competition. The posters were evaluated by judges on criteria which were scored on the title of the poster, the relevance to the theme of the conference, the methodology, results, legibility, reference, and outlook of the poster that included color, spacing, and balancing. So here we have the results now. The first award for the category Best Technical and Research Project on a competitive topic goes to Dr. Uma Yadav, who holds a PhD in life sciences. And her topic was on machine learning approach for drug response prediction in diffuse large B-cell lymphoma patients. I would like to congratulate her. Uma, are you here with us? Can you please unmute yourself and also turn on your camera?
Yes. Yeah. So can you hear me?
Yes, I can hear you. Congratulations.
So, yes, thank you. I would like to take this opportunity to thank Omics Logic, Pine Bio for this platform. This has been a great platform and is a great platform, not only for graduate students, but also for continued learning and upgrading your skills and knowledge, which is the need of time. I'm grateful to my mentor, Dr. Raghavendran, for his guidance. I thank Sonalika for immense support throughout the project. Thank you all at Omics Logic, Sparsh, and Srigori. I thank all the judges, and I also congratulate all the participants for their work. Thank you all.
So much, Uma. And the second award that was for the category
So now,
of
Sonalika,
most
if I could ask,
promising
I'm sorry, if I could ask Uma one question while she's still on, what do you plan to do next?
sure.
What's the next big thing for you?
Regarding the continuing on this topic of project?
Sure, your career, however you wish to answer it.
Sorry, I couldn't get your question. If you could repeat.
What are you planning to do next, either with your research topic or your career?
So regarding the research topic is that for this work, what we have done, we have considered just the gene expression data. Now we want to try to integrate it with copy number variation and mutation data. One. Number two, regarding career, I'm looking for postdoc opportunities wherein I would like to explore in the field of data science and machine learning and applying in field of life sciences and precision health, precision medicine, oncology, precision oncology. That is what I'm looking for.
Okay, fascinating. Thank you so much. Just like to hear where this is going. And it's, again, congratulations. Sonalika, go ahead. I'm sorry I interrupt.
Thank you.
Perfectly fine, Ran. Thank you so much. And the second award for the category of promising research direction for future potential goes to Ms. Ayushi Notra. She has completed her master's in bioinformatics honors, and her topic was identification of critical genes associated with intrahepatic cholangiocarcinoma, weight gene co-expression network analysis. Hi, Ayushi, are you around? Can you please turn on your camera and also your mic?
Yes, hello. I would like to take this opportunity to thank Pine Biotech and Omics Logic for giving me this opportunity to present my research work and get it reviewed by judges from all around the globe. And I would also like to thank the judges for selecting my project as the most promising research direction for future potential. In this project, I tried to correlate the genes associated with this cancer with the tumor microenvironment. And as we know that the tumor microenvironment plays a very critical role in the cancer progression, development, and metastasis. So it was a great area to work on. Thank you.
And thank you. And congratulations. That's a wonderful award. So where will you go next? What are your plans next?
Well, I'm pretty much very interested in studying how a particular disease occurs in the body at the transcriptomics level. So I would want to carry on my research further on this particular topic only, where I study diseases and how that particular disease is caused and the underlying genes responsible for the disease occurrence in the body.
Do you have a recommendation to others for trying to work on their own projects or trying to win?
Yes, I would like to recommend everybody that do a lot of literature review, because that enables you to have a particular research interest ignited inside of you. So a literature review is must. And so that you can, find your particular interest in a particular research domain, because there are n number of things that you can do using bioinformatics. So do the literature review and find your particular interest and then work on it.
Fascinating. Thank you so much. And again, congratulations. Sonalika?
Thank you.
Thank you, Ayushi. Thank you, Grant. And the third prize that was for the category of most innovative approach in an established domain goes to Mr. Ojasvi Datta and Mr. Shubham Kumar, Masters in Bioinformatics. And their project topic was in silico reverse mutation of spike protein RBD from different SARS-CoV-2 carrier host. So, Ojasvi and Shubham, do we have you both here with us?
Yes. So, thank you for giving me the opportunity for participating in the Omics Research Symposium poster presentation competition. and it is good to evaluate your research work at this platform. And I would also like to thank my college faculty and my PI, Mr. Sandeep Saini, for their constant support. So we worked on the structure analysis part of SARS-CoV-2 RBD protein, like spike protein. And we tried to do the reverse mutation in our project. And we tried to find the, like how can the mortality rate can be like get down, and what are the next competition, like what are the next techniques which can be used for further making the medicines in the field, like to get these SARS-CoV-2 down as a pandemic.
Congratulations to you both. How was
Thank
this
you.
experience developing your own project meaningful to you both?
So we worked like we used multiple tools for our project. We used 7 tools for mutation analysis, and then we used different docking tools to get our results for the protein-protein interactions. And the structural analysis was done in this whole project, and it was good to like complete whole project in terms of structural analysis part in bioinformatics.
In addition to that, we have also analyzed the phylogenetic analysis of the SARS-CoV variants with humans.
Fascinating stuff, guys. What are you planning to do next?
Next, we'll generally try to pursue PhD in structure biology, or like drug discovery or drug medicine, precision medicine topics. Or main topic would be our astrobiology. So in these 3 fields, we'll try to pursue PhD.
Well, thank you very much. And again, congratulations. Most Innovative Approach is a really great award. Thank you, gentlemen.
Thank you.
Sanalika?
Congratulations once again to all the winners. And now the stage is all yours, Fran, to proceed ahead with the talks. Over to you.
Very good. Thank you so much. Well, we're very excited on our next guest who is coming up, Professor of Virology and Biotechnology and the Department of Pathology. Looks like I'm getting a note here. I want to make sure I'm on the page here. Am I going forward too fast, gentlemen?
All right, we have with us Dr. Julia Panna. She was one of the judges, and she has few comments for the winners. So, hi, Julia. How are you? Yeah, over to you.
Hello, everybody, and thank you again for inviting me. And congratulations to all the winners, to Dr. Uma, Ayusha, Oasvi, and Shulpan. Congratulations. I really enjoyed looking at all the posters. It was very exciting to actually see that how you use the tools and how you use the data that is available. And I think it's a very great opportunity to see what, what our investments also, not our in particular, but also from the whole field, putting the data out there for everyone to see and to use is a very big endeavor, and a lot of people put a lot of effort into it. And of course, also the tools that are available, many tools are today available freely. So it's really exciting to see how others use them and how you make up your own great research projects and come up with great ways to use different tools. So congratulations, and thank you for the opportunity to be here with you today. And also, if I have this couple of minutes, I wanted also to thank the organizers for this event. I know that enormous organizational effort has been put into this to to bring the whole community together, to organize the talks, to find the speakers. So again, thank you very much for organizing such great event, and thank you for inviting us to be part of it.
Julia, what was the hardest part in the judging sequence?
Yeah, the judging sequence. Well, most of the posters were very nicely organized, so it was very easy to understand the flow, how the person got the idea from the introduction, the methodology, how people have used the tools, and what conclusions they came up with. So that wasn't a very big challenge in most of the posters that I have been looking through. Sometimes it was challenging to actually follow this logic, but then it was obvious how to judge. But of course, some posters are very good, and it's always hard to put a number, the final number. So I think it's a problem for all the judges all the time.
Right. How many judges were on the team?
I think Sonamika can answer that one better. How many judges were there?
We had Dr. Abhishek, Dr. Harpreet, Dr. Julia, and Dr. Raghavendra. So we had 4 judges from different institutions and labs here.
Very good. So Julia, since you're on the spot here as the only judge on the screen, what could others do that are watching this right now on the next competition to be move higher in their work? What did you notice anything that was lacking or some way to help them?
I think the really well, from what I seen from the posters, there were some posters that I was really sad to see that just because of technical issues, I couldn't even read the text. Some of the texts were put as pictures instead of the actual text. So instead of judging the actual work that the person put into this project, and the thoughts that were going on, I had to rely on very partial information just because I couldn't see the text. So if you are preparing a poster, just make sure that we can read it, and then it will really improve your input, and we can really judge your work and not just how the picture appears. So that was something that is very technical and I think easy to correct. Another thing that I would also mention when you're preparing posters, and this is of course, with the posters that were top posters, this is not at all a problem. You could see the flow of the events. The introduction was really closely related to the methodology. We could understand what the question of the research was and how that question was supposed to be answered. And then in the methodology, we could see how the person answered these questions, which tools were used, which data were used. It was all very, very clear. So when this link is missing, then it's, it's unfortunate we cannot read or we cannot understand what actually was done. And then it's just the project is not clear. Why is the person actually doing this project? So just keep the flow, the regular flow of the project. You have the introduction where you have a hypothesis, you are checking, and then methodology, how you decided to check, which data, which tools you used, and results, and of course conclusions. So the regular things.
I almost hear you saying it might be worth it for those that are doing this next year to maybe ask a mentor to review for them, or someone maybe is in the competition before, maybe review their work to make sure that's all clear.
that, that is always the best thing to do, I think. Even if it's not an official mentor, just somebody else who doesn't know your work, who hasn't been with you in the project, just to read through your poster to see if there is this logic of a regular project. So anybody, a co-learner, a co-student, a co-researcher, anybody, I think any poster benefits from that. Because when you're in the project, it seems like everything is already clear, right? We have been in this work for so many days, nights sometimes, that we lose track of what's clear and what's not clear to an outsider. So it's always a great idea to have somebody else read it and look through it together.
Great. Sonalika, can you speak to how many entries there were? Is that released?
Correct, I didn't get you. Yeah, 25. All right. So there were total 25 posters that we got, but only 16 of them were selected to be put together on the poster presentation landing page that we prepared. And out of these 16, we have these 3 winners.
Very good. Well, thank you. It's even a, I don't know if that's a bigger honor, but to be chosen out of 25, the only 4 that move forward, that is fascinating. What, any one of you that wants to speak to this, what was the biggest challenge in answering what Julia just spoke of, the flow, or what was your biggest challenge to make sure, you, even though you knew it, that someone else could understand it? Anyone that wants to unmute and speak to that, please. Somebody jump in and help me here.
Yeah, so starting from introduction till results, it is very hard to like relate all of the steps, like hypothesis in the introduction, then in the methodology. Like we have used more than 10 tools, so it is very difficult to like mention all of those in the methodology part in the poster. Then again, in the further part, result and conclusion, there we have to showcase all the results which we have got from the project we have done. So it is really hard to like relate all of those together, but somehow we managed to do that.
Okay, very good. Someone else, please. This is really great information for those that would compete again, so we really appreciate your helping us out here. Go ahead.
Yeah, I would also like to add the same, because we have so much of information when we do a particular project work that putting it in just like a piece of paper all together becomes really, really difficult. So you have to choose what you need to put out over there and what you need to leave, like what is the thing in the project which is not of that importance, that if you leave that out, it still makes sense. So we need to be very critical on that, and we need to put on the important things, and we need to like leave out 1 or 2 minor things which can be understood if they're not over there as well.
Very good. Well, thank you all again so much. I think we're ready for our next speaker. So we're going to go ahead and let you guys go back to all your research projects and number crunching and all that stuff. If you plan on entering again next year, best of luck to you. And for those of you that would like to enter next year, please watch for notifications on that on our website. And of course, I'm going to put it in the chat box again if you'd like to register for your free learning portal so you can learn more about everything we're doing at Pine Biotech and Omics Logic. Please jump on that and get involved. Again, thank you all so very much, and we'll go ahead and roll to our next speaker. A professor of virology and biotechnology in the Department of Pathological Sciences and a director of the Division of Biotechnology and Molecular Medicine at Louisiana State LSU Biomed, Dr. Koulousis has extensively utilized viral vectors for vaccine development and cancer treatment. His other research interests lie in the structure and function of proteins and glycoprotein and the use of virus-like particles for drug delivery, bioinformatics, and the development of new drugs to combat infectious diseases. Constantine, if you are there, the stage is yours. And we may be having a connection issue, so if you would all hang with me for just a moment, we will make sure that we get him on screen. Dr. Kasoulas, if you are out there and on, we would appreciate you connecting.
Grant, you have to make him a co-host for him to be able to speak and share his video.
I cannot do that. We'll have to have somebody else on the team pull that off, Elio. Again, our apologies for the technical issues here. We'll be fixing this right away.
Sonalika, I guess you are the one that can do it.
Well, this is always the fun part of Zoom and webinars across the world. We thank you for your patience. If anyone wants to ask, let's do a quick little poll. We forgot to ask this on the way in today. We see we have about 65 people participating on chat. Where are you in the world, and what is your name? We'd love to see where you're at, and maybe drop in your focus of omics as you're moving forward. Let's have a little poll, see who we can get going here. Irina, thank you for the opportunity. You're very, very welcome. We're glad you're here today. There's Dr. Kuslis. Good morning, sir. How are you?
Very good. Finally, we got through.
Sorry about all the mix-up there. We had you all introduced and then we didn't have you on stage. So thank you for your patience with us.
Okay.
We are ready for you to go ahead.
Absolutely. I do have some slides I would like to show. So hopefully I can share.
You should have screen sharing capabilities and just go into slideshow mode.
Okay. Let's see here. Okay. Can you see my slides?
Not yet.
Okay. How about now?
We're getting there. I see your desktop, but no slides.
Okay. Must be the wrong one then. Hold a second.
Beautiful island, however.
Yeah. Let's see. What's going on here?
There we go. And go ahead and go to your slideshow menu and get in slideshow mode, and you should be.
Okay, you see the slides, right? Can you hear me?
Yes, we do. Go right ahead. Thank you.
Okay. Yeah, so I guess good afternoon, everybody. My name is Gus Kasoulas. I'm a professor of virology and biotechnology at Louisiana State University and head of the Department of Pathobiological Sciences. I also have the honor of being the president of the National Association of IDEA Principal Investigators, NEPI. NEPI is a not-for-profit organization that represents 23 states and Puerto Rico, and all the NIH funding that goes into the special program from the National Institute of General Medical Sciences, which is called IDEA, which is really funding biomedical research at multiple levels, including primarily undergraduate institutions, flagship institutions, clinical translational research centers, as well as a number of other co-funding opportunities through NIGMS. And I will tell you a little bit about NEPI first and everything else that we do here. But I'm delighted to participate in this Bioinformatics conference. We have a long-standing relationship with Pine Biotech, and we have embraced the platform and a lot of the training, as well as the software and a number of other things that we do. And Pine Biotech is instrumental in helping our programs here at LSU, as well as through NEPI, providing content and research opportunities, as well as training modules throughout the 23 states and Puerto Rico. So NEPI aims to protect and promote the IDEA programs. As you'll see later, it's approximately $450 million per year, allocated to approximately 200 research centers throughout the 23 states of Puerto Rico. It fosters interactions, promotes research sharing, enhances the visibility of all these programs that I just mentioned, and develops consensus on priorities, identifies and disseminates best practices, and also identifies opportunities and developing strategies. One of the main concept is that by promoting biomedical research within these 23 states in Puerto Rico that traditionally do not receive a high level of NIH funding, that we be able to develop manpower and lift those states to be able to compete for traditional NIH funding through all the categorical NIH institutes. This is the leadership team with that currently you can see on the NEPI website, and all the programs fund the organization through a setup fee, and NEPI does a number of different activities that I will mention to you as we move forward. So the mission again is NEPI is to provide leadership and communication and to improve the biomedical research throughout the 23 states in Puerto Rico. We want to foster interactions among the IDEA programs and all these 23 states of Puerto Rico to promote sharing among resources among these different programs. And importantly enough, that all the INBRIs, as well as the co-bridge CTRs, especially the INBRI programs, have a special emphasis and mandatory emphasis on data science and bioinformatics. So one of the major aspects of the INBRI programs is actually the promoting data science and bioinformatics. I am currently the principal investigator of the Louisiana Biomedical Research Network, which is one of the INBRI programs. There's only one INBRI per state, so there are 24 of those programs, approximately 20 million over 5 years, and is devoted to really support primarily undergraduate institutions, creating a pipeline with a mandatory activity to be promoting data science and bioinformatics. So again, we're developing priorities and consensus. In fact, all the INBRI PIs are meeting October 18 in New Orleans to arrive at a consensus on priorities and new directions. And we want to identify, disseminate best practices, and to develop strategies to achieve the common goals of the entire IDEA program, which is really a pipeline coming from the undergraduate to the graduate program, to the postdoc level, to the faculty level, and beyond. So history of the IDEA, it started in 1993 with approximately 50 million. Over the years, has really become much bigger, approaching 1% of the NIH funding totally. And of course, the program is very unique because it serves unique populations, such as rural and medically underserved communities in these states. So health disparity is one of the big topic that's actually addressed through those states that predominantly have a majority of these health disparity issues. This is the up to 21, the funding right now sits at 2022 at 430 million. Hopefully in next fiscal budget, it will go approaching the 500 million, which will be approximately 1% of the NIH budget. And of course, a sizable part of every state's funding is actually this IDEA program, as you see in Louisiana here, Mississippi. You can see 21 million of the 176 million in 2021 was actually IDEA funding. The different programs that I mentioned include the Centers for Biomedical Research Excellence, the COBRIs, and those are for mentored by senior investigators, and their goal is to take young faculty, typically tenure track assistant professors, to become independently funded through an R1 mechanism. And the program can transit to a phase 2 and phase 3 based on how successful it is in graduating independently funded investigators. The IDEA network, like the Louisiana Biomedical Research Network, has a different goal of promoting biomedical research in primarily undergraduate institutions. Typically, they go for R15 type of grant applications, but even beyond that, and to create a pipeline of undergraduate and graduate students that ultimately feed into COBRIs and other programs. We also have the clinical translational research centers that are sort of small clinical translational centers. The bigger ones are in Massachusetts and elsewhere. And those are approximately funded about 20 to 25 million, and have a different scope of really translational medicine, translational research. The Pennington Biomedical Research Center is now the headquarters of the Louisiana-based Clinical Translation Center. It's called the LACATS. The IDEA program also provides co-funding. It's very important because if you submit R1 R15 and you're not within the pay lines, you could actually get funding by having co-funding provided by the National Institute of General Medical Sciences. And there are also technology transfer accelerator hubs that were funded previously, and they may continue in the future, creating an entrepreneurship activity to promote also economic development within these 23 states in Puerto Rico. The NIGMS promotes the additional programs over the years. We had collaborations with other non-IDEA NIH programs, including the maternal and infant mortality research, the COVID-19 patient registry, cloud computing, and affiliation with the NIH STRIDES program, Google and Amazon. In fact, they really discount prices for IDEA participants to participate in these particular programs, and including Google and Amazon related products. They're also continuing new activities in collaboration between NGMS and other ones. And I would urge you to look at the NGMS website to actually see these new initiatives that are co-supported by the IDEA program and categorical NIH institutes. These are the IDEA states in green here that you can see. And the programs include, again, the COBRIs, IBRIs, CTRs, co-funding, and the entrepreneurship hubs. The NEPI in itself has a number of activities, and I would urge you to look at the nepi.org website. It funds IDEA undergraduate travel funds to Washington, D.C. to visit Congress. It awards undergraduate, graduate students, and assistant professors best presentations. We have 4 regional IDEA meetings in 4 of those regions that NIGMS has, be able to enlist us. And we also have a national meeting, the National IDEA Symposium of Biomedical Research Excellence on NISBRI, that was held every other year in Washington, D.C. In person. Those were basically postponed because of COVID-19. The next virtual conference would be held December 12th through 14th. It would be from noon to 6 p.m. to accommodate the different time zones. And we hope to make these announcements and start accepting abstracts and all presentations very soon. We also will be, through the NEPI, you'll be able to access IDEA-wide databases and research expertise and network with other investigators. And we have offered discounts to virtual courses in data science and bioinformatics in a collaboration with the Pine Biotech that you see on the NEPI website. A number of activities included NEPI-sponsored symposium. This was a 2021 symposium Other symposia have also been promoted through the NEPI, including, I think, this one here, this conference. I hope it's also on the NEPI website. And the last thing I wanted to talk to you about is the Louisiana Biomedical Research Network. This is the website for the network itself. And you can see through that a number of information about Louisiana-specific activities. We have a summer program that is also funded by the Louisiana Border Regions. We had over 50 to 70 people participating in different projects over the summer relating to bioinformatics and computational biology that we execute every summer. And of course, the LBRN has two major cores. One is the Bioinformatics and Biocomputation core, which is headed by Dr. Chris Taylor, who's a professor at the LSU Health Science Center in New Orleans, and a protein production purification, proteomics, lipidomics core that's headed by Dr. Johan Lee, who's a professor here at the LSU campus in the Department of Biological Sciences. The LBRN has an annual meeting that's scheduled for most likely the second week of January. And we do invite people from all over Louisiana to participate, especially people that are funded or participate in active LBRN programs, but others can actually visit. We also sponsor a Bioinformatics regional conference, and that will be planned hopefully in New Orleans at the University of New Orleans campus sometime next April. Through our LBRN website, you'll be able to see recorded lectures or previous bioinformatics conferences. We had one last year, which was virtual, that had 10 fantastic speakers from all over the country, included Michael Snyder, the chairman of the Department of Genetics at Stanford, promoting this composite of transcriptomics, proteomics, metabolomics to ultimately arrive at a digital twin that would enable to predict diseases and follow patterns, changes in the body, in addition to a complete scan, head to toe, that provides new information of how basically everything works and integrating this data into a digital twin. It was, I think, a fantastic presentation. And that's where basically, ultimately, the science of data science and bioinformatics will go, especially for health, where you'll be able to combine huge data sets, whether it would be genomics, transcriptomics, proteomics, metabolomics, health-related factors and clinical readings, to be able to combine all those to create a digital twin that ultimately would predict or basically detect any potential perturbations in the body system to allow for prophylactic or therapeutic approaches to disease that may occur much later in life. So this is what I wanted to sort of formally discuss with you, and I'll be happy in the remainder time to answer any questions that you may have with regard to NIH, the IDEA program, NEPI, LBRN, or any other topic in bioinformatics. I had the pleasure working with Pine Biotech to offer an advanced course in transcriptomics a few years ago. We are now promoting the creation of a bioinformatics, data science bioinformatics course in every primarily undergraduate institution in Louisiana. Those are 8 institutions that are primary institutions. However, there are 27 that participate throughout the state of Louisiana. So we hope to start with LSU Shreveport to create with Dr. Esquez-Vec a bioinformatics course at the 4,000 level for undergraduates and graduates. And that will be using content from Pine Biotech as well as faculty members from the network. And we hope to repeat that and create a data science bioinformatics course in every of the 8 principal primarily undergraduate institutions. And this is really the purpose of LBRN and the IDEA program, to provide opportunities and training for biomedical research with a special emphasis in data science and bioinformatics. So I'll stop sharing, and I'll be happy to answer any questions.
Thank you. Wonderful. All this help. We just went through the poster presentations and the awards, and to have this much help out there is wonderful. So how is NEPI working with the IDEA community to promote biomedical research training and advance the importance of all the NEPI programs?
So NEPI is the principal or co-principal organizer of the national NISBRI meeting, and also participates in all the regional meetings. All the registrations and some of the expenses are really picked up by NEPI. However, LSU has a U13 grant to support the conference. So it's actually an NIH, LSU, NEPI collaboration to run those major conferences. And we provide stipends, honoraria for bioinformatics speakers, and we organize symposia within those conferences for bioinformatics. The other important thing that we already have started is to provide subsidized or discounted courses, trainings. We started with Pine Biotech throughout the network. Also, we have asked content that is being created by any of these programs, 200 programs, whether that would be special projects or exercises or even, a recorded training that can be uploaded on the NEPI website and shared among all these individuals. Would like to also to ultimately create a forum for job seekers and placement of graduate students, especially in the data science bioinformatics sector. And we're moving towards this goal as we try to populate the website more than we have at this point. I don't think I can hear you. For some reason, you got muted.
Yes, I wonder who clicked my mic button. It's probably me. This is from the chat. This is specific to African students, but you may want to answer more generally. Are there internships or workshops that are fully funded for students regarding bioinformatics, GWAS, and transcriptomics data analysis, possibly specifically? Any of that?
So obviously, through these programs, all the IMBRE programs have fellowships and support students, but really, they're limited to those state participants. So for instance, the Puerto Rico IMBRE would support Puerto Rico, Louisiana would support Louisiana through LBRN, and so on. At this point, we don't have fellowships for international students to participate in particular programs unless they're incoming students to any of the participating institutions here in the US. However, it is possible through philanthropic donations to NEPI, and we've had some funding before that could be something that we could work in the long run.
Very good. And in relation, the backside team helped me out with a career opportunities link on the NEPI website. So that was posted to chat for those of you that are interested in it. So we've got that link up there also. So make sure you check out the chat box. So how can all these programs from the whole program work together more efficiently? Is there a way to do that?
Well, this is why the IMBRE programs, which are really all of them, including the principal investigators and the coordinators, are meeting in New Orleans October 18 in a whole day meeting to actually find ways that we could communicate better together. There's already annual meetings where the principal investigators are meeting to discuss potential collaborations. We do participate in regional meetings because, again, 5 or 6 states have their own regional meetings. So we all get together and discuss different things. There haven't been formal ways to actually get programs to work together. Part of the difficulty is because the funding is limited to individual states. So we cannot use Louisiana funding to send our students easily to another state, and the vice versa. So it has to be some kind of an exchange where, somehow we'll be able to do this. Some of that has been occurring already. So we can send students, for instance, to the Mississippi proteomic training courses. But if you look at the NEPI website and you look at all these programs, it's pretty amazing the amount of resources for biomedical research that they have. And NIH is really promoting this synergy and collaboration. I think we started doing this, but obviously we could do much better.
So we've posted 2 links now, one at the Omics Logic website and at NEPI. So what are opportunities for students pursuing data science or other omics studies at NEPI? And are there formal certificate courses that can be obtained? Do they have to go for, please talk into that, please.
Yeah, one of the discussions has been whether individual programs, whether it would be LBRN for Louisiana, creates a certificate program. The difficulty there is that we have multiple institutions, and in order to actually get students enrolled and receive credit, a particular program has to be accepted by all campuses. So the alternative to this is to actually help through LBRN and maybe NEPI to have courses in individual campuses that are supported through NEPI or LBRN. I do envision, though, the possibility of having a NEPI specific certificate or a state-specific certificate. However, that certificate would be a NEPI certificate, not necessarily a full credit certificate that the typical higher education institution will give. And the enrollment for this would be hopefully partially supported by philanthropic donations, as well as registration that would be paid individually, hopefully at the minimal cost, to have as wide participation as possible.
Do the universities recognize the NEPI certificates as access to other programs, or like almost like gained hours that they don't have to get later?
They do not right now, because most institutions keep very close control of their credit hours. However, obviously these certificates do count for the job market, as well as potentially for incoming graduate students or preparation, advanced preparation, so they could actually meet expectations when they get into a regular credit-oriented curriculum.
I know little about these certificate courses, and I'm sure they may vary, but is there a time? Can a person get a certificate in 8 weeks? Is it 8 months? Can you speak to that?
Well, we are really at the planning stage right now, and the anticipation is that there will be a very specific program where you'll be getting different levels of certification by providing a variety of different modules that will start from very basic going to very advanced, and the certificate could be awarded at the different levels: level 1, level 2, level 3. Whether with the expert certificate, that type of thing. So that's something that we have been discussing, and we hope to launch maybe at the beginning of next year, after we get everything that we need to do established already, so we can actually pilot it first and then move it to having a bona fide program.
So what I hear you saying is that someone might be able to go a shorter period of time and get a lower level certificate, but then build on that, and build on that possibly.
Yeah, exactly. And I think, Pine Biotech has a lot of content right now. NEPI works very closely with Pine Biotech. Like I said, we hope to get some philanthropic donations to be able to allow people to register at a lower cost through NEPI, and then use the NEPI credentials to validate, those certificates at that level. Because don't forget, there are really practically 100 institutions of higher learning in 24 states that are basically support NEPI at this point.
Fascinating. So we already talked about internships and job opportunities, but what can a student do after they complete a training or get a certificate? Where can they find research or things to bite into? Any other
Well, it
resources?
depends on the level of the student. Obviously, if you're undergraduate and you seeking to go to graduate school, that becomes very important because you hit the ground running and you know a lot of different things. And depending if you go in engineering or in bioinformatics or data science, you'll be able to really just go through that curriculum very easily. Obviously, in industry right now, anybody that has credentials in data science specifically, but bioinformatics in general, sought out, and that creates a lot of job opportunities in the clinical setting, as well as on the research enterprises, where it be biotech startups and so on. So it depends. Ultimately this is a data science intensive environment. I think anybody that works in data science and bioinformatics, depending on the field itself, could secure probably a very bright future, either in the higher education, academic setting, or in industry.
Fascinating. Well, thank you yet again. We're going to go ahead and release you to the rest of your day. And we thank you for being here. We appreciate all the input on NEPI and all that you're involved in. And again, just thank you so much. And we will hopefully see you again. Thank you, sir.
Okay, thanks, Grant. And then anybody that may have some questions, please, contact either NEPI directly through the contact information, or LBRN, the Louisiana Biomedical Research Network. Okay, have a great day. Bye now.
Thank you. Well, yesterday I introduced him as one of the tech masterminds behind this symposium. Well, he still is. Ilya Brodsky is one of the co-founders of Pine Biotech and serves as CEO. His background is in applied mathematics and data science, and currently completing a degree in business and management from Tulane. But you haven't been working on that this week. But before I pass the mic to him, I want to remind you one more time to make sure you register at omnicslogic.com for your free learning portal. And as soon as Ilya comes on the screen, I'll post that link one more time just in case. And again, that's a free sign up, so go at it. So back to the matter at hand, Mr. Brodsky oversees the strategic development of research projects and manages the team responsible for bioinformatics analysis, training, collaborations for biotech, and a big part of the symposium. So ladies and gentlemen, let's kick off with Ilya. It's all yours.
Thank you, Grant, and thank you to all the speakers before me. I'm also going to share my screen. And I will speak a little bit on why we are here today. What is the goal of actually bringing together the whole community? And I hope to kind of connect the different parts of this symposium. And in general, this symposium really represents what we do throughout the whole year, even though the symposium only happens twice a year. So first of all, let me tell you a little bit more about what we do and how what we do fits in with what we see as a community consensus about bioinformatics training and kind of the current challenges and gaps and what currently exists. So one of the challenges I think we just heard from Dr. Kasoulas mention how different universities, different networks, different branches of the same network can have very different opinions about what bioinformatics training and needs are. And I think that there are ways to work more efficiently and faster outside of that framework of academic institutions to really address these challenges at scale, right? Because that's one of the problems here is that bioinformatics degrees, data science degrees, they tend to be really narrowly focused on this unique combination of engineering and biological skills or biomedical skills, and therefore exclude the majority of the students that are just not exposed to the domain and are not able to appreciate the way that this skill set fits into their future research. So another component here is that bioinformatics training is really not a single track. Not only is it a very dynamic kind of a field where all the time you have new methods, new skills, new types of data, it is also something that varies across many different domains. So for example, if you're learning oncology as opposed to applying bioinformatics in infectious diseases, maybe the types of skills that you're expected to have vary as you advance in those fields, right? In the beginning, they might look very similar, but as you advance, those types of databases, methods, maybe specific types of questions that you need to be able to answer, they start to be very, very different. And that's why we kind of arrived at this set of fundamental principles that guide our Omics Logic program. And that includes being able to combine several of these essential elements into a training experience. And those are the courseworks, practical activities, and case studies and projects, which really I hope that you were able to experience in these couple of sessions that we had over the past 2 days. Coursework, of course, relates to just traditional elements, modules, if you will, that kind of take a particular topic, data type, or a question and break down how that topic could be understood, mastered, or even just kind of applied in a specific way. Practical activities, a lot of MOOCs, online training coursework exists out there. You can watch lectures, you can learn about fascinating topics, but really you get to experience the component of problem solving and challenge, kind of addressing different challenges that arise from data when you start working on things with your own data sets and in your own kind of a project. So practical activities that we have include building pipelines and understanding the logic of bioinformatics pipelines, but also a lot of coding in both Python and R to really understand how to clean data, wrangle data, prepare it for analysis, visualize your data, and also apply different statistical and machine learning techniques, either specific to the domain of bioinformatics or really in a more generic way. And finally, this component of case studies, I think, is very important because, as I mentioned, these domains vary as you get more and more deep into the project. And so we've been working to source high quality curated data sets from publications that have an impact in a particular field or domain and prepare them as case studies that are open-ended. So you get started as a tutorial, and at the end, you can kind of use that data set in many different ways. So it introduces you to the topic, gets you into the topic deep enough for you to understand the context, but still the ending is open. And another, I think, challenge is to really have clear objectives and outcomes that are measurable. And so the way that we do it is we try to address this question of what do students need to know for them to get started, right? And I think that's a different question from what do they need to know to master a particular topic. Because the majority of the students are not going to pursue a full degree in bioinformatics, and they're not going to specialize with those bioinformatics skills to address a specific topic. I think what's more reasonable to expect is for them to be familiar with the way bioinformatics is used in various domains, and then to determine whether they will participate as a user, as an algorithm developer, as a technician, or potentially a person who is involved in the conversation, right? And so because of this dynamic technology and software and tools that are being developed, this is an important aspect to just introduce them to this field, I think, early on. Now, another topic that we already heard throughout this whole symposium as well is that, bottom line, you have to be allowed to not only play with kind of small demo data sets, but actually work with larger data sets. And that requires processing power for large data sets, and effectiveness of how your code is organized, and maybe help from a technical team that can prepare your pipeline for those larger data sets. So that's why we are kind of focused on collaboration with academic and industry partners to make sure that those tools are available as well. And I think finally, another challenge is with faculty, right? Because when we speak about introducing bioinformatics at an earlier stage, that means that the faculty have to be comfortable introducing those topics at an earlier stage to answer questions, to maybe, guide and monitor progress of students, evaluate them. And I think that's another set of challenges that really we would like to, find a solution for, because it is extremely difficult, I think, in some areas to attract faculty who are trained and well-versed in the domain of data science and bioinformatics, and ultimately having to deal with a variety of topics that they might not be prepared to do. So this is kind of like our operational motto, which is SOS, standardization, organization, is systematization, right? So how can we take fragmented curricula about bioinformatics applications in many different domains, find a way to agree on what all of them need to cover, and organize them in such a way that they could be systematized and implemented in basically any place, from top universities to universities that maybe are liberal arts and not exposed too much to the area of data science and biomedical research in general. And another aspect of that is really bringing it earlier on in this journey of a student, so that they can master and embrace the terminology and the kind of thinking behind data, obviously mastering skills as they go along and as they choose the specific direction and focus of their own journey and path. I think what's helpful in the context of organizing and kind of structuring these things is Bloom's taxonomy of learning, where we kind of see from basic to more advanced how the learning actually happens, and start to introduce that into what we call training in bioinformatics. And that means from kind of knowing terminology, knowing specific details and elements of what bioinformatics is, like genomics, how do you know what genes and mutations are, to eventually kind of growing in your ability to analyze, use tools and algorithms and techniques, and eventually becoming creative. And that's why we kind of organize the resources in such a way that anyone for free can register on our portal and start the learning process, and then develop skills that they would need, and a logic behind the analysis that they really need to potentially become experts, or just become aware of these topics and being able to try them at least before they commit to a long-term and demanding path to mastering bioinformatics and data science skills in full. Now, with the help of organizations like the National Association of Idea Principal Investigators, as well as many other collaborators that we work with, be it academic or industry experts, we have developed this core set of topics that we cover in our training. And that includes an introduction, a very high-level introduction to bioinformatics and different omics data types, deeper into different specific types of data that are commonly used, like genomics, transcriptomics, metagenomics, epigenomics, and then more technically into the area of coding and specific statistical and machine learning algorithms that we kind of put under the umbrella of data science. Beyond that, we also have divided the areas of our training into the domains of oncology, infectious diseases, neuroscience, and agriculture. Now, I realize that that might not cover every possible application of omics, as we heard even today, in beauty, wellness, in space, in other domains, but nevertheless, it's a framework for us to kind of start expanding from the basic set of skills into more specialized. And as we are able to build relationships and kind of find areas of mutual interest, we hope to further expand the number of specialization tracks and expand the number of project examples and experts that can lead those specialization tracks across the board. Excuse me. So one of the, I think, important parts of working with complex data is being able to utilize complex algorithms without knowing how to overcome technical challenges with coding. And so we offer both coding and no coding paths to mastering bioinformatics. And I think that is a prerequisite really for biologists to be able to get started. And we have quite a number of good examples of individuals that came from a life sciences and clinical background that are able to leverage such tools to get started, and eventually that builds enough momentum for them to take on a more technical skill that they see as a necessary requirement for what they want to do, and doesn't kind of introduce this feeling of, oh, I have to do a full degree in engineering now that I have already maybe multiple degrees in life sciences to just even get started, right? I think it kind of helps push them over the board, over that first threshold, and gives them momentum to keep going. I think that, again, this is more about kind of making sure that you're addressing a set of outcomes as you are planning these kinds of degrees. But I think this is very important for, planning and developing a strategy for effective implementation, right? When we come to outcomes and measuring those outcomes, we have to have a very good sense of what that is in the short term as well as in the long term. And I think that what we have discovered for our participants is that, this is a very long-term process. Someone might get introduced to a specific project in the summer and then have the time and availability to take a full course in the winter, or maybe complete it the next summer. And so that's why we have focused on kind of a profile that you build over time. And building that profile includes a technical measurement, like gaining points for completing coursework or practice exercises, as well as more of an open-ended portfolio that you can start building from, connecting your portfolio on LinkedIn, let's say, connecting your publications on OrcID, developing independent research projects and being highlighted for those projects, winning prizes or presenting them at different conferences. So really, that's where we hope that students that participate in such training can build out their personal profile, having that as a place that they can also share and apply for opportunities in research as well as industry positions. And so we kind of have these tools for tracking progress with points, as I mentioned, research assessments where we bring mentors to evaluate the efforts and the projects that students prepare, and then bringing together life and data sciences. What we mean by that is really trying to expand beyond a narrow focus of bioinformatics skill sets to be able to talk about actual biological questions or biomedical questions that are being answered in the context of mentorship and projects. And I think today was an excellent way to kind of see that in practice, right? All of the different projects that we saw that got the prizes, but also we have a full page on learnomixlogic.com where students have already submitted tens of projects that they have completed either individually or in teams. From around the world, from places like Louisiana State University, Georgetown University, Amity University in India, and universities in Ghana. So we really have this international representation of different teams and projects that have been completed by students working through these this kind of training. Now, as Grant already mentioned several times, this is free to get started. and really affordable, I hope, across the world to get into. We offer very low fees for a basic subscription, and I think very affordable fees for specialized training, but you can start for free. So I do encourage all of you to, if you don't have an account yet, go to learnomiclogic.com/sign up, and there you can sign up and create a profile. Now, another resource is our group on LinkedIn, where we post regularly job opportunities and opportunities to learn from experts in this field. So you can join there and also learn about those, build your connection and make sure that people know about you as well. Now, briefly, let's talk about this symposium and what we have done over the past couple of days. We still have a few speakers that are going to present today, but yesterday we heard about this need for bioinformatics training and research in India and in Africa, had an excellent discussion about, I think what we really see is that local efforts are almost as equally as important as international, right? And so instead of trying to say that, this is one center that will do everything for everyone, really what it is there's a community that needs to share and exchange opinions and kind of best practices to be able to build natural locations where this kind of training can happen. And also we heard about the need to actually go and translate those early efforts in training into translational research, where data from data scientists or maybe students or citizen scientists can be analyzed locally. So Indian challenges are maybe different from African challenges, different from challenges in the US, right? And so building out that community and pool of talent locally is critical for those communities. And so we heard quite a few successful programs that are being developed in those regions. And we hope that our regional teams in Africa and in India and in South America could be a part of that effort as well. Now, we also heard about the India Partnership Program, quite a number of universities that have signed up to that, which means that they will contribute their content and benefit from the type of resources that we make available. And that includes some of these great universities like Omics University and others there. We also heard today and yesterday from great speakers from both Louisiana State University or Louisiana Biomedical Research Network, from international locations like Dr. Joseph Borg, interesting applications of new technologies like single cell from Sweden. And we heard about the T-Bioinfo platform from the Tauber Bioinformatics Research Center. So it really was exciting to see how these different locations are offering interesting insights into different applications of bioinformatics. And we heard today, and we saw judges kind of give evaluation to student projects, which are starting to follow the lead of these experts, right? So we saw projects in space, in oncology, different applications to different kinds of biomedical areas of biomedical and basic research. And I think that was really exciting to see that we actually see this translation of ideas from, such presentation as this, where an expert speaks about what they're doing, and then students being able to pick up on that, find project or data sets and kind of develop their own independent projects. So I think that is really a validation and a path forward for us to move further into kind of building out this international community. So another component that we announced was the listing of jobs and opportunities for internships that you can find remotely in your area or by moving to a new location. So we have this new page that will continue to grow and expand job listing on our portal, and we will link it to your profile so that you can actually showcase your skills, your effort, and your project examples that you've completed to be able to be more successful at applying to those positions. I think these events kind of bring together an international community. So it was really exciting to see these projects that got highlighted as the best ones by independent judges that didn't even know about each other in some cases, but they have agreed on the best technical and research project altogether on a competitive topic, the most innovative approach in an established domain, and a promising research direction for future potential. And knowing these individuals, some of them, and interacting with them, I've really seen their growth, both in technical ways, but also conceptually, that they were able to not only follow somebody's tutorial and somebody's guidance, but also start bringing new ideas, not from, just being creative and kind of thinking about something, but from analyzing data. So really data-driven research projects that came from students who are, basically not very experienced yet, but have been able to do this nevertheless. So really exciting to see that. Another component that we don't talk too much about, but I think industry participation, right? So we heard a little bit about that, but we also heard about interesting opportunities for students that master data science and bioinformatics to be innovative and provide practical solutions to real challenging problems. So for example, in our last symposium, we heard from individuals that work in bioinformatics at the therapeutic companies like Camp4, or companies that are developing infrastructure for bioinformatics and for teaching bioinformatics like Google. Or we had hosted panel discussions and experts from the Faba Academy that talk about industry perspectives on bioinformatics and biotechnology in general. So I think that's another very important component to what we're doing, because ultimately there's not a place in academia for everyone to succeed in the long term. And I think that knowing about those opportunities outside, and also for companies to know that talent is being developed and is growing, that is also an important agenda for what we do. We will talk a little bit more in our next slide about the significance of taking bioinformatics training further to address these two important needs, and that is the diverse types of data that continue and develop. We heard today about nanopore. We heard yesterday about 10x genomics. Probably recently, most of us have been paying attention to Ultima genomics and the price of sequencing going dramatically down. That leads into the need for, I think, a broader framework for analysis, integration, modeling, and visualization of different types of data. And so that is a recurring topic and challenge that I think we will see being addressed in the future. And for us, what that means is that we really need to focus on making that continuous workflow from raw data to interpretable biological insights that can integrate effectively across these different technologies. And so I was really happy to see, Dr. Julia Panov and also Elizaveta Taranenko, who showed advances on the T-Bioinfo platform that we also offer as a tool in the training that we offer and as a commercial solution for our research partners. But that is really exciting to see that, this solution exists and continues to evolve and develop to address these important needs in single cell, or in the future in multi-omics integration, user-friendly multi-omics integration as well. Just a brief reminder of what we heard yesterday. We heard about machine learning and multi-omics integration in the area of single cell research from Dr. Nikolaj Oskolkov from Sweden. We heard about interdisciplinary forensic sciences, integration of omics technologies in forensics. And I thought that was really interesting from Dr. Sarah Zapico. We also talked about networks and graph theory in application to databases for patients in oncology. And we heard about precision medicine in pediatric oncology and the way bioinformatics is informing state-of-the-art in new emerging places like Brazil. So really fascinating set of topics that I hope we can use to turn into another set of case studies and kind of leverage these connections to offer students opportunities to explore these topics in much greater depth than what we heard there. Now, we also heard today about solutions in medical informatics, which obviously is kind of the other side of the coin when we talk about bioinformatics and omics technologies. It's really about how to integrate them in many cases into that clinical workflow. So we heard about predictive analytics, clustering, time series analysis, classification, and others. And I hope that those topics are really, something that ultimately converges, because omics technologies can inform a lot of those questions as well. Another presentation that was really exciting for me to hear is the presentation about the CPGR bioinformatics network, or this effort in South Africa, where we see them being able to both train, support research, but also include services into their model, so that they are successful, at addressing industry and academic needs. So really great presentation there. And finally, maybe one of my favorites was bioinformatics in the beauty industry from Dr. Raya Khanin, who talked about her company, but also in general, the need and interest in developing new targeted, personalized solutions for wellness and specifically for beauty. And I think that, that's a component that aligns well with what Dr. Kasoulas mentioned about entrepreneurship. Ultimately, states can invest, government funding and research funding into research projects, so getting more grants. But there's also a component of innovation and entrepreneurship that exists, and I think that's a way to translate these ideas and innovation and skills out of highly specialized and qualified students into this industry domain. So as a result of, partially this presentation, but also our internal planning, we are going to host a bioinformatics hackathon focused on omics wellness and also health. So we're going to make available some publicly available data sets that have been curated. We will make tools available for specific types of analysis related to this domain, and we will work with our academic and industry partners to organize this hackathon in November. So November 17 to 22, this year, we will host this hackathon, and then we will present the results of this hackathon at a later date. But the ultimate product here will be that each individual will be able to join a team, build an actual application, and then publish and share that application that we will host for 1 year with our industry partners, so that anyone can try and use that and you see that as an MVP, as a prototype for some practical use case in the omics wellness and health domain. So with that, I also want to mention that right after this symposium, our team is actually going to continue working on expanding our code or no code bioinformatics paths on the Omics Logic portal. And we are excited to be able to, bring this across the network that we heard about today, the National Association of Idea Principal Investigators, and work with those partners to make sure that it is more accessible and effective throughout the whole network. And because of these efforts, and because of gradual progress that we are making with these relationships, it is really exciting to be now seeing the results of this effort across the network. So we recently shared this update. We have individuals from University of Hawaii, University of Nebraska, Delaware, and others that have graduated from this training. So really, I think it's over 100 people that have gone through this training from across the network. And we are now also going to announce new research fellowship opportunities for students that have basic skills that would like to work over the next few months on developing independent research projects, leveraging bioinformatics and leveraging their skills that they will be able to use. And finally, my last slide, we're excited to be selected as the semi-finalists, for the EdTech Cup in New York, the largest EdTech accelerator in the US. So we will be sharing more information about this in the coming days, and we hope that you will support us in that journey as well. So again, thank you. And with that, I will leave some time for questions.
Great. Thanks again, Ilya. You did mention interest in Dr. Kanan's work. Is that due to need in the beauty area? I just thought I'd ask it. Never mind. So we wanted to get to some questions. Here's one from the chat, and this is a long one, so we'll go here. It seems that Omics logic is all about medicine, health, etc. It seems other domains are less targeted till now. Please, could you lead me where to find Omics related to the fisheries, aquaculture domain, environmental DNA, etc. And then, or how can we use the Omnilogics platform to bring improvements to the domains that I just mentioned?
Yeah, excellent question. So I think one of our challenges is actually finding a long-term relationship around those topics and significant interest in those topics for people to be able to kind of, fully develop and leverage those resources. So we do have people that have worked on fishery and environmental projects. We just don't have enough volume to turn those into actual kind of specialization tracks. So yes, you can still use bioinformatics, and Omics Logic provides resources to be able to do that. And also, yes, we would love to work with others who are, experts in those domains to prepare those specialization tracks. And I hope to see that just over the next few months, actually, because we have worked on those briefly over the summer.
Seems like Omics is touching literally everything. It's such a great post suffix, as it were. So what are some of the specific objectives taken for development, promotion, and improvement of data-centric research experienced by Omics Logic?
Well, one of the major objectives for us is to allow students to go even further. So right now, we're kind of at the stage where a student can develop their project and talk about it, right? Share it. What we would love to do is take it further by showing that they can actually develop solutions with those skills. And by that, I mentioned the hackathon. That would be one of the first steps in that direction, is to allow them to build applications that other people can use, leveraging these efforts that they have, analyzed data, created a model, maybe created a database or something like that. So really create a platform for applications. And then as we develop that, I think that will lead into the next phase where we will offer support to research projects or to even, innovative entrepreneurial ideas to take that further as well.
Okay. What are the bioinformatics skills that the Omics Logic Research and Training Program intend to enhance?
Well, like I mentioned, we want to provide a universal education about bioinformatics, but I think there's a lot of effort in that domain already. We want to provide specialized skills that are data science, coding, data management, data wrangling, data visualization, things like that. But I think what is important is that within that broad umbrella of skills is also the integration with biology or life sciences skills, application in this domain, and soft skills, right? Being able to think critically, come up with innovative solutions, communicate your results, things like that, right? So that's really why our program, I think, is different, is because we put that emphasis on logic and developing logic based on data, and also providing opportunities for them to interact, speak, and describe their projects as well.
Cool. I got one more for you. If we can keep it under 60 seconds, I'm going to challenge you. How can the student and research community benefit from the training provided by the Omnilogics programs platform to gain research experience?
I think the best way is to find a mentor. We have a network of mentors, and develop an independent research project. So the research fellowship is really the best program for that.
Great. Ilya, it was great to hear about all these updates, and it looks like we have an exciting path ahead. We hope that after the event, we get to further discuss the way all of these international efforts can be coordinated for the benefit of all the students and young researchers that are learning and applying data science and biomedical research. So thank you so much. And if Dr. Paulson can be available and go ahead and come on screen, he is an associate professor in the departments of Computer and Information Sciences, Plant and Soil Sciences, and Biological Sciences at the University of Delaware. His research interests lie at the intersection of genomics and microbial ecology, examining the ways in which microorganisms and their viruses affect and are affected by their environments. In light of COVID and other viruses, this could be a very relevant topic for our world. Dr. Poulsen, you are on the stage.
All right, great. And can everyone hear me?
I can hear you fine, and your presentation is showing fine, so it's all yours.
Excellent. All right, well, thank you to the organizers for the invitation to talk today. I put together a talk that dives a little bit into my research and actually kind of looks at a bioinformatics education thread with undergraduates that has kind of shaped my research program over the years, and we'll get into that in a little more detail. But as Grant mentioned, I am at the University of Delaware, and I wear a few hats there. In addition to my faculty role, I'm also director of Bioinformatics core facility and the Bioinformatics and Data Science core aspect of the Delaware Embry program. So I work with investigators pretty much across the swath of biomedical data science and bioinformatics in trying to figure out how to interpret data that they have, how to actually move forward their research using different tools, and how to educate people in bioinformatics. So a lot of what we saw in our last talk was great, really kind of hit the nail on the head with a lot of the topics that I'm usually talking about in my presentations. So as I said, I'm going to talk a little bit about my science today, but I did want to briefly mention kind of our bioinformatics data science infrastructure at the University of Delaware. I think the University of Delaware was really forward-looking about 15 to 20 years ago and making a really big investment in bringing in a strong bioinformatics aspect to the campus. And one of the unique aspects our bioinformatics programs have is that our campus, we are not associated with a medical school, which actually means that we have a lot of life science research that is going on that is not only in the biomedical sphere, but goes beyond that to agriculture, biopharma, bioengineering, chemical engineering. So there are just tons of related disciplines that end up feeding into our bioinformatics center that I am part of. So we have a really vibrant kind of population of people that are interested in omics, data science broadly, and our center, which has been around for about 15 years now, is both holding those core facilities that I mentioned that I host, and also a really strong educational program where we've developed a number of different graduate degrees in bioinformatics, data sciences that we have dozens, hundreds at this point of students have passed through our various graduate certificates, master's programs, and of course doctoral level programs. And we're really engaged in education throughout that entire realm. But one thing I did want to include in this talk is we are really excited that our already what we feel like are really good bioinformatics education programs have just gotten a huge boost from an NIH investment, which I am lead PI on this new T32 graduate training grant in computational biology, bioinformatics, and biomedical data sciences that we're going to be hosting at the University of Delaware, and actually just gave the launch orientation for that a couple of days ago. We're really excited about the opportunity to bring this in. It comes with a number of new slots for students that we can fund and bring into our program, but also is going to let us really work in those kind of interesting experiences with working with industry, working with government, and working with people in the medical industry as well. So I just wanted to put in a plug for this. We are recruiting for our next cohort starting next year. Anybody who's interested in our program, please feel free to reach out to us at the bioinformatics@udel.edu email address. So without further ado, on to the research talk that I wanted to give for today. So my entire career has been based around looking at microbes and what they do. And I think we've seen in the past decade or so that the role of microbes, the microbiome associated with humans and other organisms, they've really started to take a much more central role than they ever had in the past. So if we look at this kind of extent of genetic diversity on Earth, and we look at this tree here, you can see that this little red circle at the bottom, pretty much every organism that you can see with your eye is within that red circle. All the rest of this diversity, it's microorganisms, from bacteria to archaea to microbial eukaryotes. So it really is it's a microbial world. I don't think this is any secret. But even this tree does not capture that full extent. There's obviously one missing piece. What is it? Well, it's viruses, right? So viruses obviously have been very much in our consciousness the last couple of years with the COVID epidemic. But I tend to research more on a different group of viruses, which are the microbial viruses, which would be any virus that infects one of those microorganisms that I mentioned. And these are, by far the most diverse group of biological entities on the planet. If there is a gene on this planet, probably a microbial virus is carrying around some version of that gene. And they're probably one of the most poorly or least poorly understood groups of biological entities. I always hasten to call them organisms on the planet. And that's a lot of what my research has been based around, is how to figure out ways to understand viruses and how they impact their environment. So I have a joint research group here at the University of Delaware. I've actually teamed up with two other faculty members, Eric Womack and Jeff Furman, and we've developed a joint group that looks at different aspects of these virus, microbe, environment interactions. In that environment, really, it could be anything. I've looked at, this is just a small selection of things I have looked at, but it could be anything from, you see seagrass here, corals, seawater, but also humans, right? Humans are an environment, and a major environment for microbes. So looking at humans and biomedical models, agricultural models, you can see soybean here. These are the types of projects we do, but what unifies them is this interest in the microorganisms, how those microorganisms interact with these either environments or eukaryotic organisms, and then ultimately how the viruses that infect those microbes play into that. So it's kind of a tripartite system we're looking at. And to study this, obviously DNA, those types of omics techniques are the bread and butter of looking at these types of systems. So those are really kind of the unifying themes that go through my research. So, just as a basic background, viruses are really difficult to study. If you can go out and isolate a virus and sequence its genome, you're golden. You can do a lot of research with it, but it is very, very hard to do that. Even when we know what the host is, say human, it is often very difficult to go out and find specific viruses that are infecting that host, much less if you're looking at environmental samples where there could be thousands of potential hosts and thousands or tens of thousands of different types of viruses, and figuring out which virus goes with which host, and what can you culture the host? Can you culture the virus? So there's a lot of complexity in trying to study viruses. So DNA-based methods have really become important for looking at viruses. However, a lot of the tools that allow us to use omics-based techniques in other organisms, like marker genes that are universal, say the 16S rRNA gene that gets used in bacteria, don't exist in viruses. There is no one gene that every virus has. So it makes it quite difficult, quite complex to look at. So using shotgun metagenomics, which would be going out into an environment, taking a sample that has a broad mixture of organisms in it. Typically what we'll do is we will filter out the cellular organisms into a separate fraction so that what's left is just things of a size that should be viruses. And then we sequence that entire sample, right? So we get a huge amount of DNA sequencing data back, and it's about many, many, many different types of viruses that might be living in an individual sample. So then we use various techniques to go through and look at that. And some of those techniques can be quite computationally expensive, but as computing and other technologies have advanced, that's really not a limiting factor in doing these types of techniques at this point. At this point, really our limiting factor is our ability to make biological interpretations out of those genomic data that we get. I'd say in viruses, it's really hard because, as I mentioned, you can't culture many of them. So for us to actually have phenotypes in the viruses that we could potentially correlate with things we're seeing genotypically in that metagenomic data, it becomes a real challenge for us. So, you know, other issues that we run into are things like, if we look at any given environment and we metagenomically sequence it, and then we cluster all the putative proteins that are there, and we look at them from the most common to the least common, among the most common proteins, one study we have here, we found that 62 of the 100 most abundant putative proteins were uncharacterized. And of the ones, which is saying they were not in a genome at that point, and of the ones that were actually in a genome, all the ones that have arrows pointing to them here were, you know, they were annotated as putative uncharacterized protein, which might as well be uncharacterized or unknown for all intents and purposes. So this is a major wall we also run into, is that, you know, there's a lot of genomic data out there, but we don't even know what gene it corresponds to, so much less what type of activity it may have. So it's been an ongoing challenge, and probably one of the things that keeps me interested in this work is that it's challenging to go through these data and figure out ways to make meaningful biological conclusions based on them. So I mentioned, you know, undergraduate researchers have played a huge role in shaping this research program. And this is actually a slide from about 11 years ago. This is a program that we've had going on for quite a while. But what we started to do with my collaborators was to each summer recruit a cohort of undergraduate researchers into the lab. And we're lucky to have excellent summer undergraduate research programs at the University of Delaware that have helped us to do this and to bring these students in. And we really put them to work on doing metagenomic analysis, on doing some of the lab-based steps downstream of it, so that we can start to understand a little bit more about this diversity that is out there. So one of the main approaches we've used with them is we'll take these protein clusters that we see in metagenomes, we'll have them explore them, we'll have them adopt a particular cluster or a particular type of cluster. I have ribonucleotide reductase clusters kind of pointed out in this graph here. So one undergraduate researcher may take these and they will actually go through the metagenomic data, pull out the sequence reads for those that contain those genes, do assemblies, do phylogenetic analyses, ultimately build some primers so that they could look at this and try to actually identify it in environmental samples. And then, you know, take some environmental samples that we have in our library and see if they can actually re-identify these genes in those natural samples. And through doing this, it really increased our bandwidth to be able to look at all that metagenomic data that we were holding on to and really start to understand some of these genes that we see very commonly in the various metagenome sequences. And you know, we've definitely looked at both unknown and known functional genes through these types of methods. And we have interesting stories on both. Today, I'm going to follow through with one story that is from a known gene, which would be DNA polymerase 1. So if you're not familiar, it is the proofreading polymerase in many bacteria, but in viruses that carry it, it is the main polymerase. It is the one they use for replicating their genome. And it is a fairly common gene for a double-stranded DNA virus to carry. We see it in about 25% of known DNA viruses, double-stranded DNA viruses. So that's actually a pretty good percentage for us. So looking at this gene gives us the ability to look across a lot of different viruses. So one group that has looked at this, and there are a couple of grad students, Jess Chopik and Dan Nasko, also Eric Sikowski, who's not on here, and in particular, one undergrad, Helen Schmidt, who really adopted this DNA polymerase gene over the course of several years and made some really interesting discoveries with it that I wanted to walk you through. So first is a little bit of background to remind you, viruses and microbial viruses in particular tend to come in two lifestyles or flavors. There are the viruses that will enter a cell and immediately they're going to go into the replicative cycle, make a lot of copies of themselves and immediately lyse the cell. So those are the ones that are virulent or live the lytic lifestyle by default. The other group are the temperate phage or temperate viruses. Those are the ones that enter the cell, incorporate themselves into the host genome, will lie dormant for some period of time, and then eventually some trigger in the cell or the cell's environment will cause it to essentially activate itself and then do a replication. So understanding which viruses are lytic and lysogenic is a pretty important thing when you're trying to understand viral biology and how viruses impact their hosts. So it seems like this would be a very easy thing to identify within a sequence, but it's not always. And actually many phage that are out there, we have complete genome sequences, but we still don't know for sure which one of these lifestyles those phage tend to follow. So Helen Schmidt, as I mentioned, she was an undergraduate biochemistry major, and she came into one of our summer undergraduate internship opportunities and adopted this DNA polymerase I cluster and started looking at it in much more detail. And through her work and her particular interest in biochemistry, she noticed something within this gene that has led to 12 years of subsequent research for us. And that was the fact that within this gene, when you look at the protein sequence for it, there is an individual amino acid residue within it that seems to be predictive for the phage lifestyle. If we look at this residue at position 762 relative to the E. coli version of the gene, the default that you tend to see in bacteria is a phenylalanine at that position. However, if tyrosine is at that position, there is biochemical data out there that indicates that that makes the polymerase about 1,000 times more processive, faster, essentially, but also makes it more prone to errors while it's replicating. There's also a mutation that's been biochemically characterized to leucine at that location. And when that amino acid is in there, we get about 3 times more accurate, but about 1,000 times slower than the phenylalanine. So it's quite a spread in the speed and the accuracy that occurs based on this one amino acid change in polymerases. And what Helen noticed was that this individual amino acid residue, when she overlaid its identity onto a tree, very neatly clades with our groups with the clades that were on the tree. We can cleanly see there are 2 groups here that are leucine, but there's a single group each for phenylalanine and tyrosine. So to us, that was an indication, wow, there is a real biological phenomenon going on with this amino acid residue. And we really saw that when she started to look at the lifestyle of the phage that we did know their lifestyle. And that was that the tyrosine version was always lytic. We had no exceptions at the time of a lysogenic phage that had tyrosine. Phenylalanine was almost always lytic, a few exceptions there. And leucine was almost always lysogenic. So just this individual residue gave us that information that otherwise is very, very difficult for us to identify out of these types of sequences. So if we just do the thought experiment of why this might be important to viral biology, let's think about, lytic viruses are the ones that come into their host, they replicate fast, they produce lots of copies of themselves, and then they're out of the cell. So for those guys, if they have errors in a few copies, maybe that's okay. Maybe occasionally you're going to get a mutant, and maybe that mutant's actually a good thing, right? Maybe that's new diversity that's going to allow them to succeed. But if that mutant actually is bad, then it's okay. You've got lots of copies that are faithful to the original. And occasionally you're going to get a really crazy mutation, which may or may not be a good thing. But you have to see how that works out with natural selection. But the really interesting thing with this trait is you're modifying your polymerase, and your polymerase is ultimately responsible for the mutations that could lead to natural selection, lead to evolution. So really by modifying their polymerase in this way, or naturally selecting for the polymerase in this way, they're evolving to increase their rate of evolution, which that's a phenomenal thought, right? They're really giving themselves the ability to be more diverse. If we look at the other side of the coin, temperate phage tend to replicate slower. They're sitting within their cell for a long time, so they really need to be more in sync with the cell. And then when they actually do start to replicate themselves, they're doing it at a critical time when there's probably a stress on the cell, and they really need to get accurate copies and get out so that they can survive and reinfect a new cell. So in those phage, selecting for a more accurate mutation, this leucine mutation, even though it's a bit slower, may have significant evolutionary potential for keeping it in sync with its host. So for the rest of my talk, are actually one more slide on this. So the first students I talked about, they were doing that whole process very manually. We've actually since done a lot of work on streamlining it, making it easier, building bioinformatics tools. I've had a number of grad students who have come through, and we've just made an entire suite of bioinformatics tools that make this process of investigating these viral genes in this way very straightforward and easy to do. So for the rest of the talk, if we go back to this DNA polymerase A, we've looked at plenty of other clusters, but this one has really been one that has really captured our interest. And I mentioned this 1,000 times faster, but not quite as accurate, or 3 times more accurate and 1,000 times slower. Those are really, fairly widespread in biochemical literature, but when we really traced it back, they all went back to single studies where they tried some in vitro mutations. So really, we didn't know for sure that all polymerases followed this rule, or that it's something we would actually see in the environment. So a current study we have ongoing now is actually taking the bioinformatics back into the lab. And I have a student, Rachel Keown, who's been working with some collaborators at Verigen Biosciences. And what we've done is actually taken our metagenomic sequences for DNA polymerase A and have picked out different variants of them across the tree, represent these different types of amino acid residues that come from different sources. And we've had them synthesized, had the DNA synthesized, and then clone those into expression vectors so that we can actually produce the protein and do biochemical characterizations of it. So this was something a little bit out of our wheelhouse, and we were really hoping it was going to work. And fortunately, working with these experts at Verigen, it's working. We have actually taken, we've had about 50 of these synthesized at this point, and we've had about a 60% success rate of being able to isolate a protein out of it, and that protein actually having functionality. So, it's really cool that we can actually take a sequence from a metagenome and make a protein out of it. But what we're ultimately interested in is we want to find viral phenotypes, and the biochemistry of these enzymes ultimately determine how fast that virus can replicate, how fast it can copy itself and get out of its host cell. So understanding the different phenotypes biochemically for this enzyme is something really important that we can now layer onto other data that we have. So, I won't go into explaining this figure because I'm getting a little short on time, but the bottom line is, if we look through these different ones that we have synthesized, we have found quite a few of them that we can show have primer extension activity at various speeds, various rates. We also have several of them that have a strand displacement activity, which is an interesting activity on its own that our partners in industry are very interested in, but we also are interested in is that it's another phenotype that organism has, or that virus has, that it's carrying around that we can potentially look at. So Rachel is moving forward with this work. She's gone a little bit beyond only looking at this biochemical data that she's been producing, but she's also looked more at what else goes with the polymerase. It's not living in a vacuum. It's ultimately working with other genes to replicate the genome. And we really see that there are some really constant associations between our different tyrosine polae, phenylalanine polae, and leucine polae with different types of helicase genes that are specifically tuned to work with individual ones. So this is another piece of evidence that this processivity accuracy hypothesis that we've been testing may well be true, in that GP4 helicases, for instance, are known to be among the fastest helicases out there. And if you're unwinding the DNA for a very fast polymerase, you would think you're going to need a very fast helicase. So that we're seeing these associations is very promising, and we're now starting to move beyond the pol A and also look at the biochemistry of these accessory genes like helicases, like ribonucleotide reductases that we see in association with these, and look at that broader genome net neighborhood and figure out how that module of genome actually impacts the biology of the phage. So where are we going with this work? Ultimately, Rachel's still working on purifying and biochemically characterizing those various enzymes. She's worked with another group of undergraduates this summer in doing optimal pH and temperature assays, which can actually tell us a little bit about the biology of the cells that the phage are infecting based on where their optimal amplification or where their optimal operational temperatures and pHs may fall. As we're expanding this database of biochemical information about different phage from different phylogenetic parts of the tree, we're correlating that data with other information that we either have based on where they were isolated, or abundance and infectivity information that we're producing in other areas of our lab through various collaborations. And our ultimate goal here is we're going to take this massive data, both on biochemistry, observable phenotypes when we can get them, as well as the genotypic features that seem to correlate with these. And we're going to integrate them together and begin to see if we can make models that will allow us to predict phenotype more accurately based on the genotypic features that we've seen within these replication module proteins, and how well it can interpret or predict these types of infectivity characteristics or environmental characteristics that may apply. And we've done this on some fairly simple basis so far, just looking at combinations of ribonucleotide reductases and DNA polymerases that we see occurring together. And we've actually had some really interesting results come out of that, where we can see correlations between versions of ribonucleotide reductase that require oxygen versus don't require oxygen, and the available oxygen or metal cofactors that are available in their system. So we have some great preliminary data here, and we're really hoping to build a more substantial model moving forward with our work. So I think I am wrapped up now. Thanks to everyone in my lab, my collaborators, and of course our funding sources.
Thank you, Dr. Polson. That is, again, fascinating stuff. If you don't mind, you can quit sharing your screen so we can see you. How are these in silico tools like AlphaFold for research you're working on? How useful are they?
They're actually quite useful. I actually have a grad student, Zach Schreiber, and a collaborator, Madi Belkade at the University of Hawaii, and they are looking at, particularly at protein structure that we can predict with these new in silico AlphaFold 2 and so forth, and how well that can be used to classify these proteins in metagenomes. So that's actually a different direction we are going, but it is something we are investigating in this area.
Very fascinating. How important is mentorship and student participation in this research? You've mentioned a whole bunch of names that aren't yours during this.
Yeah, absolutely. So, student participation is, our research could not happen without it, both undergrads and grads. And we really try to set up a kind of a group support mechanism. We don't just take an individual student through, we bring through a cohort of students, which brings a support mechanism with it in itself. And then we pair those undergraduates with graduate students. And then we have mentor PI meetings with them on a regular basis. So through that kind of multi-level mentoring, it both teaches graduate students to be mentors, but it also sets up multiple levels of support for those students as they move forward to get at these types of new skills that they're trying to learn and apply.
Thank you so much. I've wrapped up our time with you, and we really appreciate that. I think we could ask questions for another half hour if we could, but we need to move on. So thank you again for your participation here this afternoon. We really appreciate it. Thank you.
Thank you.
Our next featured speaker is Dr. April Wright, and we are really happy to have her here. She's currently the Assistant Professor of Biological Science at Southeastern Louisiana University. Dr. Wright has several currently funded projects in the lab on deep time evolutionary dynamics in a variety of groups of organisms. Secondary focus in the Wright Lab is domain-based education research in computational biology and how biology undergraduate students learn computation and mathematics. Dr. Wright, I can't wait to hear what a Bayesian model is and the latest fashions they're wearing. Oh, that probably isn't the right kind of model, right? I'll stop guessing and let you go on. Go right ahead.
All right, thank you. Really happy to be here today. I'm going to try and put this into slideshow mode and see if you can still see me advancing. All right, can you guys see me advance my slide, or are you still seeing the title?
I can, and we can hear you just fine. Everything's good.
Perfect. All right, well, thank you so much for having me. It's really nice to be here today. So today, I'm a little bit of a different type of scientist than your other past couple of speakers. So I'll be talking a little bit about some of the mathematical modeling that we do in the lab. There actually won't be any genomic data in this particular talk, and instead we'll be focusing on what we do with models and how we can do that confidently. I just realized I have the wrong year in my slide. It is not 2021, but it feels like it's been a lot of years lately, so that's fine. Like I said, I'm a little bit of a different biologist than the other speakers in this session. I don't really do very much in the way of genomics. And in this lab, we predominantly work on complex and particularly hierarchical models for estimating phylogenetic trees, particularly from the integration of a variety of data sources. And Ilya had asked me a little bit to talk about how I ended up interested in something like this. And I didn't start interested in phylogenetics at all, in fact. So when I went to college, I started taking intro bio classes, and I was really bored by that. You know, you memorize the Krebs cycle, and you know, you have to know all this stuff. And so I was bored by that. And I ended up dropping the biology major and picking up a journalism major, actually. But being the only person on staff who had any sort of STEM background at the newspaper, I got handed a lot of science articles. And this was back in 2006, when a lot of the challenges to teaching evolution started to happen. So, for example, the Dover v. Kitzmiller trial and things like this. And so it was a really interesting time to be writing about and thinking about evolutionary biology. And because of that, I actually realized that biology wasn't just memorizing a bunch of stuff and regurgitating it onto an exam. It was a living discipline. And I became interested in the the role of phylogenetics as sort of something that would allow you to study a little bit of everything, right? So you can build a phylogeny of fish, you can build a phylogeny of mammals. We work on ants in this lab, we work on stem mammals in this lab, we work on things that have been extinct for 500 million years. We work on things that you could walk out your front door and collect on your doorstep. So I got interested in phylogenetics because it was this way to get to do everything a little bit at once, as well as a way to sort of engage with the deeper philosophy of biology. And when Ilya and I had a conversation about this as well, he wanted me to talk a little bit about primarily undergraduate institutions. So when I went to college, I went to an undergraduate only institution, St. Olaf College in Northfield, Minnesota. And I decided that I really liked sort of that modality of education. And I decided to pursue it following my PhD. So I've been here at Southeastern Louisiana University. This is the start of my 6th year. I was awarded tenure last year. And so I feel like the balance of responsibilities that I have in this type of lab is very good. I teach 3 courses a semester, I have a couple of master's students, and typically 4 to 6 undergrads in my lab. And the work that you're about to see has been sort of touched on by all of those personnel at least some points. All right, so what we have here are 2 phylogenies. In panel A, this is a phylogenetic tree that is what we refer to as not scaled to time. So in this case, typically you're going to see a tree like this. You saw some in the previous presentation where the branchlings are usually given in a rate, right? The number of substitutions expected per site. So a measure of either the genetic or the morphological change, whatever you happen to use for your data. On the right-hand side is a phylogeny that has been scaled to time, or what we might call absolute or geological time. These types of trees allow you to make, like I said, absolute or geological statements about when lineages diverged. Did they diverge 10 million years ago, 5 million years ago, 100 million years ago? These are the types of questions that we can answer with a time-scaled tree. And over the next couple of slides, I'll show you a couple of applications of these types of phylogenies in the literature. So for example, right now, we might all be very familiar with this. This is a screen grab from the website NextStrain, where they're tracking different variants of COVID, right? And you can see there's a time axis on the x-axis of this. They're keeping track in absolute and real time of what strains are arising and how they're diversifying. You can see the different strains are in different colors here, with some of them labeled by sort of their common name. And you can see over time, some of them sort of go extinct, some of them continue and diversify. So for example, Omicron right now is sort of the dominant strain. You can see that dominance kind of over here on the right-hand side of the phylogeny. So this is an example of a time-scaled phylogenetic tree. And another example here, this is an example from scallops, where they've built a phylogenetic tree of scallops. And what they're looking at is, if you've never looked closely at a living scallop, they're bizarre. They have a bunch of eyes on them. You can see by the scale down at your left, the eye abundance goes from, black is like 37 eyes up to 151 eyes in the blue there. And then on the kind of right-facing phylogeny B, we have the life habit. So are they abyssal? Do they sit on the ground? Do they really attach to the ground, so cementing? Do they free live? Do they glide? Do they do other things? And so this is the study of trait evolution. How does the evolution of the eye impact the evolution of life history? And these types of studies typically also assume a branch length in sort of millions of years, so absolute or geological time. And so how do we get a phylogeny with branch lengths in millions of years of time? And that is sort of the real question. The family of methods that I work with are referred to as the fossilized birth-death model. And it has 3 different phylogenetic components. For this reason, it's been sometimes referred to as the tripartite model or the tripartite approach. So under this model, we have our data. So our phylogenetic characters, these could be morphological characters, these could be DNA characters, and we have our fossil ages. And fossil is a little bit of a general term. If you're working with actual organisms that might fossilize, then yes, you have fossils. If you're working with viral data, these would just be the ages of the thing that you isolated. So if you did a blood draw and you found that somebody was carrying HIV and you wrote down the date, that would be your fossil age. And this model has 3 different components. There's the substitution model, so how your phylogenetic characters evolve. The clock model, which describes the distribution of evolutionary rates over time, and the tree and tree models. And this describes the process of diversification that generates your actual phylogeny. And what I mean by that is this. This part of the model is referred to as the fossilized birth-death. What we have in the middle here is this character T that represents the phylogeny. And you can see there are 5 different parameters that are all pointing towards the phylogeny. These are the parameters that govern the tree shape, how many branches are on the tree, so how many lineages are on the tree, the age of the tree. We're going to walk through them. So right here we have the origin time. When did the process of diversification that leads to your observed tree start in your group? The speciation rate is what is the rate at which new lineages are added to the phylogeny. The extinction rate is the rate at which they are removed. The fossilization rate is the rate at which we recover lineages in the past. So this could be via, sampling viral loads. This could be the rate at which fossilization occurs in your group. And then the sampling probability. Of the things that currently exist in your group, how many of them have we sampled? Right, this is, you'll notice that this probability, the sampling probability, is in a square. That refers to this parameter being fixed. We typically assume in this model that you know how many of your clade of interest you've sampled. So if you have, you're estimating phylogeny of bears, there is genetic data out there for every bear. You can include every bear in your sample. And so your sampling probability would be 1. For many groups, you probably haven't sampled everything, but you might have some good idea of what proportion of the extant diversity you've sampled. And different tree shapes are going to different model parameters are going to imply different tree shapes. So for example, if we are not including, any tip sampling at all, that would sort of imply that we are going to sample absolutely everything. As we, increase or as we increase our extinction rate, that impacts the tree that exists in reality, and it impacts the tree that we can recover from our data. Continue on there. Right. So I think a lot of us are fairly familiar with Bayes' theorem. We have that right here. We have the posterior probability on the left-hand side of the equation, which is kind of the probability of parameters given data and a model. On the right-hand side, we have the likelihood, so the probability of the data given your parameters in a model. Any priors you have, right? Prior probability distributions that specify, knowledge that you have about those parameters. And that's all divided by the marginal probability of the data, which is sort of a complicated quantity to get a handle on. And I'm not going to spend too much time on it here, but if you have questions, I'm happy to answer them. So in the case of the fossilized birth death, in a phylogenetic context, that equation looks something like this. And there's a lot of stuff on the screen. So this is a little bit tricky to deal with. And so we're going to take a look at how we can sort of break this down usefully to do good model selection for each of our 3 hierarchical submodels. And I'm actually going to present to you some results that we have from actually an entirely paleontological data set called the Synctons. These are an extremely temporally restricted group of organisms. These are invertebrates, marine invertebrates. And they're found basically from about 510 million years ago to about 498 million years ago. And that's it. So these have a pretty good amount of phylogenetic characters for a small invertebrate, about 60 characters for those 27 taxa. And something that's interesting is that we have some idea about how diverse they were over time. So I'm going to call your attention to panel C on this one, where basically between the first 2 points, this one down at the origin and then this one over here at about 6 million years, there's a huge jump in what we call the disparity, and that's an index of how different organisms are from one another. And so very early in this group, we accumulate a lot of morphological change. Things begin to look very different very fast. And so that's a very common pattern in the fossil record, sometimes referred to as the early burst. And so this might imply something about our distribution of evolutionary rates. In fact, it might imply that they are very quick at first, and then they slow over time. And so we decided to test 4 different molecular clocks. The strict clock says the rates of evolution are the same all across the tree, all through time. Probably not true in our case. The autocorrelated clock says, well, the rates of an ancestor are going to be similar to the rates of its descendants. Okay, that seems plausible. Early burst, like I just said, it kind of looks like our data is that. And then the uncorrelated means an ancestor and a descendant can have strongly different rates of evolution, no limits, sky's the limit, do whatever you want. Right, so again, that's sort of this is a visual representation of that, where you can see on, the autocorrelated, generally the cooler colors are the low rates, and generally you have to kind of pass through intermediate rates to go from low rates to high rates. On the uncorrelated plot there, you can see when the branches are colored by evolutionary rate that you can have jumping from very low evolutionary rates to very high ones, no problem. Right, so we had a look at those. And something that's also the case here is that we have pretty good geological information about the time period when these guys existed. So for example, we had pretty rapid accumulation of lineages here in stage 5, and then slower lineage accumulations in these stages. And so, while we might expect evolutionary rates to change over time, this could potentially also be implicating the process of diversification over time. Perhaps it's that we're diversifying very rapidly early on and therefore accumulating disparity. So perhaps it's not the rate of evolution, but the rate of diversification. And so we took a little bit of a look at this and got some overall estimates from extinction rates and origination rates or speciation rates. And this allowed us to sort of have some idea of ballparks for these sorts of models. So we're able to use these to place informative prior distributions on each of these values. And we use a set of models that are referred to as skyline models, which allow us to have a different kind of diversification process for each of our time periods. So we had a look at dividing up the time period into 2 chunks, but also 3 chunks. So each of these individual stages getting its own diversification process. So we're not totally out to lunch on at least 2 of our models, right? But what about this part here? So most of the time we're going to be working with the MK model of Paul Lewis. And that's pretty much what we did in this paper. I'm starting to run out of time, so I'm just going to keep moving. We also looked at, if feeding and non-feeding characters evolve differently. So from kind of an infinite set of models that we could have assembled from all of those parameters, we were able to distill down to about 10, which is great. We used a constant data set of fossil and phylogenetic characters. We did all of our estimation in a model or in a software called RevPhase, which is something that my lab works on for development. And this allows us to calculate a precise model likelihood to make comparisons between models. And what we found is that for each of the hierarchical subcomponents, there was one clear model that was best. All right, so for each we could fit a best fit model and use that to infer our phylogeny. So our uncorrelated log normal model was the best for the clock, which I was a little bit surprised by. And a model in which we have diversification changing per stage was also good. Right. And so this is computationally expensive, but we were able to get things to work a little bit better by not evaluating every single model and instead choosing models that have some prior evidence for them. Right. And so when you have these types of extremely complex models, that really calls for a return to hypothesis-driven work. Right. Why do we think this is the right model? What is our intuition telling us? Right. And so we were able to estimate a phylogeny, but there's some stuff about this that's intrinsically unsatisfying. So I mentioned earlier that we could have an early burst model that manifests in either the clock or the tree model. What if those interact? Are these subsets independent? And is it valid to do model selection on each of them? And my answer is a little bit no. And so my lab has been piloting what we call reversible jump model selection. This allows us to sample the number of model parameters as part of a Markov chain Monte Carlo analysis. And so in this case, we could have subsets of models being selected at once. So we could be doing the model fitting for each of our submodels together rather than separately. So we can sample models simultaneously to determine the correct model for all of the sections together. And finally, the other technique that my lab has been piloting is the posterior predictive model adequacy testing. These are a family of methods that allow us to simulate data from our Bayesian posterior predictive or Bayesian posterior distribution, and then see if when we simulate those data, they look like the empirical data. And if they do, we have some test statistics to describe the goodness of fit there, and they're assumed that then the model is sort of adequate for the data. I'm running out of time, so I'm just going to sum this up really quickly. With these new hierarchical statistical models, we have non-independence of model subsections that could be misleading for our analyses. And so we're kind of looking at evaluating a new generation of tools for their effectiveness for dealing with these sorts of problems. All right, so thank you to all of these right lab members and collaborators, as well as to funders. And I think I am out of time.
Well, not quite. You actually have a few more minutes. We have about 10 minutes we can fill up. So we're going to ask you some on-the-spot questions here. Are you ready?
Yeah.
All right. You can stop sharing your screen there if you would.
Yeah.
And that way we can be just, there you go. Now we can see you. So a question from chat and stuff. You mentioned that you are not working with genomic data. That was at least what was understood. How do you validate that model predictions, or how important or even possible it is to validate the models?
Yeah, so we work with a lot of simulated data in the lab and try to simulate data that looks like, you know, the real data. I mean, and that's exactly the same as you would do with genomics, right? So we work with a lot of simulated data for model validation. And then we do quite a bit of sensitivity testing as well. So, you know, you run the model and then, you know, you take a look at how well it's kind of working with your data, and you run it with maybe some different assumptions and see how much of a difference that makes to your actual conclusion. And then the other thing that you can do for kind of model validation would be the posterior predictive model adequacy testing, which is that very last thing that I kind of thought that I was out of time to discuss. But the idea with this is that you take samples out of your posterior distribution, simulate data with them, and then you use test statistics to determine how closely the correspondence is between you know, the data that you've simulated under your model and your empirical data, right? So if they look very similar according to your test statistics, then that's, you know, a good sign that you're probably modeling the correct things.
Okay, well, thank you. What are the limitations of using hierarchical Bayesian models?
I mean, I would say that the limitations are mostly sort of, actually, we had an annual reviews paper that came out on this last week. But there are lots of limitations, and a lot of those limitations are computational. So when it comes to Bayesian methods, you know, we're really looking at the idea of, you know, having these models converge to a stationary distribution where you can assess that you've drawn enough samples from the posterior to approximate it. That takes a long time in practice for complex models. So there's a time sort of factor, and then the computation time associated with that. It's very hard to do this type of work, for example, in the cloud. So if you don't have, you know, a server or an HPC unit, a lot of this isn't at this point kind of out of your reach. It's not something that can effectively be done via AWS or anything like that. So there's that. That's a big limitation. Another limitation is sort of the quality of information that you can come up with, right? So let's say you have, you know, genomic data, but there's no fossils in your group. Your ability to have any sort of resolution in the past is therefore extremely limited. And there are some groups where that's true. Fish, for example, are one where, because of the nature of sort of the sedimentology and how they get deposited, it's pretty hard to tell when a fish is from. Other soft-bodied organisms are also quite tricky. But, you know, if you're working with something like disease biology, you often have a lot of extremely high certainty data because you know what day the patient came in and you sampled their flu strain, right? So in the biomedical realm, methods like this are often extremely effective. And actually, one of the founders of this kind of series of methods was on Swiss national TV every week for the coronavirus update because they were the ones who were tracking it. So you encounter a lot less limitations when you're looking over those short timescales, like a couple hundred years or a couple thousand years for a disease-bearing organism. I think the other big limitation is just your ability to think about all of these things at once. You know, it's complicated. It really is. And developing the statistical literacy, the computational literacy, knowing each of your data sources and its individual limitations, that's a lot to ask of anyone. So I think, yeah, just our ability with human brains to think about these things is considerable.
That's been my greatest impression over the last 2 days is just how everybody's keeping all this even remotely close to what they can think of. Okay, here we got one from chat. Can the models be used to determine gene evolution for potential vaccine candidates for tick species?
I mean, I don't see why not. So for a long time, phylogenetics has been a component of the flu vaccine selection, right? And so this was initially described back in the early aughts with Mike Warby et al., where they would show that, you would get oftentimes kind of a trunk lineage that would give rise to the flu strains for that year. And so phylogenetics has a deep kind of history with that. And they typically do that based on a couple of loci associated with sort of the host genome evasion response. And so, yeah, definitely. I think that sort of thing is very possible. So you can build individual gene phylogenies and see what they're telling you that might be different than the whole kind of species tree. So oftentimes there's conflict between what individual genes will tell you versus when you look at them all together, what they'll tell you. But those techniques are very well explored. And so I think that would absolutely be possible. Yeah.
Okay, thank you. What are the ways you can combine hierarchical Bayesian trees with other methods like your standard neighbor adjoining tree, etc.?
You can't.
There we go. That was a simple answer.
Yeah, I don't normally like to give answers that simple, but yeah, doesn't work with neighbor joining.
Okay. Here's another one from chat. Have they used K nearest neighbor models against real data for synthesizing data? Has this included both clinical and omics data? For example, using Cynthia patient data with 1 kg genome data or using UK biobank data with clinical phenotypes, etc.?
I have never heard of any of that. So
you and me both. So we're
potentially someone else. Yeah.
All right, very good. If it has limitations, then what is the biggest motivation to use it?
Well, I mean, everything has limitations.
Right.
I mean, if we didn't do things that had limitations, you wouldn't have a computer, right? Like we wouldn't be in this meeting.
Carving in stone would make this very interesting, right?
Right.
Okay, so what
exactly.
prompted you to focus? What prompted you to focus on this specific topic of research? And did you consider other domains also? Why did you get this direction?
Yeah, so for me, I think like most kids, I grew up watching, dinosaur TV shows and reading books about dinosaurs, and you think you might want to be a paleontologist, but that's a pretty tough career path. Whereas going into the computational side of things, you get to do a lot of different things in a day. And so, when I was first thinking about, evolutionary biology seriously as a career path, I looked at lots of different things, functional trait studies, kind of evolutionary ecological studies, all sorts of stuff like that. But then as I started to think about kind of, well, how do we integrate those things together? Phylogenetics kind of kept coming up as sort of the organizing framework in which we study the evolution of traits in organisms. And so I got interested in phylogenetics that way. And then, the statistical problems that we have in phylogenetics are extremely difficult, and hard problems keep us in jobs, and hard problems are interesting. And so that's how I ended up here.
Nice. Well, we're within a minute of our official closing time. So instead of potentially starting another question that we can't answer and working into Emmanuel's time, I'm just going to thank you for a fascinating, interesting decision-making, modeling, and how it all relates. Thank you so much for being here today. We really appreciate your time.
Thank you for having me.
You're welcome. Bye-bye. We all know this field is in constant change, and it's defined and pulled and twisted and redefined. And our next speaker, Dr. Emmanuel Ruiz, will discuss how omics data is changing our molecular applications. Dr. Ruiz was awarded a PhD in molecular and cellular biology. And throughout the years, she was confronted with the need for computational biology tools in order to answer biological questions. And she had to acquire an expertise on bioinformatics and biostatistics. Dr. Ruiz, can't wait to hear what you got for us. You're on stage. Can you turn your video on, by the way? I cannot hear you or see you, Dr. Ruiz.
Here we are. Hello, sorry.
There you are.
Let me try again. All right.
Yeah. I can hear you fine. And if you want to go to slideshow mode, that would be great. There we go. You're
It's
on
perfect.
your own.
Thank you.
Good luck.
Thank you. My name is Emmanuel Riz, and I am a senior postdoc bioinformatician in the Veterinary School of Louisiana State University. It's an honor today to present my work experience from the bench to the computer and how bioinformatics with omics data have drastically changed my career path. So I was born, raised, and got my education in Lyon, France. I completed my PhD in the Cancer Research Center of Lyon, and the main purpose of the research project was to decipher the interaction and the collaboration between the epithelial mesenchymal transition regulators with oncogenic stresses that lead to mammary transformation. This research project was initially a basic project with bench or in vitro data that would have been validated in animal models and with luck, finally, in human subject. However, as you will see in the next slide, very soon the bench results obtained could not anymore be interpreted or analyzed further with basic molecular or cellular biology tools. The goal was to understand if and how the EMT transcription factors were collaborating with oncogenic stresses to transform non-cancer mammary cells and compare these new oncogenic abilities or properties between these EMTTFs. The methodology chosen has been a screening where the 10 transcription factors will be compared in 4 oncogenic stresses conditions. The deletion of P10, of P53, the overexpression of EGFR, or the mutational activation of beta-catenin. That are all oncogenic stress found in breast cancer. So briefly, the virus contained the 10 transcription factor sequences were produced and infected in mammary cells that were already altered by an oncogenic stress. The original mammary cell lines did not possess oncogenic properties or transformant abilities, and adding this oncogenic stress was not enough to make them cancer cells. The infected mammary cells were grown in a soft agar plate that mimic the transformation process. And only cancer cells or transformed cells can grow in soft agar. After several days, clones were collected and amplified, RNA were extracted, and PCR were run in order to identify which EMTTFs have been overexpressed in the clones. In parallel, functional tests as soft agar, proliferation, and migration assay were performed to compare their oncogenic properties. Initially, we anticipated to analyze clones that have maybe 1 or 2 transcription factors overexpressed. However, because of a screening, we obtained clones that contain from 1 to 6 at 7 transcription factors per clone. And furthermore, the functional tests were showing that these clones presented a high diversity of transformant, proliferative, and migration properties. So a bigger picture of what was originally saw was building up, and all this information have to be computed and analyzed. But how? And here is bioinformatics and biostatistics were the keys. And so we collaborate with a bioinformatician, and we gave her this simple Excel files that contains the combination of the EMTTFs in the column for each clone that are in the row, and for the different conditions, transformant and non-transformant, and for the specific oncogenic stresses. We ask a specific question, as we all the time have to do when we talk with bioinformatician, one question at a time. Which EMTTFs combination or signature were the most associated with a specific oncogenic stress? She thought that a tree-based model classification could help to answer our question. But at this time, so 10 years ago, more or less, the trees that we obtained didn't make sense for me, and I was quite confused on how to use them in order to answer this biological question. So I decided to go deeper in the tree classification process and understand the different steps of how they were built, and how we had these complete trees. At this time, I only have Excel to help me, but I succeed to find a meaning, and also I succeed to create a new visualization that computes the number of the EMTTF that we see on the circle, the colored circles, the EMTTF signature, and also that predicts the importance of the rank inside this signature. And so finally, also, this visualization could predict the ability to drive mammary transformation represented by the width of the cycle. The triangle shows that their transformant abilities depending only on their rank if there were not other parameters altering these transformant abilities. And we could see that we can see on the visualization that the first EMTTF that have the strongest ability to predict the transformant properties of the cells will be on the top of the triangle with the first on the signature, and the one that has the least abilities will be on the bottom. However, when we put them together, we saw that we had a different signature according the transformant, according the oncogenic stress, and also inside the gene, this EMT signature, the transcription factors have an importance that were different. And so we could identify specific EMT signature according the oncogenic stress. This analysis was the beginning of understanding the bigger picture of the interaction and collaborations between EMTTFs in the context of a specific oncogenic stress during mammary transformation. In parallel to this project, the years of my PhD were also associated with the emergence of transcriptomic and genomic data online platforms. It was a decade now that the first human genome had been sequenced. Different institutes like the National Cancer Institute, the NCI, and the National Human Genome Research were joining forces to propose genomic, epigenomic, transcriptomic, protein, and also clinical data on cancer tissue with the TCGA, the Cancer Genome Atlas, also on cell line with the Cancer Cell Line Encyclopedia. And all that for the international research community with a free access. The possibilities to validate and understand the bigger picture of what we obtain from our bench results were now within everyone's reach, even the ones that didn't have any bioinformatics expertise. My bioinformatics curiosity, already initiated by my own project, were fed by my boss that wanted to use these new tools, and I took the challenge to learn how to use them. What I didn't know at this time is I will fall in love and be addicted to bioinformatics. The bench work, as maybe a lot of you know, where we can spend hundreds of hours trying to accumulate results and dispose them as a kind of puzzle pieces, were not anymore very exciting for me. And I largely prefer to spend the same amount of time resolving bioinformatic puzzles. So as I said previously, I was using Excel as the first tools, and Excel showed very quickly its limits, and I had to learn from scratch real bioinformatics and biostatistics software like R. So I didn't take a university classes, but already at this time and more today, there are excellent tutorials online that are free or not much expensive, and forums where I could and I continue to ask the dumbest question without any shame, and people are very patient to answer them. And so all this, the fruit of this effort led me to have different collaborations during my PhD, and one of them led to a publication in Nature Medicine. So I was quite encouraged by that. So after that, I left France and worked as a postdoc fellow in the University School of Medicine with Dr. Kendall as a PI. Dr. Kendall is an endocrine surgeon specialized in thyroid cancer. And so my research work was strongly driven by a clinical perspective. it was actually a very good training to never forget that my research project, even is driven by a strong curiosity, have all the time to have in mind and that in the end, the research project have to help patients. And so today I would like to share with you 2 projects that show how much bioinformatics is a tremendous help to answer clinical and translational questions and problematics. So the first project is related to cancer and aging. So cancers are aging disease, and so more we get older and more the cancer incidence increases. However, the link between aging and aggressive cancer is not clearly defined, except in one type of cancer, thyroid cancer. Thyroid cancer is the only cancer where clinicians have established standardized prognosis marker using age as a marker component, because older patients, older than 55, have a higher risk of cancer-specific deaths. However, it is not the case for every older thyroid cancer patient, and it is crucial to identify which all patients will present a high risk of poor outcome and aggressive tumors. The new identified by prognosis marker will assist clinicians to establish personalized and more accurate therapeutic strategies. First, for the high-risk patient with more aggressive therapies, and also for the low-risk patient with less aggressive therapies or active surveillance, where no surgery is proposed, but a regular controlled follow-up will check the evolution of these low-risk tumors. The active surveillance is the future of thyroid cancer, as a majority of these cancers are low risk, but consequences of surgery and treatment are high. So in order to discover this biomarker, we use the TCGA thyroid cancer and the GTEx thyroid normal tissue datasets. And different gene expression analysis were performed with a DEC2R package, so using the R software, in order to select which genes were the most significantly differentially expressed in the supposed aggressive aging cancer population. So we perform different comparison analysis between aging cancer with aging normal tissue, young cancer, young normal tissue, aging normal and young normal tissue, in order to select specifically the aging cancer gene. And we found 198. From this first selection, other DEG analyses were performed comparing the clinical and survival parameters, and we filter the 198 genes on 72 cancer aging specific, but also aggressive genes. As we focus on an aggressive phenotype associated with the aging process, the same analysis, but adjusting the parameters with age, were next performed, and the 72 were filtered as 48 cancer aging specific and aging dependent aggressive genes. Finally, as internal validation, the same analysis were performed only on the aging cohort to be sure that the gene selected continue to be associated with an aggressive phenotype in this aging cohort. And we finally define 23 genes that are cancer aging specific and aging dependent aggressive. A score computing the expression value and the beta coefficient of regression analysis of these 23 genes were calculated. This score validated the strong association of the 23 gene signature with aggressive phenotype and also with the poor outcome. The score powers to predict the aggressive phenotype of this poor outcome were estimated with a receiver operative curve analysis. The interpretation of the curve is the following. The area under curve AUC. The closest is to 1, and the strongest the predictive power to classify is. And so as you can see on this curve, the 23 DEG panel presented a very strong AUC, superior to 0.7 for most of the clinical parameter analyzed. Hence, this 23 DEG signature predict aggressive tumor phenotype in the all thyroid cancer patient. We went further and we compare this 23 panel with abilities to identify the high-risk patient with the already standardized clinical markers that are used in clinic today. And so we observed that actually the 23 DSG panel presented a significant and strongest prediction that the AGE system, the AGCC system, when we were predicting the overall survival, but also against the ATA risk classification that predict for recurrence with a disease-free survival and progression-free interval. Hence, this 23 gene signature is a potential prognosis marker for aging thyroid carcinoma and also can be used complementary to the standardized prognosis markers. Finally, through a principal component analysis and hierarchical clustering, two subclusters of this aging cancer population could be identified: the less aggressive aging cluster and the more aggressive aging cluster. So we have discovered a 23 DSG aging panel that predict which all patients will present a poor outcome and aggressive phenotype. Validation in other databases and also in human subjects will be needed, of course. To conclude, this panel may lead to a more accurate personalized therapy and a better understanding of how aging aggravates cancer progression. So the second project that I want to share with you is related to the cancer process, the resistant process after BRAF inhibitor treatment in melanoma patients. So more than 60% of melanoma and also in thyroid cancer present a mutation of the kinase BRAF. The main mutation found is the V600E with the valine, the 600 amino acid of BRAF protein is replaced by glutamic acid. The mutation leads to a change in the protein conformation, making the kinase pocket being overactivated. The overactivated kinase leads to an increase of proliferation and survival of the mutated cells, and so create and drive tumorigenesis. The overactivated kinase, so drugs were designed to be competitive substrate, especially on this mutated kinase. And so under BRAF V160E inhibitor treatment, melanoma shrinks and patient will be in remission. However, or unfortunately, most of the patients will relapse in the next 18 months after the end of the treatment because cancer cells will have induced resistant mechanism against the specific drugs. So it is crucial to discover new biomarkers as gene panel in order to identify which patients have a high risk of recurrence. The new biomarker will help clinicians to adjust the therapeutic strategy by proposing a combination of treatment with the BRAF inhibitors and drugs preventing the resistant process, or by choosing another frontline therapy as a immunotherapy. So the project began at the bench where our team was looking to understand the resistant mechanism after a BRAF inhibitor treatment, because these mechanisms are quite still unclear. So we performed an in vitro melanoma model of resistance. We used, an in vitro melanoma model of resistance, and we performed a proteomic assay. Among more than the 80 different proteins, 18 were differentially expressed in the resistant samples compared to the sensitive one. Next, we were interested to see how these proteins were behaving in human melanoma resistant models. In our knowledge, there were not protein data available online to answer our question, so we decided to look for transcriptomic data. In the GSC dataset repository, we found 5 datasets of human melanoma samples that were treated with BRAF inhibitors and present resistance. We focused on the first, the GSC 19898, has a discovery panel, and we validate our results in the other datasets. So first, we checked the differential expression of the 18 genes according to the resistance stage, and we saw that most were deregulated, but few presented a significant difference. However, when we ran a principal component analysis in order to estimate the importance of the contribution of the actin gene to explain the heterogeneity observed across the samples, we saw that the actin gene could significantly differentiate the samples according to their resistance stage. With the pink samples represent the sensitive one and are in the left of the plot, whereas the blue samples that represent the resistance samples are the right of the plot. Furthermore, the genes presented a variety of correlation, as we see on the right plot, but also a variety of contribution to the PCR, with the red one having a high contribution, whereas the blue one having a lower contribution. And so, as we observed that all the actin genes didn't have the same contribution to the resistance discrimination, we decide to filter them and select a signature that presents the strongest predictive power to stratify the sample, according to the resistance stage. To this aim, we use the supervised machine learning linear discriminant analysis methodology for every combination possible between the actin genes. So 260, 200,000, 153 possibilities. All these combinations were tested, and the criteria of selection used was a ROC analysis with the estimation of the area under curve of the AUC. And so the combination presenting the highest AUC have been chosen. It was a combination of actin gene, and this actin gene signature presents a discriminatory range of 0.98 as a AUC in the discovery dataset. Linear regressions and other statistical tests shown strong significant p-values, and other datasets used as validation tests presented similar results. Finally, a merge of the 5 datasets in order to obtain a dataset that have a high number of samples were built with the computation of the corresponding z-score for the individual previous datasets. And this merge shown also a very strong discriminative power of this 13 gene signature to predict resistance in human melanoma samples. The ROC curves illustrate the table, the previous table with the 6 analyses, the 5 dataset and the merge of the dataset. And we observe that all of them have an AUC superior to 0.7. The actin gene score has previously for the actin gene were also differentially expressed according to the resistance stage in all the datasets. And through a PCA, we could see that the actin gene explains the difference observed across the samples because of the resistance process. So we have discovered a 13 GEG resistant panel that predict which melanoma patient will relapse after BRAF V600E inhibitor treatment. As previously said, validation in other datasets and human subjects are needed. To conclude, for this project, this potential therapeutic marker might help clinicians to accurately establish a personalized therapy. This panel analysis will improve the understanding of drug resistance mechanisms. And so today, I work full-time as a bioinformatician, finish bench work, and I work at the vet school of LSU at Baton Rouge with Dr. Kousoulas. Being part of the, I am part of the LBRN as the assistant director of the Bioinformatics, Biostatistics, and Computational Biology core. And I will be the liaison between the LBRN community and the Pan Biotech. It will be my pleasure to collaborate and help and mentor you for your bioinformatics or biostatistical needs. Thank you.
You're on mute there, Grant.
There we go. Thank you, Emmanuel. We appreciate all of that information. we have learned so much from everybody today, and you're working in a changing world with changing data structures, and it must be a huge challenge. Thank you for delving deeper and giving us more understanding in what you're doing. I'm going to ask you a few questions that we've got in, and I'm going to get you a start here. What are the challenges in bioinformatics in relevance to personalized medicine?
On my experience, what I observed is there is the wish of clinician and researcher is to find the most optimized signature marker that can be used for the health problematic. However, bioinformatics have a diverse methodology to answer this question. And there is no one that is better than the other. So the challenge for me was how to choose the best methodology and how to justify the why I have chosen this methodology and not the other one. That was for
thank you.
A challenge that you haven't resolved.
What prepared you for this position, and what do you think other students might learn and master as they embark on their own bioinformatics analysis?
What was the first part of the question? Sorry.
What prepared you for the position?
What prepared me? During all the years, every time I have a new question, a new necessity on bioinformatics, I have to learn it. So it's a learning process. What prepared me for this position is during this my postdoc particularly, I had the opportunity to collaborate with different persons that present me in the end, Dr. Kousoulas. And I think the fact that I have this background in molecular biology, in science research, where I understand the bench work, I understand the molecular process that we are trying to understand, it's an advantage to help by researcher or student for a project, because I have this balance between the molecular biology and biology context, but also know which kind of bioinformatic tools are available to try to answer the different questions. And furthermore, also when you have this balance, it's maybe easier also to ask biological questions. I think sometimes what I heard when bioinformaticians were talking is it's difficult for them to have a clear question of what they need to answer with their tools. And so having this two expertise in a way help to have a clear question to be answered.
Very good, thank you. So what do you think, we have a lot of students on our calls, I know, what do you think students might want to learn and master as they embark on their bioinformatics analysis? What kind of advice do you have?
I think it would be interesting for them to at least maybe beginning to master some free software as maybe R, that is very easy, the ones that I use, on very basic statistic and bioinformatic tools, has correlation analysis that can be used for different field on research, can be master, beginning to understand also statistic that can be used in very diverse field and are not very much specific for molecular biology or immunology or this or other things. So yeah, it can be helpful to have a kind of basic understanding of the bioinformatics tools that are available.
Very good. Thank you. We're down under a minute for our this session, so we're going to go ahead and close this, but we're going to head towards our panel. So just hang in there, if you would, while we gather our panelists again. For those of you that are on the panel, if you could go ahead and join back in, we would be most grateful. I thought the audience might be interested in our statistics from our 2 days. I'm going to put this in chat just so you can all kind of absorb what we've had here. We have had more than 1,000 users registered for the event, 8 speakers from India, 5 different speakers from Africa, and 14 from the Americas. So, man, everybody's been mentioning diversity here and there. That's a lot of diversity. So we really appreciate that. Also, we've got one more reminder about the learn.omicslogic.com for your free learning portal account. It's free. Sets you on a path to learning. So if you haven't done yet, I'm going to put the link back in chat one more time as we head towards our panel, and you can get that before we close. Over the last 2 days, we've had amazing presentations, and today we covered diverse topics from cancer, forensics, space to beauty, drug discovery to Bayesian hierarchical phylogenetic trees. And what brings us all together is our appreciation of the increasing role that bioinformatics is playing in biomedical research and the need for high quality bioinformatics education, mentorship, and opportunities for students to participate in research. So thank you all for all of this. And we want to thank our speakers once again from today. We will be sharing the recorded sessions on our YouTube channel. And since I seem to be really good at plugging in links to YouTube and all that, there is the link to our postings that we will have. And let's review our esteemed panel. We have Dr. April Wright, Dr. Emmanuel Ruiz, and Ilya Brodsky, who's stayed on with us. And panel, we want to dig in on how students, this is all about the students, how students can learn about bioinformatics and advance biomedical research across the NEPI regions. So who wants to start with tidy data and what it means to us all?
Yeah, I can start. I think there's often a lot of hesitance to try and get students to do any sort of bioinformatics or computational research. You know, people think, oh, it's going to be really hard. The students aren't going to understand it. And, you know, I'm not saying that it's going to be easy, but I use R in my introductory biology class, and we do some, you know, basic linear regressions to do things like understand organismal scaling or think about how mutations move in a population. And, you know, most students are game to try things. Like that's what they're really good at, trying stuff. And so I think, you know, if we think it's important to get students involved in learning these sorts of skills and to get them to think quantitatively consistently, you know, we're the ones at the front of the classroom. So, you know, there's no one else who's going to do that if we're not going to do that.
Okay, fascinating. Thank you. Emmanuel?
Yeah, I think is as a student, the possibility to learn the basic tools of bioinformatics, I think is quite crucial and interesting in a way that first I think it will, how says that, illustrate all the theory that is learned on mathematics, statistics courses in university, and it gives a wide examples of how what we learned in classes can be used and be useful for very global topics, and I think it can help them practice this theory that they learned, and yeah, we need to try. I mean, we need to students need it's easy. I mean, it doesn't need a big brain to begin to learn bioinformatics. It's, as I say, and with the Omics logic tutorials that help very much, it's quite easy to learn with internet and how to use the basic of bioinformatics, and with very simple examples of how it can be useful. It's quite interesting to learn. And
Thank you.
Sorry, I think it's also very much important because more today, we know with all this media crazy stuff, learning bioinformatics, biostatistics help to see that all the information maybe are not very significant, but we need some tools to select what can be significant and can be kept after. So it's a kind of also a kind of mindset that is learned to be more, how to say that, more, I don't know how to say that, but more respect.
I'd like to help you, but I don't want to put words in your mouth.
You can have like that, I don't see it. So yeah. Yeah, more take a step back of what we think, and before to confirm and give a big conclusion on anything, we can use tools that help to say, oh, maybe not, it was not true. It may be, but in the end, it's not true. So we have to be careful. And bioinformatics help to teach this kind of mindset.
Okay. Ilya, I'm going to let you kind of close that up if you want to say anything more about it, and then talk about how students and diversity of experiences. Lead that off too.
Well, I think here we have a great diversity of experience, right? April was saying how she did, a degree in biology and then went on to journalism and ended up doing what she's doing now. So it's a very non-linear path from, where you start, where you end up. And we had a lot of speakers that illustrate that. I mean, people that, maybe started in a completely unrelated space, end up doing bioinformatics. Now, what does that mean for students? I wonder if it is easier for them to see how bioinformatics can be a useful and not an intimidating skill set if they are exposed to it early on, and not necessarily only after they said, I will, master bioinformatics and become proficient at it, right? How can they do that? How can they just try something small, in an elective course or maybe a mandatory course, but not with a high stakes, not like you either succeed or fail, there's a lot of in between. And maybe another thing is diversity from the perspective of this kind of a community, right? I think when you are just like 3 people that do something, it's also very difficult, right? You got to see that there is a greater population of people in different regions, in different universities at different levels that maybe use this in a variety of ways. So I think that it's very important to bring that community together and let students see each other and kind of realize that, it's not just one single type of people that end up doing this.
And one of the things I've heard over the last 2 days is how diverse the paths have been. So yeah, April, I'm sorry, go ahead.
Oh yeah, I was just going to say, I think one thing that is worth remembering is that, when it comes to who can see themselves doing computation, almost every high school offers biology, right? A much smaller proportion of schools are going to offer advanced mathematics, computer science, these sorts of things. And so I was writing a grant last year and I came across this stat that, 90-something% of students who major in computer science had it in high school. And I was like, wow, that's incredible. And then I realized, well, probably 100% of students who major in biology probably had biology in high school, right? And so if we restrict this sort of information and this sort of training to people who have already declared an interest in it, we're basically restricting this sort of information and this sort of training by zip code in a lot of places. And so I think, that integration, like Ilya mentioned, in lower divisions, that integration in coursework where it might not necessarily be, the bioinformatics course, but it's something that everybody has to take, I think is probably a good thing if we believe all students should have access to these skills.
Any expansion on that, Emmanuel?
Yeah, no, I don't know, but yeah, it's true. It's true that bioinformatics is so diverse that it will, even for the undergraduate students that still don't know exactly how they want to evolve in their career and how they want to specialize, they will have tools that can help them very easily, because bioinformatics have it's a very, very diverse. And these tools also can be used for other, the same tools can be used for other fields, as finance, business, or other activities. So it can be quite very useful. And yeah, and we are, I imagine, because it's a long time I have been a student, but we try to be in the top of technologies, and right now biology have been is so high that in the technology are very high tech, and so it would be very interesting for the students to have an access about how this new biology are because of bioinformatics in part. So it's yeah.
So one of the things I could imagine, a student having a hard time asking questions and going beyond the guide direction and going self-directed and coming up with creative thinking. If you don't know how to ask questions, how do you ask questions? Can you guys talk about that a bit? Ilya?
Well, I think that's kind of what's missing in bioinformatics, right? Bioinformatics is almost viewed as this like set of skills, do you know how to code? Do you know how to do this? Do you know how to do that? So if somebody gives you a coding problem and they explain to you, this is what you need to do, this is kind of the result I'm looking for, it should look like this. I think, yeah, anyone can follow that. It's much more difficult, I think, for students to feel like they can be creative and kind of thinking beyond those kinds of instructions, and even sometimes spot what they are finding, what is interesting about their finding. Because ultimately as a student, you do a lot of digging, right? And a lot of digging and reworking your problems is because you don't get them right. But as sometimes you kind of stumble upon something that is interesting, right? And then sometimes your expectation might be, well, maybe that was another mistake, but it's not always like that, right? And so you have to almost have that anticipation that every once in a while, you're going to stumble across something that is interesting. And when you do, you need to have, I think, a good way to explain what it is, communicate it to somebody who's more experienced than you, and have that discussion of equals almost, like, this is what I see in data. What do you see? How is this like the correct way to interpret that? So I think that's a skill that you cannot master by, reading a textbook. You almost have to be mentored, be shown an example that, yes, we all make mistakes. We all stumble across interesting findings. And that's what papers are, is like, we found an interesting finding. Now let's try to describe it and validate that it's a real thing.
That's interesting that you mentioned the aspect of digging, because, one of the things I feel like we teach kind of poorly is how to read scientific literature, right? How to pick up the narrative and how to understand it. And so I do some domain-based education research, and I have, one class where we're looking at kind of more straight lecture versus if we incorporate more like learning how to read a scientific paper and then doing sort of Socratic method discussions. And I'm really interested to see how these data come out, because I feel like, one of the most common things that I see in students is not that they come into research and they want to do things, and it's not that they're not bright or they're not, intelligent people. It's that they stumble with the independence, right? And it's like, you learn to be a good writer by reading a lot, right? But when you learn to be a good scientist, you do that by sciencing a lot. But how do you do that in the undergraduate curriculum in a way that, we're constantly shuffling them around, right? You're going to chem lab and you're not a chemist, so you have a lot of instructions. If you're not a physicist, you have a physics lab and a lot of instructions. And there's not a lot of room for independent exploration in a lot of these. And so, having them read the literature and see how other people have done independent exploration, I think is really important. That's a good point.
So, Ilya, you kind of talked about something about in the classroom and stuff like that. How much of that stuff, how much of a responsibility do we put on a mentor or a teacher to make sure a student understands it's okay to be wrong, it's okay to walk down the wrong path, it's okay to ask questions?
Well, I'm just going to incorporate this message that I just see in chat from Dr. Amitabh Jayaswal, who's saying, as a bioinformatics learner, my difficulty is that I find it hard to understand concepts of computer science and statistics, although I understand biology fairly well, but those things instill loss of confidence and consequently productivity. Also, when I approach some professors or practitioners of the subject, even they don't truly appreciate the depths. Doing a series of clicks or following an algorithm does not impart real understanding hold. So this disenchant. So I guess what this message means to me is that whether, Dr. Jayaswal here is not a student, right? He's already an established maybe researcher and learning bioinformatics, right? So I think if you go into any new domain, you're going to be following somebody else who's saying, this is pretty much the formula of how you get from A to B, right? And I want you to be at B, and at B, something else happens, right? You get a degree. Now, we call you a doctor or whatever it might be, right? So I think this kind of a view of research, if you want to be in research, that's kind of like a parallel world that you're living in, because in research, one of the more important things is getting to a point where you stop relying as much on other people that tell you this is how you do things, because otherwise you find it difficult to kind of get away from established truths, which is what you're going to end up challenging in most of your research. So I think that it's something that is much more advanced in the West than maybe in some other more traditional systems of education. So that does exist. On the other hand, I think that the effort to get people faster to this higher level where they can become more independent could have this downside that people view this as restrictive. So I think that's where you have to kind of leverage the abundance of resources that are available to, skill, understand, ask questions about how things work and get to that point. But, it's not something that I think you can get from that formal education. The goal of formal education is probably not that.
And also, there is something that I realized on my own experience is sometimes you don't have results, or you have results that you are not able to explain. That can be in biology, but also in bioinformatics. But it's not because they are wrong. It's just maybe they are largely more complex than what we saw initially. And so we need maybe more time, or maybe someone else at one point will understand it. And so you need to be to lower a bit the pressure of the results and say, okay, maybe it's more complex than what I saw, but it's not wrong per se. Is that something that it can help and not be completely discouraged in the research world?
Anything to add, April?
No, I have to run and get my kids from school though. So thank you for having me. It was really nice to be here.
Well, thank you for being here yet again. So
nice to meet you, Emmanuel, as well.
Nice to meet you too.
Thank you. Emmanuel and Ilya, what's coding in R and Python, computational training? Are those going together? How important are those things? I've heard those names throughout the last 2 days.
I guess I want to also ask, what Emmanuel thinks about it. I started learning R first, and I really thought, this is so great. You can just produce these charts right away. They look nice. You can just put them in publications. It was really kind of like an exciting, simple way to get quickly to meaningful results. In Python, just using matplotlib with all the loops and trying to get your chart to, display different colors and annotations, that was such a pain. until I realized that Python maybe is much easier, in fact, to use in other domains, like machine learning, for example. I find it very easy and kind of intuitive the way that those packages have been built. So I think that there are benefits to, either R or Python in different applications, but it is dependent on kind of where do you see yourself going. Like if all you need from R is to get that plot and everybody's using that complex heat map to produce that nice figure, maybe that's enough. But if you are trying to go much further beyond that, build your own application, start kind of working with other types of repositories here and there, that's, I think, where you have to know enough of both to not be afraid and to also be able to kind of specialize eventually down the road. But I would be interested to know what Emmanuel thinks about that.
But me, it's simple. I'm still not in Python. I am in R. I imagine I am familiar now with R, and I still haven't found on my application the limits of R where I couldn't use R. But yeah, so for me, R was simple. I will say that I found the tutorials that let me understand R in a simple way and going step by step. And so it's very useful, as you say, for plots, for simple analysis. And like that, in my head, to be honest, I don't see publications that show Python, but most of them show that they have done their analysis with R. So I think it's very useful. But as you say, we don't have to be closed on one software, but see how others can be also helpful. And with the Omics logic tutorials, I'm going to begin to learn Python. But
nice.
New language. I didn't want to do both at the same time, even if a bioinformatician told me you need it. But I was a bit afraid that if I learn both at the same time, I will mix them and I will don't succeed to improve my skills. So I stay on R. Yeah.
Nice. So let's switch a little bit of this over to the social and emotional aspect of research, especially as it applies to students. Now, I've heard in my world here how many people throughout the last 2 days have stumbled onto something that became important in their lives, but how much impact do students have into research, and then what happens in our world?
I understand this topic to be extremely important from the aspect of research being very discouraging sometimes, right? So I think that research is a very long game. It's not something that you achieve results quickly. And it's something that you're going to fail a lot more than maybe if your goal was, much less ambitious. So it's definitely for people that have the perseverance and the emotional strength, I would say, or ability to, disconnect from their emotions to go over, overcome those challenges. So I think that's one part of it. The social part of it is that being a scientist, you wish you were the person on TEDx talks, discovering, things that change the world, but most of the time you're not, right? So most of the time you're kind of behind the scenes and your PI is who's out there talking about the discoveries made in the lab. So I think that, if you choose this area of research, and I think that's where bioinformatics actually is going through sort of a transformation. My father is a bioinformatician and has been for decades, and so he was always the one providing data to people who went on stage and talked about the discoveries. And I kind of saw this disconnect between the hard effort that he was putting into his analysis work and whatever the biologist was then presenting. I think today that is becoming much more balanced. Bioinformaticians, there was an article a few years ago that bioinformaticians can finally be in the driving seat. I think that is a little bit of a change and an encouraging change for those that have chosen bioinformatics as their science.
Yeah, I agree with you that the best quality of researcher are perseverance, because that's you will need it. I mean, because It's maybe less in bioinformatics, maybe more on bench work, but you spend most of your time having negative results before to have one positive result. And it's not something that is easy to manage, so you need to have perseverance. And so I think there is one thing that a skill that need to be learned, and it's long process because it's more a kind of a routine to know how to separate your professional life to your personal life, because research doesn't have end. And it's like when we do a big puzzle, we don't want to stop until it's finished. But at one point, we need to have also a healthy life and a healthy separation between what is research or professional life and personal. And that, I think, honestly, is a skill that need to be learned, because it's not easy, it's not innate. And more when we are students and we are working for PI, PhD, postdoc, when we are working for people that put us a pressure, it's because they have their own pressure, okay? It's not easy for them too. It's difficult to put this separation. And so maybe learning these two skills, perseverance and separate professional to personal life, is very helpful to have a success in research. And yeah, I think that, as you say, not every researcher will have the Nobel Prize, but it gives us a kind of humility and a kind of encouragement to know that for a Nobel Prize, there was maybe thousands of manuscripts that this Nobel Prize have read, tried to understand, used to his own project that have this reward. So in a way, all work participate to the whole science evolution. And that is, it's a beautiful feeling.
Fascinating. So you guys touched on something, and I want to kind of go back to it. For students and doing all this research, is there an issue with expectation of result? And I think you kind of walked around a bit, hit it a little harder, because I'm a new student. I know I'm going to find this, and then it's not even close.
Well, I think actually one of the speakers, Dr. Paulson, mentioned that they involve students. It's almost like as a PI, you have to have this, maybe decades or years long project. And if you are able to involve students at different stages of this project, you end up sharing these exciting milestones with them that they feel like they've gotten to a result. So it is, a lot of it is about mentorship with an intent to offer students, not squeeze work out of them, but allow them to get into the field and feel like they are contributing something meaningful, right? So like, for example, in my experience, I've had a number of very established PIs that have included me in publications that, I honestly did not contribute maybe that much, but the fact that, my name ended up on that publication, gave you like, we published in Nature, whatever my contribution was, but, that was a very empowering feeling for me.
I understand. It's, yeah, we have similar experience, yeah, where we work hard. I know my PhD, my principal project have not been yet published. So it was quite hard, because when we spent 4 years working on it and by discovering new truths by yourself, it was quite hard. But what I have done, even it didn't get published as we wanted, it helped other teams to build their own their project, and so driving and discovering more about their science and the breast cancer at this time. So yeah, it's frustrating. Research is frustrating. It's not, you don't have to be blind and go there saying, oh, it's a happy place. It's a frustrating work, but yeah, knowing that even indirectly we participate to this knowledge evolution help to overcome these frustrations. And so to put the pressure lower, because unfortunately, the success of researchers are very dependent of what they publish. And so when you work years or months or years on projects that in the end, you didn't see any publication, it's quite hard. But there is other indirect way to have encouragement. So it's nice to remember that.
Yeah, very good. Well, thank you all. We are wrapping our time here as it comes to an end. Thank you all, especially Ilya and Dr. Emmanuel, for sticking around this afternoon and answering some questions, especially as students, because I've seen a lot of student questions in chat over the last 2 days. So thank you all. To those of you in the audience, please engage with us. I posted our social links a little bit ago, and if you can comment and share there, that would really help our mission. And of course, go sign up for your free account. That wraps up our 2 days of international worldwide learning. I'm just stunned by the level of intelligence and integrity with all the researchers and speakers. Thank you all. Thank you again, Emmanuel and Ilya. And that wraps us up. So thank you all so very much.